and food security across North Africa remind us that genomics is not a tool for discovery, but a bridge between science and policy, between data and people, and between biodiversity and health. We must never lose sight of our common objective, which is to responsibly and wisely use biodiversity genomics to better serve the communities from which biodiversity originates. Through this initiative, such as this one, AfricaBP, we unite universities, research centers, and industries to promote ethical, inclusive, and sustainable genomic science. Over the coming days of our workshop, we will explore how genomics, bioinformatics, and artificial intelligence enhance conservations and accelerate breeding for climate-resilient livestock and crops, and also illuminate the invisible threads linking the environment, the animals, and the human. We will learn from pioneer and emerging scientists during this workshop how Africa's growing capacity to generate world-class genomic data using local resources, original partners, and open science. This workshop is also a living example of One Health approach in action, where genomics informs not only agriculture, but biosafety, biosecurity, and ecosystem health. Our practical sessions across North African countries for this year, from Nextflow for RNA-seq and artificial intelligence, CRISPR-Cas9, NGS, and bioinformatics and marine bioinformatics hackathons, show that AfricaBP is not confined to theory. It's a movement of practice, mentorship, and empowerment, shaping the next generation of African scientists. To our students and also to our early career researchers, you are the reason we are here, because you carry the promise of an Africa that sequence its biodiversity, curate its data, and define its priorities. You are the future of Africa, and you are the guarantee of the independence of Africa at the political, economical levels. Let this workshop inspire you to innovate boldly, collaborate openly, and lead ethically. I extend my heartfelt gratitude to our organizing committees, partner institutions, speakers, sponsors, and volunteers. I'm equally grateful to every participant who traveled from near and far. All of you, you transform this workshop into genuine Pan-African dialogue. Welcome to Rabat. Welcome to the future of African biodiversity. Welcome to AfricaBP. Thank you. Now, I will give the floor to our Dean of the Faculty of Science at Mohammed V University in Rabat to welcome you for this event. Please, Professor Ragavigi, you have the floor.
Vous avez tout dit. I agree all you said. Distinguished guests, esteemed colleagues, dear participants, dear students, dear professors. It's a great honor for our Faculty of Science at Mohammed V University to host this African BioGenome Project Open Institute North Africa Regional Workshop 2025. This gathering reflects what science should be at its best: collaborative, open, and deeply connected to the realities of our continent. By bringing together researchers from across Africa, North Africa, and beyond, we affirm our shared conviction that knowledge must serve both nature and humanity. The theme of this workshop, biodiversity, genomics, and One Health, reminds us that health, environment, and food security are inseparable. Through genomics, bioinformatics, and responsible innovation, we are building the scientific foundations of a sustainable Africa. I would like to express my sincere appreciation to Professor Boabid Badawi and the entire organizing committee for their remarkable effort, and to our national and international partners for their continued trust. I wish you all a productive workshop filled with meaningful discussions, new collaborations, and ideas that will carry far beyond these walls. Welcome to the Faculty of Science. Welcome to Rabat, and thank you.
So thank you very much, Professor Ugagi, for welcoming our host and for supporting this event. Thank you very much. So now we will, okay. She is online? She is online?
Yes.
So
yes,
now we will give the floor to Professor Anne. Professor Al, please, could you hear us?
Yes, I can hear you. I
Okay.
hope you can hear me.
Yeah, she's online, Boabid.
Yeah, we can hear you. So please, you have the floor, Professor Anne. Go ahead, please.
Thank you very much. And thank you, the Dean of the Faculty, the Chair of the African BioGenome Project, North Africa, that is Professor Boabid, and all the organizing committee, the faculty members. My name is Professor Anne Wigai. I'm the chairperson of the African BioGenome Project. And I'm going to give a short presentation giving you a background of the African BioGenome Project. And this presentation will be given on behalf of myself, the entire African BioGenome Project, and my co-chair, Dr. Thangod Ebenezer. So the African BioGenome Project was conceptualized in 2019 and launched in 2021. And in March of 2022, we published our position paper in Nature. And in this paper, we gave our goal of sequencing 100,000 species of African biodiversity in 10 years. This caused a big interest around the world, and the paper was featured in over 300 news outlets. People were very, very excited to see this. Among the questions that were asked was whether it was worthwhile to spend over $1 billion over the next 10 years to sequence biodiversity when Africa has so many challenges, including the challenge of provision of clean water, food security, and all that. So I guess the question you would ask yourself is, why Africa? As Professor Boabid has said, Africa is home to some of the world's most diverse ecosystems. Many of these species are endemic. They can impact global science, and this can be very, very impactful in the area of medicine, in agriculture, and also in the areas of conservation. And we need to leverage on our indigenous biodiversity, because as Professor Boabid has highlighted, we have numerous plants and animals that are very, very unique and have various advantages that are only found on the continent. Some of these traits that are very unique include disease and pest resistance, drought tolerance, stress resistance, And many of these plants have not been fully characterized, so we don't know what genes they carry and how they can help us as we go forward in science. And we have to recognize that we're racing against time. We can wait for others to do it for us, but we can do it better if we do it for ourselves. This will have even more benefits towards ourselves as a continent. And this is where Africa BioGenome Project comes in. African BioGenome Project, which is also known as AfricaBP, is a large-scale initiative. It's a continental effort. It is led by African institutions and scientists, and we all bring in our expertise, be it in genomics, be it in bioinformatics, be it in taxonomy, to sequence and catalog the diversity and Africa's flora, fauna, and microbiome data. So our goals are to coordinate all non-human genome sequencing initiatives. We are not doing any human genome sequencing work because there are other groups on the continent that are doing this, and they're doing it very, very well. So we don't need to replicate this. We want to initiate, develop, and support any new efforts in sequencing non-human genome sequencing. And as I have mentioned, our goal is to sequence 100,000 species in 10 years. And then we want to use this information to help advance bioinformatics, genomics, the medical sciences, the human sciences, and to contribute to African regional biobanks. We also want to inform policy and in the process also build capacity in these specialties in Africa. So African conservation is one of the key points, and we recognize that the flora, fauna that is found in Africa is very, very diverse, but we have to recognize the threats that are coming by to threaten our ecosystems. That is climate change, the fact that we're continuing to destroy our habitats. We have illegal trade of animals and plants that is causing issues. We have issues of pollution, we have issues of overgrazing and agricultural expansion. So on one hand, we are trying to conserve the environment that we have, but we have another arm of ourselves where we are not conserving this effort. And therefore, we need to work very, very important. And genomic data can provide data-driven solutions. So we don't have to imagine what is the best thing to do. We sequence, we get the information, and then we advise policymakers what is the best way to move, because we'll already have the data to support this. We also want to understand what is climate change, what is happening in our ecosystems, what's happening in these species, what's happening in the environment. Only when we do this will we be able to react properly, because we will have the information that we need. And it's also important to impact how species are beginning to adapt to climate change. And therefore, this will also help us as we begin to effect new climate conservation strategies. One of the things AfricaBP has done and will continue to do is to forge better collaborations within Africa. We recognize that many of us in Africa have forged very, very strong partnerships with partners outside Africa, in North Africa, in Europe, and across the rest of the world. But within Africa, we've not done as well. We don't know each other, and we don't know who is where and what they do and what they're doing and what sort of infrastructure that they have. And if we don't have that information, we cannot learn from the best practices that they have. And therefore, AfricaBP is one of the forums where we're beginning to forge Africa partnerships. We learn what is being done across the world, and we run in to support this and also to showcase this as a best practice across the world. So our goal is to sequence 95% of the genomes in African institutions on African soil. And we recognize that this will generate an estimated more than 200,000 gigabases of DNA bases. And we will take a very long time if we are not cognizant of that, and if we don't address some of the gaps. What gaps have we been able to identify? We've been able to identify infrastructural gaps, and we've also been able to identify genomics and bioinformatics capacity gaps. By the way, as I scroll along, you will see photographs showcasing capacity building initiatives across the 3 years that we have worked in these regional workshops. So this is showing a practical that was conducted in Central Africa in 2023. So to address these infrastructural and capacity building gaps, we launched the AfricaBP Open Institute for Genomics and Bioinformatics. And this is, you could call it like our training arm. And we aim to train, equip, and build local capacity, institutional capacity with skills and capacities to drive biodiversity and genomics, so that when we have this genomic data, we have the people and the hands and the institutions to be able to analyze it and store it on the continent. So to do this, we need to develop early career scientists. These are the young, vibrant people who will be working in these particular areas. And we need skills in taxonomy, in genomics, bioinformatics, and computer science. And here you see a practical that was done in your region, in North Africa, in 2023. So how do we build African talent in Africa? First of all, we need to place them in African institutions. We need to be able to have the infrastructure in these African institutions. We need to be able to harness the skills for home-based capacity building. And only through this will we be able to have homegrown solutions. This also saves our young talent from moving to overseas countries to look for better jobs and to look for institutions that will be able to host them. And this now, then, retention of this talent in Africa is actually a good thing for Africa. It will build our economies, and it will also physically have an impact in conservation and biodiversity on the continent. So what are some of the milestones achieved? With this workshop that is being done in Northern Africa, this region, we will have successfully completed 3 years of regional workshop. We started in 2023, where we had 4 workshops. We continued through 2024, we had 4 workshops, and now with this conference, we complete our 4th workshop this year. So we have a hub in Nigeria where we do this same training. This is a picture from 2023. We have you in North Africa, where you do the same. South Africa, in Cape Town, we have a hub. And in East Africa, we have a hub in Nairobi that also takes care of the East and Central Africa region. So we've completed 2 days conference-style presentations, and this is normally followed by 3 days hands-on practicals. This is really important because, you know, we cannot only focus on the theoretical training. We also need to focus on the practical training of our young scientists. And here we showcase 3-day practicals that was done in the Inca Biotech Laboratory in Eastern Africa. Here we have, on the right-hand side, we have the practical workshop that was done in the separations lab in South Africa. And here we have the first CRISPR-Cas9 practical training workshop that was done at the African Genome Center at the University of Mohammed VI Polytechnic in your region in Morocco last year. And I'm happy to see that we also have a CRISPR-Cas9 practical workshop. So every year we register participants. In 2003, we had 3,700 workshop participants. 408 of these received practical hands-on training, molecular biology, genomics, and bioinformatics. And we are so delighted that there was a very, very high representation of ladies, and it was attended by students, faculty, scientists, and industry players. Of the 54 African member states, we had 49 countries represented. And this means that we have all of Africa covered, and we hope that we can get more from all the 54 African states participating. Every year, at the end of every workshop, we prepare a manuscript. This manuscript that came out last year in 2024 showcased the findings of what we had seen in the workshop that we carried out in 2023. The 2024 workshop manuscript has already been written up. It's been submitted, and it's coming out anytime now. And we are currently in preparation of the workshop findings that will be done at the end of this year, and this will come out next year. Why is this important? Because we need to share our best practices, but more importantly, we need to share our experience. What have we seen? What have we learned? So that others who are coming after us will be able to learn from us and also move very, very quickly ahead. In addition to this, we have established AfricaBP Fellows for Genomic Leaders, and this was launched in 2024. It means that we select some of the best young early career scientists who are established and linked to African institutions. When we put out a call, these young scientists apply for it, and we select the number depending on the call, and we are able to now follow through the hands-on training for a longer time. This was a fellowship that was done in 2024 in partnership with Carl Walls Institute for Genomic Biology in the University of Illinois in the USA. And it was also sponsored by the African Genomics Center. And we had 4 young African scientists. You can see Ishraq from North Africa was one of them. And they had an opportunity to spend 3 months in this workshop with travel between the 2 institutions. And they really, really learned a lot. The picture here, they're pictured with the editor-in-chief of Nature. In my young early career days, I wish I had met a high-ranking editor such as this, because meeting this has a huge impact, and it means that the quality of the publications that come out of these young early career scientists already begins to be very high from when they're very young. So I'd like to leave you with some food for thought from my co-chair, thank God Ebenezer, who says an African-led institution is not one where the primary investigator has a primary African affiliation, but rather where 100% of the research is performed on Africa's soil. Many of the research projects that we have conducted in Africa have actually been to ship samples outside Africa. And these samples, even after the work is done, the samples are completely lost. And therefore, we have to think and say whether that has really been a benefit for us. But when we have sequencing initiatives on the continent, we train young scientists. We are bringing project money into that institution. That institution is able to build infrastructure, and the samples that are collected are retained in this institution for future work. And I think that is a bigger benefit, and that's what Dr. Thangod is alluding to. We also have to think that when we're training early career scientists, we also need to think about the mechanisms of retaining them on the continent. We cannot just train young early career scientists and leave them, because we will lose them. So when we train them, we also need to have well-paying positions in African institutions, have the African institutions have good infrastructure where the scientists will be able to find career satisfaction, and then also give them opportunities for promotions to higher ranking positions as scientists, senior scientists, professors. This way they will be able to be retained in the African institution in Africa, and therefore they will continue this training of others, and therefore we as Africa stand to gain in more for the future. The important role of AfricaBP Open Institute Regional Workshop cannot be underestimated. We have seen huge numbers applying for the limited positions that we have. So for instance, of the 1,500 applicants that applied to North Africa, only a few of these, maybe 400 or 100 of these, will be able to have hands-on experience, which means that about, more than 900 are left out. And therefore, there are huge training needs, and this we need to address as we go along. And therefore, partnership with African institutions that can be able to help us train and retain these young African scientists will help us as we go along in the future. And then we also need to make sure that the curricula that are being taught in our institutions is current and in line with the industry developments in this area of bioinformatics and genomics, which changes very, very rapidly. And we need to consider removing barriers that are impeding the growth of science in Africa by going fully open access, fully open science, and providing access to training which is free, providing access to training in situ rather than sending a few, maybe 5 Africans overseas. We can be able to train more if that training is local. We need to think about publication fees to journals and how these are presenting barriers, and then access to conferences for young early career scientists where we charge registration fees, and these fees, we can sometimes see them to be a barrier. So we prevent scientists and young scientists also from attending these workshops. And then we need to have common curricula that can be addressed across the African continent. And I thank you very much. Please visit our website, the africanbiogenome.org, for more information. And congratulations to the Northern African Regional Workshop for this magnificent initiative that you have. And I wish you all the very, very best. Thank you very much.
Thank you very much, Professor Anne, for this nice presentation in which you highlighted the context of AfricaBP, the achievements and the perspectives. The main message that's come back in each presentation of AfricaBP is the sovereignty on AfricaBP. The main objective of AfricaBP is to make the African people sovereign of their land. We need African youngs that sequence their university, curate their data, and defines their priorities. And we don't want that others decide what should be done in Africa. Thank you very much, Anne, for this nice presentation. Thank you very much. I think now we will jump to the first session, genomics-driven One Health. And this session will be moderated by Professor Laila Sbabou. Please, you have the floor.
Good morning, everyone. I'm truly pleased and honored to moderate this session held as a part of the African Biotechnology, African BP Open Institute for Biodiversity, Genomics, and One Health. I would like to first warmly welcome all of you, those joining us here in person, and those that are connected online from across the world. Your presence today reflects our shared commitment to advancing genomics and strengthening the One Health approach across Africa and beyond. Today, we explore how genomic science is reshaping the One Health paradigm, connecting animal, human, and environmental health through a shared biological language written in our genomes. Our first session is very inspiring as it is led by 4 outstanding Moroccan women scientists, leaders who have each built bridges between disciplines, bridges between research and policy, and between Morocco and the global community scientists. Let me first introduce our first speaker, Professor Wafa Fassi-Fihri. Maybe I'll ask the 4 speakers, Professor Wafa Fassi-Fihri, Professor Bushra Al-Qadi, to join us here, and Dr. Zakia, please join. And I think Dr. Alilush will be online. Our first speaker, thank you for joining, ladies. Our first speaker is Professor Wafa Fassi-Fihri. She's a veterinary virologist, one of the Morocco leading figures in Morocco in viral infectious diseases. She's the former director of the DIV Hassan II, the co-founder and the former president of the One Health Moroccan Association. We're glad to have you today. Thank you for joining, and the floor is yours, Professor.
Bonjour tout le monde. Alors, je vais bien sûr m'exprimer en français, mais ne vous en faites pas, mes slides sont en anglais. Donc, je voudrais tout d'abord remercier Professeur Bedaoui de m'avoir invité à introduire le One Health qui dure depuis presque 20 ans maintenant qu'on en parle. Et bien sûr, ça a évolué au cours du temps dans l'évolution de ce concept. Alors, bien sûr, je ne peux pas parler du One Health sans parler des 4 organismes qui sont impliqués sur ce concept, la FAO, bien sûr, l'OMS, l'Organisation mondiale de la santé animale, ex-OIE, donc elle vient de changer son nom il y a 3 années. Et le 4e organisme, c'est le programme des Nations Unies pour l'environnement qui a rejoint en 2021 donc cette tripartite qui est maintenant devenue une quadripartite. Alors, tous ces éléments, ces notions et ces experts ont mis leur expertise en commun pour, bien sûr, placer ce One Health dans tous les domaines pour une bonne santé humaine, animale et environnementale. Alors, ils ont également mis en place un One Health High Level Expert Panel qu'on appelle OLEP et qui ont donné la dernière définition de ce concept. Donc, une approche intégrée fédératrice qui équilibre et optimise durablement la santé des hommes, des animaux et des écosystèmes. Ajouter à cela la santé des humains, des animaux domestiques et sauvages, des plantes et de l'environnement au sens large, y compris les écosystèmes, sont étroitement liés et interdépendants. Et enfin, le One Health mobilise de multiples secteurs, disciplines et communautés à différents niveaux de la société pour travailler ensemble. Et je vais beaucoup insister au travailler ensemble. Favoriser le bien-être et la lutte contre les menaces pour la santé et les écosystèmes tout en répondant aux besoins collectifs d'eau, d'énergie, d'air propre, d'aliments sains et nutritifs, d'agir sur le changement climatique et contribuer au développement durable. Alors ça, c'est la dernière définition, donc 2021, bien sûr, où il y a eu la création de ce panel d'experts pour le One Health. Alors, bien sûr, j'ai dit que j'allais revenir sur travailler ensemble. Donc, bien sûr, tous les acteurs, et tout ce qui est santé humaine, animale, environnementale, écologue, sociologue, économiste, agronome, etc., doivent travailler ensemble pour appliquer donc ce One Health. Et comment ils doivent l'appliquer? C'est en coordonnant donc les actions, en communiquant entre eux et en collaborant entre eux. Vous savez, on a l'habitude de travailler depuis des décennies en silo, donc chacun dans son coin traite son travail et sa spécialité sans communiquer donc les données de sa recherche. Et bien sûr, tout cela peut contribuer à une meilleure santé animale humaine et de nos plantes et de notre environnement. Alors, pourquoi ce One Health est important? D'abord, on va penser à l'augmentation spectaculaire de la démographie mondiale et bien sûr, avec une connexion étroite avec les animaux, de plus en plus, les animaux sont proches de l'homme. Et ensuite, il y a le changement environnementaux, donc avec bien sûr les changements climatiques et l'usage des terres, avec tout ce qui est catastrophes naturelles, incendies, déforestation, etc. Je vais en parler plus en détail après. Bien sûr, tout ce qui est voyages internationaux et commerces internationaux. Maintenant, on traverse la Terre dans toutes ces directions, d'ouest en est et du nord au sud. Et bien sûr, les animaux ne veulent pas seulement réclamer à manger, mais ils ont besoin de beaucoup plus que ça. Alors, tous ces facteurs, bien sûr, réunis, font exploser des maladies qu'on appelle les maladies émergentes. Et vous allez voir que sur un autre slide que tout ce qui fait le cocktail pour avoir une pathologie, une maladie émergente, et ça va de plus en plus rapidement avec ces changements globaux. Alors, je dois parler de ces sciences et ces sciences multifactorielles donc, qui sont impliquées dans le One Health, avec santé au centre, la santé humaine, animale et de l'environnement. Et vous voyez toutes les disciplines qui doivent travailler ensemble et être impliquées pour pouvoir lutter contre les problématiques du changement climatique, des maladies émergentes et de tout ce qui nous entoure actuellement. Alors, il y a la médecine humaine, la santé publique, les sciences sociales, dans l'environnement, l'urbanisation, les cycles et réservoirs des maladies, l'écologie, l'écotoxicologie. Le cadre légal également est impliqué, la pratique des cultures, la relation entre l'homme et les animaux, la domestication. On voit toujours ce qu'on appelle les nouveaux animaux de compagnie. Maintenant, il n'y a plus que le chien et le chat. Il y a d'autres animaux de compagnie qui habitent avec l'homme. La médecine vétérinaire, les zoonoses et les épizooties. Et j'ai vu qu'il y avait des présentations sur les maladies infectieuses. Je ne peux pas oublier la résistance antimicrobienne et bien sûr les maladies non transmissibles. Alors, vous voyez qu'il y a plusieurs disciplines qui interréagissent entre elles pour le One Health. Alors, j'aimerais que vous vous concentriez au schéma du bas. Donc, quel est le cocktail qui fait qu'on doit appliquer ce concept? Alors, on est à 8 milliards d'êtres humains sur la Terre, 20 milliards d'animaux d'élevage dans le monde, 100 millions d'hectares de forêt coupés depuis 20 ans. Ensuite, un tiers de la Terre a connu une utilisation des sols, un changement d'utilisation en 60 ans. Plus de 20 mégapoles sont au niveau du milieu intertropical. On a 10 puissance 31 espèces de virus, 4 milliards de voyageurs par an, 57 000 navires de commerce en activité, 75% des pathogènes humains sont transmis par les animaux et 400 millions de personnes sont infectées donc par des pathogènes impliquant des rongeurs. Donc, voilà le cocktail explosif qui fait qu'on doit utiliser ce concept, travailler ensemble pour pouvoir lutter contre cette situation. Alors ici, c'est un travail qui a été effectué sur 20 ans, 2 fois 20 ans, la première de 80 à 99 et le deuxième de 2000 à 2019. Et vous voyez toutes les catastrophes auxquelles on est confronté, la sécheresse, les séismes, les températures élevées, les inondations, les glissements de terrain, l'érosion, les ouragans, les activités volcaniques et les feux, les incendies au niveau des forêts. Et vous voyez que entre les deux décades 80-99 et les deux dernières 2000-2019, il y a une nette augmentation des catastrophes naturelles, surtout au niveau des inondations. Et bien sûr, on voit que pratiquement c'est multiplié par 2 pour les inondations, également pour les ouragans et donc tout ce qui est storm qui est multiplié par 2. Alors, on va voir qu'on a beaucoup également travaillé sur la population et l'économie. Et vous voyez que les désastres reportés entre les 80-99 et 2000-2019, pratiquement tous les désastres reportés sont multipliés par 2. La mortalité humaine également, la population affectée en billions également. Et à la fin, les pertes économiques sont pratiquement multipliées également par 2 entre les 80-99 et 2000-2019. Voilà l'état des lieux. Alors, je vais zoomer un peu sur la biodiversité parce que le parent pauvre du One Health, santé humaine, animale et environnementale, c'est surtout l'environnemental qui est un peu à la traîne, donc qui doit rattraper donc, les 2 autres santé. Alors, je vais en parler un peu plus que les autres. Alors, comme vous voyez, le cœur de la biodiversité est impliqué dans plusieurs domaines, dans la pharmacie, dans la médecine traditionnelle, dans la santé mentale également, le développement durable, la sécurité alimentaire et hydrique, l'agriculture, la nutrition, les désastres, les risques de désastres, bien sûr la santé, les maladies infectieuses, le changement climatique, la qualité de l'air, de l'eau, et bien sûr également les écosystèmes et la diversité microbienne. Alors, ici, c'est un exemple sur les feux de forêt donc, en Amazonie. Donc, vous voyez l'image à droite qui représente les feux de forêt en Amazonie et à gauche, vous avez vue de l'espace. Donc, comment on voit donc ces feux de forêt amazoniennes et ici à gauche, comment dire, la pollution due à ces feux de forêt à Sao Paulo au Brésil. Donc, vous voyez l'air est irrespirable. Également, l'augmentation de la température au niveau de la surface de la Terre et au niveau de la surface des océans. Donc, une étude a démarré à partir de 1960 jusqu'à 2020 et on voit en bleu l'augmentation de la température de la surface de l'océan et en parallèle l'augmentation de la température à la surface de la Terre. Donc, il y a pratiquement 1 degré et demi d'augmentation de la température à la surface de la Terre et de l'océan. Alors, la déforestation, alors quand on déforeste, plus rien ne repousse. Et ça fait des altérations au niveau climatique avec l'augmentation des gaz à effet de serre, altération de la pluviométrie, de stockage de carbone, disparition des espèces et des habitats, la santé, bien sûr, augmentation des maladies respiratoires, cardiovasculaires, ophtalmiques et psychiatriques, et augmentation, bien sûr, des maladies vectorielles, puisque maintenant, les vecteurs qui transmettent la maladie, les pathogènes, on les voyait surtout en Afrique, mais maintenant, on les retrouve au nord, donc dans les pays européens. Également, il ne faut pas oublier les peuples autochtones qui vivent de ces forêts, de ces cultures locales. Et bien sûr, il y a de plus en plus de pauvreté à ce niveau-là. Donc, l'impact socio-économique et politique et une pauvreté, inégalité des peuples, des conflits de plus en plus au niveau des frontières et bien sûr des ressources naturelles, économie et emploi. Donc, les catastrophes naturelles causent des perturbations dans les activités économiques et la croissance également. Et les droits humains, bien sûr, avec ces peuples autochtones dont j'ai parlé, populations déplacées dans des situations de détresse et plus exposées au travail des enfants, au mariage précoce et d'autres formes d'exploitation. Alors, la disparition des espèces, vous voyez ici à notre gauche la disparition des coraux. C'est un schéma qui débute en 1990 et jusqu'en 2020. Donc, également, disparition plus lente des oiseaux, des mammifères et des amphibiens. Donc, le cri d'alarme des scientifiques depuis 1992, où le taux d'extinction s'est multiplié par 1000 par rapport au taux naturel. Disparition de 60% de la population des vertébrés en 40 ans et bien sûr la destruction de la population d'insectes. Et vous avez bien sûr l'exemple du rhinocéros blanc donc, qui a disparu. Le dernier est mort au Kenya, le dernier rhinocéros blanc. Alors, on a 38 500 espèces menacées d'extinction, 41% des espèces d'amphibiens, 14% des oiseaux, 37% des requins aérés, 28% des crustacés, 33% des récifs de corail, 34% des conifères, 26% des mammifères, et le varan de Komodo est menacé d'extinction également. C'est le grand lézard au niveau de la planète. Alors, un mot sur l'agrobiodiversité. Donc, au niveau mondial, nous avons 250 000 espèces de plantes et nous utilisons que 7 000. Et sur ces 7 000, ce qui prend la plus grande surface, c'est le riz, le maïs, le blé, et qui donne plus de 50% des calories à partir des plantes. Bien sûr, sur ces plantes, nous avons 12 cultures qui prennent la plus grosse partie. On consomme 5 espèces animales, ce qui fait 75% de l'énergie totale mondiale. Donc, l'agriculture, le rôle de l'agriculture, c'est 1/3 des terres, utiliser 3/4 des ressources en eau et 20 à 30% des gaz à effet de serre. Alors, si on voit un peu cet impact sur la santé humaine, nous avons un retard de croissance chez les enfants au nombre de 150 millions. Des enfants qui sont en détresse alimentaire, 52 millions. L'obésité chez l'enfant, c'est 41 millions. Et 2 billions d'adultes en surpoids et en obésité. Alors, avant de conclure, j'aimerais donner ces éléments. 75% des zoonoses émergentes proviennent de la faune sauvage. Donc, quand on déforeste, on a de plus en plus d'animaux sauvages qui sont à proximité de l'homme. Une nouvelle maladie émerge tous les 10 ans en 1970, contre 1 à 5 par an depuis 2000. Chaque année, les zoonoses provoquent 2,4 milliards de malades et 2,2 millions de morts. Donc, ces organismes internationaux, donc la quadripartite, se sont réunis pour lancer des domaines d'action rapides. Donc, le domaine 1, c'est augmenter la capacité et renforcer les systèmes de santé. Domaine 2, réduire les risques liés aux épidémies et pandémies de zoonoses émergentes et réémergentes. 3, maîtriser et éliminer les zoonoses endémiques, les maladies tropicales négligées et les maladies à transmission vectorielle. 4, renforcer l'évaluation, la gestion des risques liés à la sécurité sanitaire des aliments ainsi que la communication à ce propos. 5, juguler la pandémie silencieuse de la résistance aux agents microbiens. Et enfin, 6e, intégrer l'environnement dans l'approche une seule santé. Voilà les 6 domaines d'action actuels. Et pour finir, j'aimerais vous rappeler que la journée internationale du One Health est toujours célébrée le 3 novembre de chaque année. Et je vous remercie.
Thank you, Professor Fassiferi, for your insightful presentation. Please note that we'll keep the discussion at the end of the 4 presentations. So I'm inviting the people that are connected online to write down their questions, and then we'll in the chat. And we have Salah. Thank you, Salah, for writing and taking all the notes, and of course for the presence here, the audience that is present. You just need to raise up your hands at the end of the session. Our second speaker is Professor Bushra Al-Qadi. I think all the students and people from the Faculty of Science all know her. Professor at the Faculty of Science, plant biotechnologist, working on molecular biology, working on also on some diseases. She did supervised several theses and hundreds of papers, and we are glad to have her. She is also the president of the Moroccan Association of Biosecurity and Biosafety. Thank you for joining, and we are all ears to listen to you. The floor is yours.
Okay. Bien, bonjour tout le monde. Je suis ravie d'être parmi vous aujourd'hui. Désolée, je vais parler donc en français comme ma collègue Wafa. Alors aujourd'hui, donc ma thématique ou le thème que je vais aborder, c'est renforcer la sécurité sanitaire grâce à la génomique dans une approche One Health. Donc, Wafa, ma chère collègue, elle a initié déjà ce que c'est que le One Health, donc ça va être facile pour moi. Donc, vous savez, là, on a vu que le monde, et spécialement l'Afrique, est confronté à une montée de menaces biologiques qui exigent une réponse rapide, coordonnée et aussi durable. Et donc, ça s'insère dans l'approche One Health. Et ma présentation va être donc propose une petite réflexion. C'est une réflexion sur les défis actuels de la sécurité sanitaire, sur le continent et le rôle de la génomique et les nouvelles technologies dans une approche One Health. Donc, je commence par une petite introduction du contexte actuel des menaces biologiques, mais rapide. Les défis ainsi que le cadre réglementaire, les leviers d'action et la place de la génomique dans la prévention des risques sanitaires. Et enfin, je terminerai par la contribution des activités de mon association, qui est l'association marocaine de biosécurité, biosureté et de sécurité sanitaire. Alors, le contexte des menaces biologiques émergentes, on l'a vu à travers la dernière présentation. Au cours des 2 dernières décennies, le monde a été confronté à des crises sanitaires d'ampleur croissante. D'abord, donc, le SRAS en 2003, l'Ebola en 2014 et 2016, la Covid qui a, donc, vous avez vécu en 2020 en 2020 et jusqu'à 2023. Et dernièrement aussi, l'OMS a tiré la sonnette d'alarme concernant la résistance aux antibiotiques et aussi ne pas oublier les risques émergents, les risques des biotechnologies à travers donc les nouvelles technologies de l'édition du génome. Le contexte actuel donc de ces risques biologiques, il a aussi été donc accéléré par l'urbanisation non maîtrisée, par la perte de biodiversité, l'exploitation de tout ce qui est sauvage, la faune sauvage, les pratiques agricoles inadaptées, les pratiques médicales non adaptées aussi, et les déplacements internationaux. Et tout cela donc a créé un environnement propice à l'émergence et à la propagation des maladies infectieuses. Ces défis sont amplifiés aussi au niveau africain surtout, amplifiés par l'infrastructure sanitaire fragile, par la dépendance à l'expertise et aux ressources extérieures, par le déficit de formation et de technologie, et aussi par le manque de coordination multisectorielle. Et ça, c'est pourquoi on a besoin justement de cette approche One Health. on voit qu'on a donc à travers tous ces défis amplifiés, on a besoin, donc on a la nécessité d'une préparation globale. C'est aussi l'urgence d'intégrer la gestion des risques biologiques et on voit l'importance aussi d'une stratégie intégrée et adaptée pour une résilience sanitaire. On se pose donc la question à travers ce qu'on vient de voir. La question qui se pose, c'est comment renforcer durablement la sécurité sanitaire en Afrique face à des menaces biologiques multiples et en évolution constante. Donc, ces agents pathogènes ne sont pas figés, ils évoluent avec le temps et on a vu lors de la Covid, le SARS-CoV-2, comment il a évolué avec des différents variants et le risque sanitaire, bien sûr, diffère aussi. Pour répondre à cette question, donc on va voir un petit peu la sécurité sanitaire et donc c'est quoi déjà? Donc elle englobe un ensemble donc de paramètres. D'abord la prévention des crises, donc ces mécanismes qu'il faudrait prendre en compte, la prévention des crises sanitaires, la détection et la réponse face aux menaces biologiques. Alors, cette sécurité sanitaire, pour qu'on puisse donc la maîtriser et aussi la renforcer, on a besoin d'un certain fondement technique et fondement stratégique. Alors, les fondements techniques, c'est d'abord avoir de la biosécurité et de la biossûreté, c'est-à-dire comme approche dans les différentes, donc, que ça soit au niveau recherche ou autre. Avoir aussi un autre paramètre qui est la surveillance génomique et aussi l'innovation technologique. Alors, les fondements stratégiques, c'est la coordination multisectorielle, la communication du risque et la gouvernance scientifique. Bien sûr, pour répondre à cela, et à travers l'approche One Health, qui a été déjà définie comme étant une approche qui relie la santé humaine, animale et environnementale, elle repose surtout sur la prévention, la surveillance et la collaboration interdisciplinaire, avec les objectifs de prévenir les crises sanitaires à la source en intégrant tous les acteurs et mieux gérer les interactions complexes entre agents pathogènes, hôtes et écosystèmes. Alors, d'abord, définir ce que c'est que la biosécurité et la biosûreté, rapidement, qui sont des piliers techniques de la sécurité sanitaire. Alors, la biosécurité, qui est la biosafety en anglais, ce sont d'un ensemble de mesures et de pratiques pour prévenir, pour gérer les risques et les contrôler. C'est des risques biologiques susceptibles d'affecter la santé humaine, animale et environnementale. La biossûreté, qui est biosecurity, ce sont des mesures visant à prévenir l'utilisation malveillante d'agents biologiques dangereux. Les deux sont des conditions préalables à toute stratégie de surveillance. Donc, on est obligé d'avoir les deux volets, biosécurité et biosûreté, pour une surveillance génomique et aussi pour avoir une sécurité sanitaire intégrée. Voyons voir le cadre réglementaire international, parce que la gestion du biorisque s'appuie sur plusieurs instruments juridiques et normatifs complémentaires. D'abord, le règlement sanitaire international adopté par l'OMS, qui oblige les États membres à développer des capacités de détection, de surveillance et de réponse aux menaces sanitaires, y compris les risques biologiques. Ce RSI 2005, il a 6 piliers, avec ces 6 piliers pour la capacité de laboratoire. Le RSI offre un cadre qui structure pour améliorer la biosécurité. Il constitue la base opérationnelle à la préparation et à la résilience sanitaire mondiale. Le deuxième instrument, c'est la Convention sur les armes biologiques, la CABT, qui est un traité international interdisant le développement, la production et le stockage d'armes biologiques et ou à toxines. Et le troisième élément, c'est la norme ISO 35001-2019 qui établit les exigences du système de gestion des biorisques au laboratoire, garantissant conformité, traçabilité, environnement de travail sécurisé et aussi la maîtrise des biorisques. Voyons voir les enjeux pour l'Afrique. Alors, les enjeux pour l'Afrique à travers ce qu'on vient de voir, c'est que déjà dans les conditions africaines, ces cadres réglementaires, ils doivent être adaptés et opérationnalisés. Il faut harmoniser les réglementations et promouvoir la mise en œuvre de la norme ISO 35001 dans les laboratoires publics et privés. Également, promouvoir la formation en biosécurité et biosûreté des professionnels de santé et la sensibilisation du public. Et si on intègre donc ces éléments, ça renforcerait la biosécurité régionale et la confiance internationale dans les systèmes africains de santé et aussi de recherche. Alors, ce qu'on relève comme faille, c'est le manque de coordination multisectorielle qui entrave la mise en œuvre de l'approche One Health et limite la réponse aux crises. À travers ces défis, donc, j'ai essayé de voir quels sont les leviers d'action stratégique et de l'innovation qu'on peut apporter pour relever ces défis. D'abord, déploiement des technologies de séquençage génomique et aussi de l'intelligence artificielle, donc toutes les nouvelles technologies. Également, l'adoption de laboratoires mobiles et unités de terrain qui peuvent aider dans arrêter les crises sanitaires, des coopérations régionales et internationales, telles que donc, la coopération avec l'OMS et avec aussi CDC Africa, et le renforcement des infrastructures et capacités locales. Aussi, l'engagement communautaire à travers les associations qui peut donner un boost aussi à ces défis. Ces leviers doivent quand même s'accompagner par une gouvernance partagée, favorisant la transparence, favorisant la confiance et le partage de données entre pays. Regardons la part ou la place de la génomique dans la sécurité sanitaire. Donc d'abord, qui dit génomique dit séquençage, bien sûr, des génomes, et séquençage complet qui va permettre la détection précoce de l'émergence des pathogènes, une meilleure compréhension des transmissions et aussi une réponse aux épidémies. La mise en place de systèmes de surveillance génomique en temps réel permet d'améliorer les capacités de diagnostic, permet de suivre l'évolution des agents pathogènes, de collecter des données, de partager ces données aussi sur des plateformes, que ce soit sur les maladies infectieuses ou aussi sur l'antibiorésistance. Un exemple de programme africain, un programme AfroScreen qui a permis de surveiller les variants du virus SARS-CoV-2 par séquençage haute définition pendant la pandémie. C'est un programme qui a donné non seulement une part de décision publique au niveau de la décision santé, mais aussi il a permis de former pas mal de personnel de santé et autres. Donc ce programme, il a permis aussi, il a touché déjà 13 pays africains. Il a touché 250 professionnels en termes de formation, santé, médecins, vétérinaires, techniciens. Et bien sûr, il a été utilisé dans différents risques biologiques, pas que le SARS-CoV-2, mais aussi le Mpox et le virus de Lassa. Ce programme, il a permis aussi la sensibilisation des communautés pour adopter les pratiques biosécuritaires dans les milieux de santé, de laboratoire et de l'environnement. Et donc, ces efforts, bien sûr, s'alignent avec les priorités d'Africa CDC et aussi de l'OMS qui tentent et qui visent à développer des réseaux de surveillance génomique robustes. La génomique, elle a réellement évolué en plus grâce aux nouvelles technologies. Et donc, on commence à se poser, on ne peut pas ne pas parler, bien sûr, des nouvelles technologies qui peuvent transformer la surveillance biologique à travers l'intelligence artificielle et tous les outils numériques qui ont vu le jour. Donc, d'abord, elle a permis une surveillance numérique grâce au rassemblement donc, des données, au big data, à l'intelligence artificielle et à la modélisation. Donc, il y a eu pas mal de logiciels qui ont été développés, tels que le heatmap et le ProMed, qui a servi justement lors de la COVID pour identifier les premiers signaux épidémiques du SARS-CoV-2. Également, les blockchains, qui ont permis la traçabilité sécurisée des données liées aux agents biologiques sensibles et à la logistique aussi des vaccins. Donc, la blockchain, elle a été utilisée par Rwanda pour le suivi des vaccins de la COVID-19. Il y a aussi les biocapteurs intelligents, les IoT, qui permettent de suivre en temps réel les zoonoses et la surveillance environnementale. Et là, en Ouganda, il y a eu l'utilisation de drones de détection des fièvres hémorragiques. Ces technologies, il y en a d'autres, bon, c'est pas exhaustif, les technologies donc émergentes renforcent la surveillance sanitaire et aussi la détection rapide de ces menaces biologiques. Elles favorisent aussi la réactivité, la transparence et la coordination dans une approche One Health. Alors, en parallèle avec la technologie, il y a aussi un autre volet, on a parlé, c'est le déploiement de laboratoires mobiles. Alors, les laboratoires mobiles constituent une réponse adaptée aux zones reculées. Je donne l'exemple des unités de type Biosafety Pod qui peuvent contenir les épidémies par des analyses de terrain rapides. Ils réduisent le délai de diagnostic par rapport aux symptômes et donc ça constitue des solutions autonomes, mobiles pour renforcer la biosécurité là où les structures sanitaires sont rares. Et là, je donne l'exemple du Maroc aussi, le laboratoire mobile d'Imlab, BSL3, c'est un laboratoire que le Maroc s'est doté donc à travers un projet du NATO et qui se trouve maintenant au niveau de la DGSN, la Direction générale de sûreté nationale, qui est un laboratoire d'innovation en détection précoce et d'identification d'agents biologiques, STANA 4632, à risque civil et militaire en utilisant les nanobiotechnologies. Alors, c'est un laboratoire de haut niveau avec un niveau de risque BSL3 et qui peut être utilisé en cas d'alerte pour la détection rapide de l'agent pathogène suspecté ou la toxine suspectée et aussi comme plateforme de biologie délocalisée pour une surveillance locale, nationale et régionale. Alors, en parallèle à tout cela, il y a aussi le rôle de la communauté. Donc, on parle de résilience communautaire face aux menaces biologiques. On a vécu donc la Covid. Et à travers des programmes régionaux africains qui ont montré quand même, il n'y a pas que les vulnérabilités en Afrique, il y a aussi la montée en compétences en Afrique suite à ces crises sanitaires. Je donne des exemples. Donc, l'exemple de l'ASLM, donc c'est l'association africaine pour les laboratoires d'analyse médicale. La Africa CDC, bien sûr, qu'on connaît tous, le Centre donc, de contrôle et de prévention des maladies. Et aussi le NCDC, qui est le CDC nigérien. Ces programmes, ils ont permis par exemple au Niger, ils ont permis donc de former des 2000 agents communautaires pour détecter les maladies émergentes. Ces initiatives africaines sont très intéressantes. Il faut quand même les signaler parce que ça constitue quand même des réseaux de surveillance génomique et qui illustrent un leadership croissant du continent dans la maîtrise des risques biologiques et aussi la souveraineté sanitaire. Et là, j'arrive à tout ce qui est communautaire. Il y a aussi la MBS, donc autant qu'association. C'est une association qui, l'association marocaine de biosécurité, biosureté et sécurité sanitaire, qui a aussi son rôle à jouer dans tout ce qui est formation et sensibilisation, la formation, la sensibilisation des universitaires, des professionnels de santé aux meilleures pratiques de biosécurité, également l'organisation d'ateliers de formation continue intégrant les dernières avancées technologiques et aussi l'instauration des standards de sécurité dans les laboratoires marocains publics et privés. Et l'association, elle a organisé des journées scientifiques, des tables rondes et des congrès internationaux pour aborder les défis actuels et surtout les défis africains. Donc là, c'est juste pour, dans le cadre de la sensibilisation, la MBS, donc elle a fait des caravanes et pas mal donc, de formations et aussi de journées scientifiques à travers des collaborations, à travers des conventions aussi avec les différentes institutions, aussi bien universitaires que écoles de santé, que l'Institut de santé, ISPITS par exemple, et aussi un point assez intéressant, c'est la formation de 200 cadres scientifiques en biosécurité et biosureté à travers le master MEABB, donc le master euro-africain de biosécurité et de biosureté. Quelques perspectives d'avenir, parce qu'on est quand même très optimiste aussi, à travers donc c'est déjà cette initiative d'aujourd'hui qui rassemble pas mal de scientifiques à travers l'Afrique, à travers le continent. Donc, je lance quelques appels. C'est renforcer donc les réseaux africains de biosécurité et de surveillance génomique et aussi développer de la recherche appliquée et la coopération régionale et aussi investir dans la formation continue et les laboratoires de haute sécurité. À travers cela, il faut mettre en place des politiques publiques intégrées, One Health, pour une résilience durable et une santé globale, parce qu'on ne peut pas travailler seul, il faut travailler ensemble. Et je conclus donc par, on a vu un petit peu la sécurité sanitaire en Afrique, comment on peut la renforcer grâce à la génomique dans une approche One Health. Par conséquent, on peut dire que la sécurité sanitaire n'est plus une option, c'est une exigence. Donc, une exigence stratégique, et donc, donc c'est une exigence stratégique, et en combinant tout ce qu'on vient de dire, donc innovation génomique, technologie numérique, biosécurité, l'Afrique peut construire une résilience durable face aux menaces biologiques émergentes. Et je finirai par encourager les réseaux comme donc l'AMBS et l'association One Health Maroc, qui incarnent cette dynamique, une dynamique collective fondée sur la science, la formation et la solidarité. Et merci pour votre attention.
Thank you, Professor Boukhadi, for your talk. And if I might resume a few points and take home messages from the first talk. The main points were how can health security in Africa be sustainable, be strengthened in the face of all the diverse and the multiple and constantly evolving biological threats that was presented by Professor Fassiferi and Professor Boukhadi, with the challenges for Africa, with a few points that can be as follow: well, the structural vulnerability of health systems, increasing pressure from zoonoses and the emerging epidemics, and insufficient local production capacity, and we may say the lack of coordination and regional governance. These challenges can be considered as strategic, leverage, and innovation opportunities in Africa. that can be resolved by strengthening governance and regional coordination, by investing in human capital and the capacity building and expertise, and also by building laboratory and genomic surveillance. Several examples were presented by Professor Bulqadi. Thank you, ladies, for your talk. And now we can move on to our third talk by Dr. Zahra Taqi Shibihi. She's a biologist specialized in cytogenetics and human pathology in Rabat. She's also contributing to the quality and biosafety management systems, author of several international publications on pediatric leukemia and genetic biomarkers, and also serves as on the board of Moroccan Association for Biosafety and Biosecurity. Thank you for your attending, and the floor is yours. Go ahead. So much.
So good morning, everyone. I am Dr. Zahra Taqi Shibihi, and I am honored to be with you today. And I welcome you to my session entitled Strengthening One Health in Africa Through Biological Risk Management. It's a vital topic whose mastery is essential for the future of our continent. So to guide you, I will begin with the current landscape of biological threats in Africa, then dive into the One Health framework and its importance. We will examine the main types of biological hazards and, of course, the complementary safeguards. We will present some concrete cases in African experience before, of course, concluding with advancements and perspectives. So as of 2025, Africa faces a complex landscape of biological hazards, from emerging infectious diseases to antimicrobial resistance and laboratory incidents. These challenges, as shown by WHO, are actually can quickly escalate into public health emergencies, highlighting the imperative of a cross-sector coordination, or what we called, of course, One Health framework. So, what is One Health, and why is it so crucial for our continent? And of course, we need to understand the urgency of our actions through the answer to this question. So One Health is a concept is simple but yet powerful. It's the integrated approach that recognizes the deep connections between the health of people, animals, and ecosystems. It works to balance and protect them together. Why? Because simply the health of one is absolutely dependent and has the impact to, of course, the health of the others. One Health matters for Africa because our continent is a global hotspot for emerging zoonotic diseases. Of course, there is the increased mobility that accelerates pathogen transmission. There is population growth that fuel disease spread. The climate change actually alters vector distribution, and biodiversity loss disrupts natural barriers. These interconnected drivers actually set the stage or create the conditions to collaboration with international partners to strengthen biosafety and biosecurity across AU member states. It aims to develop national strategies, build infrastructure and capacity, and improve preparedness and response. This initiative reflects a strong message, which says Africa is no longer waiting for external help. We are building our biosafety future, and this is a strong message for autonomy and resilience. There were actually some key achievements. They didn't appear in the slide, but I'm just going to tell them orally. Is that the achievements from this initiative was that hundreds of experts were trained and national frameworks developed, regional training hubs established in key countries like Nigeria, Kenya, and Senegal. And this is not the end, because Africa CDC is currently developing the next strategic phase, which is the phase of 2025 and 2030, building on these foundations towards a safer, more resilient, and One Health aligned Africa. To conclude, I'd like to quote a message that perfectly captures the spirit of this presentation, which says, biosafety is not a luxury. It's a strategic necessity for sovereignty, security, and resilience. And the key message, of course, is that together we can build a safer, healthier future for Africa and contribute to global health security. And thank you for your attention.
Thank you very much for your talk. We'll keep, as I said, the questions at the end. And we are going to our last talk of Dr. Kautar Alilouche. She's having her talk online.
Yes.
Kautar, are you?
Yes.
You online?
Yeah. Yes, I'm here. Can you hear me?
Yes. If it can be a bit louder, it can be fine. Kautar?
Oui. Yes, I'm
Okay,
here.
thank you. Okay, so let me introduce you. Thank you for joining. Thank you for accepting to be part of this interesting session. Dr. Kautar Alilouche, she's actually professor of biology at the Subtech Santé in Sawera. She holds a PhD in biotechnology and immunology from DIV Hassan Sagan. And she have an expertise on monoclonal antibodies, vaccine development, and stem cell research. She's deputy secretary of the Moroccan Association for Biosafety and Biosecurity, contribution actively with several workshops that are done here in Amphi Belmahi, like frequently. Thank you for joining, and the floor is yours, Kautar.
Thank you. I still have some difficulty to share my presentation. Hello? Thank you. So hello everyone. Thank you so much. So good morning. It's a pleasure to be joining you even virtually today. So first, I would like to thank the organizers and to congratulate AfricaBP for the remarkable work achieved so far. It truly represents a great example of the One Health approach, like building capacities, studying pathogens, not only at the genomic level, but also to understand their transmission pathways, environmental conditions, and by sharing data across sectors. And this kind of integrated approach help us to generate real indicators for surveillance and early warning, and ultimately prevent future outbreaks before they spread. So congratulate AfricaBP. That's also the goal of my presentation today, to introduce the framework that guide countries and ultimately the world in putting One Health into action. So I will start with an overview of the One Health Joint Plan of Action. Second, we will go through the preparedness pillars, and finally we will look at how One Health can be applied in practice. So first, I would like to start with these figures. It shows two very different worlds. In the first, shows a typical reactive system. When surveillance is fragmented, infections move silently from wildlife to livestock and finally to humans. So here, wildlife and domestic animals infections go unnoticed until human gets sick. By the time we react, it's too late. In the second world, shows One Health in action. So when One Health systems are in place, forecasting, early detection, and rapid cross-sector response, the outbreak curves collapses before human amplification begins. And the benefits here is shared. We have fewer animal deaths, fewer human cases, and reduce economic loss. So the message or this image summarizes the value of operational One Health better than any definition could. And this message is clear: when we act early and together, we change the shape of an epidemic. And that brings us to the question, what is driving these spillovers in the first place? So statistics shows that the majority of human infectious disease originate in animals. And this trend has been driven by major changes in ecosystems, for example, climate change, deforestation, habitat loss, which are reshaping the interface between wildlife, livestock, and humans, and forcing species to interact in new ways. And these interactions increase the risk of zoonotic spillover, when pathogens cross species barriers and infect humans, such as seen with viruses such as Ebola, Nipah, and SARS-CoV-2. What this means is that if we focus only on human health systems in isolation, we risk missing the upstream warning signs such as animal outbreaks, environmental changes, which serves as early indicators of new threats. So that is why we need a broader lens called the One Health approach. So let's remind ourselves what One Health means. One Health is an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems. And these definitions remind us that One Health is not just about diseases, it's about systems. It calls for collaboration among health, agriculture, environment, education, and researcher. So from local communities to governments. But how could the One Health vision be turned into reality? So the answer came in 2022 with a shared global framework to guide countries. It's called the One Health Joint Plan of Action. This plan was developed jointly by the quadripartite organizations. Together, they created a common roadmap for global collaboration. So the One Health Joint Plan of Action covering the years 2022 to 2026, and it is built around 6 interconnected action tracks. Tracks 1 focused on building capacities and governance, making sure One Health is integrated in national systems. Track 2 tackles emerging and re-emerging zoonoses and pandemics by strengthening risk assessment and preparedness. And track 3 addresses endemic and vector-borne diseases, ensuring integrated control of those that persist every year. And track 4 is about food safety by harmonizing surveillance and communication along the entire food chain. Track 4 deals with antimicrobial resistance, the silent pandemic that demands joint human-animal environment stewardship. And finally, track 6 brings the environment fully into the One Health system because ecosystems health underpin everything else. So these tracks work together as a coordinated package, not 6 separate programs, but 6 dimensions of the same mission. So now, how do these tracks shape our preparedness and response? So each of these tracks addresses a key dimension of preparedness and response. The first preparedness pillar focuses on governance and financing. So to operationalize the One Health approach, countries need to establish a national One Health platform that brings together all relevant sectors under a formal, legally recognized mandate. Next, it's crucial to secure sustainable financing that supports preparedness and response activities across these sectors. This means moving away from project-based or emergency funding toward long-term integrated investment. And finally, national plans should align with the One Health Plan governance frameworks, ensuring coherence, accountability, and clear coordination between institutions. So once governance and financing are in place, the next step is to work together to understand and assess risk. The second preparedness pillar focus on joint risk assessment and planning. The joint risk assessment is the core operational tool that helps ensure a shared understanding of threats across all sectors. So first, countries are encouraged to conduct regular assessment, bringing together experts from human, animal, and environmental health to identify emerging risk early. Second, they should develop integrated risk matrix that map out possible transmission pathways, whether through animals, humans, vectors, the environment, or food. And finally, contingency planning is key. So joint plans should be developed and tested through multi-sector simulation exercises so that when a real event occurs, the response is coordinated and efficient. Altogether, this pillar ensures that preparedness moves from theory to practice through shared risk understanding, structured planning, and tested response capacity. Preparedness pillar 3 and 4 focused on integrated surveillance and laboratory networks. So these two pillars work hand in hand. Surveillance provides the early warning and laboratories provide confirmation and detailed analysis. So under integrated surveillance, the goal is to link data across sectors so we can detect threats early. This means merging human health surveillance with animal and wildlife surveillance. It also means incorporating environmental data streams such as wastewater monitoring, vector tracking, and ecosystem health indicators. Finally, we need to ensure all sectors can share information in real time through connected dashboard and shared data platforms. On the laboratory and genomic side, harmonized lab capacity is the backbone of verification and action. We must align biosafety and biosecurity standards across human and animal laboratories, establish joint sample sharing and data sharing protocols, and expand genomic sequencing networks that can track pathogens across species and environments. The next preparedness pillar is risk communication and community engagement. So preparedness isn't only technical, it's also social. When people receive clear and consistent information, trust grows and response becomes faster and more effective. So the first step is to establish cross-sector communication. So that human, animal, and environmental sectors use the same channels and agreed procedure to share updates and alerts. Then it's essential to ensure targeted engagement, like reaching farmers, market workers, local health staff, and other community actors who are often the first to notice unusual events. So their participation builds the foundation of trust and early reporting. Finally, all sectors must maintain unified messaging. That means delivering consistent, coordinated information on issues like zoonosis, food safety, and environmental hazards. So will be no mixed signals, no confusion. And can we really talk about preparedness without addressing antimicrobial resistance? Actually, Action Track 5 of the One Health Joint Plan of Action focused exactly on that. Antimicrobial resistance, a true One Health crisis that demands coordinated action across all sectors. So first, we need unified policies, consistent rules for antimicrobial use. Second, we must build cross-sector surveillance systems that monitor resistance patterns not only in hospitals, but also in animals, food, and the environments. And finally, antimicrobial resistance control should be integrated into national preparedness and response plans. So to ensure accountability and progress, we need to measure success through clear operational indicators. The One Health Joint Plan of Action encourages countries to track both process and outcome indicators that show how well preparedness systems are functioning. So first indicator is the spread of detection. how quickly we move from the initial animal health event to an official human public health alert. Time saved here means lives saved. Next, environmental coverage, which look at how extensively and how often wastewater and other environmental monitoring systems are used to detect emerging threats. And then, antimicrobial resistance stewardship by tracking reductions in antimicrobial use and resistance across both human and animal sectors. On the systems side, integrated data platforms measures how many priority pathogens are actually captured within joint surveillance dashboards. And finally, joint planning, which is the number and frequency of cross-sector risk assessment in simulation exercises conducted together. So beyond metrics, we must also recognize the shared benefits of investment. Investing in One Health is not only scientifically sound, it's financially smart. So early detection and integrated interventions are significantly cheaper than managing large-scale disease outbreaks, and they lead to fewer cases, lower morbidity, and reduced mortality. Also, beyond cost savings, there are also shared co-benefits. So healthier ecosystems contribute to the Sustainable Development Goals. Safer and more secure food supply chains improve global food security, and improved animal health means greater productivity and more resilient livelihoods. Now let's see how all these preparedness elements came alive during a real event. So this story from the CDC, Center for Disease Control, beautifully illustrates how One Health prevents outbreaks, not just how it responds to them. Rift Valley fever outbreaks often start in livestock after heavy rains. In the past, human health systems react only once people were already infected. But with One Health integration, everything changed. Environmental specialists use satellites and rainfall data to predict when mosquito eggs will hatch weeks before disease appeared. Then the veterinary services move in first, vaccinating animals and controlling animal movement in high-risk areas. On the other side, human health teams are alerted in advance, so hospitals are prepared and community are informed about mosquitoes protections and safe handling of animals. And all sectors, animal, environmental, and human, share data daily. So if livestock infections drop, we know the measures are working. As a result, the infection cycle is cut off before it spills over to humans. Now the One Health Joint Plan of Action gives us the framework. Now it's up to countries and institutions to apply it. So from principle to protocols, that's our challenge. First, mandate the platform. Second, fund the surveillance roadmap. Third, launch joint exercises, whether through tabletop simulations or full-scale drills, because testing coordination in real time reveals the gaps before an actual crisis does. And finally, review and budget. Every exercise and response should end with a formal review, identifying what worked, what didn't, and linking those lessons directly to policy and funding decisions. So putting One Health into action means making it part of our institutions through mandate, financing, practice, and accountability. So before I conclude, I would like to emphasize that One Health is not just a framework, it's a mindset and a shared responsibility. When we act early across human, animal, and environmental sectors, we prevent crises instead of reacting to them. The One Health Joint Plan of Action gives us a clear path. Build capacities, link data, strengthen laboratories, communicate transparently, and act together. So this collaboration transforms preparedness into prevention and prevention into resilience. Thank you. Thank you so much for your attention.
We can't hear the online presenters, on-site presenters.
So how can we prevent the next epidemics? Okay, thanks.
Alors, merci pour cette question. Alors, pour prévenir les épidémies, on a vu à travers déjà les différentes présentations qu'il faut travailler en réseau. Donc, d'abord, avoir donc, un réseautage entre les différents secteurs déjà, et avec des programmes où on peut intégrer tous les secteurs. Bon, d'abord, le biologiste, les vétérinaires, les médecins, aussi, donc, il n'y a pas que le secteur de la santé, mais il y a aussi les écologistes, etc. Et là, c'est à travers des recherches, à travers des projets de recherche où il y a les différentes facettes et chacun peut sonner l'alarme s'il y a donc une détection précoce d'un agent pathogène, que ce soit au niveau chez les animaux sauvages, en écologie, etc. Donc, c'est un ensemble de secteurs qu'il faut travailler ensemble. Maintenant, pour ça, donc, il faudrait être armé aussi par les nouvelles technologies. C'est ça le plus important, c'est-à-dire déjà avoir des échantillons en temps réel et chercher bien sûr à détecter ces types de pathologies. Maintenant, il y a aussi le volet prédiction qui peut jouer un rôle très important aussi, c'est à travers quand il s'agit par exemple de maladies vectorielles. Quand il s'agit de ce type de maladies, pour suivre les vecteurs, il y a tous les paramètres écologiques, édaphiques et aussi climatiques qu'il faut suivre. Et donc, on peut avoir de la prédiction d'une épidémie future si toutes les conditions se rassemblent. Voilà, merci.
So,
No,
should we move to audience questions?
yeah. I think we have Kautar Lelouch. She's reconnected. Kautar, can you hear us?
Yes, I'm here. I can hear you.
You had one question. Can you repeat the question for Dr. Lelouch, please?
So you had an online question that went, how can African countries coordinate to spot outbreaks early and share alerts quickly?
There is no more good example as the AfricaBP. So it's a good example for One Health approach that integrate together all sectors. So I also recommend that as a researcher or at a researcher level, we need to have the One Health approach mindset. So when we conduct a research project, we need to also think about the transmission pathways, not just like sequencing or keep it at genomic level. We need to think about the environmental changes. We need to look for indicators that warn us earlier to prevent an outbreaks.
So I like the One Health mindset, Kautar. I like it. I'm gonna keep it. And I think it's you have a good point, because we need to rethink our research and then take in consideration this One Health approach. Thank you for your sharing. We have another question from the audience on site. Please go ahead.
My question goes for all the panel, Madam Leila included. Watching back everything, I just wanted to ask if y'all think that maybe we are prioritizing preserving tigers and trees when it is known that microbes make up for most of the biodiversity on Earth, as well as interactions in One Health concerns. So it is more of a question of priority. So.
So your question in the priority of the next steps, right?
Yes.
To be just more clear. Okay. So, yeah, for all of the speakers. So if you would identify one priority action, you know, for to advance genomics within One Health approach. Un secteur prioritaire pour vous, mesdames, peut-être que vous voyez urgent, je sais pas, une feuille de route.
Je vais rebondir rapidement sur les priorités. Au fait, la première priorité, c'est de pouvoir échanger entre les différents secteurs. Et les priorités, si je parle du Maroc, les priorités, c'est de pouvoir impliquer d'une manière durable le secteur de l'environnement. Malheureusement, nous commençons entre santé animale, santé humaine. Nous commençons. On est au début, mais on n'arrive pas encore à mettre avec nous les gens de l'environnement pour la santé de l'environnement. Donc, je pense que le plus grand effort à faire, c'est d'intégrer cette partie. Voilà, merci.
So the answer of Professor Fassifiri is clear. So the integration of sustainable like involvement of different sectors and especially the environments, like we say that we're still behind the collaboration or work between human and environment, and it's sector that is very important. And we have a researcher from the Faculty of Science working in environment. It's now you have a priority sector to work on, right? Second, another question? For a bit.
Okay.
Ladies
Professor?
first, go ahead.
Thank you. Assalamu alaikum. Thank you, Professor Fassi Fihri, Professor Belqadi, Dr. Taqi Shibih, and Professor Alilouche. I'm a master's student at the program Genomics and Bioinformatics, and my question is for Dr. Taqi Shibih. I was wondering why our laboratory incidents often underreported in Africa, despite their potential impact on public health and safety. Thank you very much.
Yes, thank you for your question. It's a very important one. You are absolutely right. The laboratory incidents have a very big impact on human health, and the fact that it's underreported in Africa, it's actually due to the fact that there is lack of reporting systems, if I might say, and the national frameworks and laws and policies that actually oblige the managers or oblige the responsible managers to report their actions. And there is also the fact that most people, when they have a laboratory incident that include biological risk, they are quite afraid to report this incident because they fear repercussions, if I might say, or they fear punishments, and they fear the view of their supervisors maybe. And there is also that, and the fact that they fear the repercussions also means that there is lack of the biosafety culture that we really emphasize on the fact that it should be strengthened more and more to strengthen this culture by these initiatives and these programs and these conferences like the one we're in today, to emphasize the fact that not reporting an incident, it's not a punctual problem. It actually can reflect to a bigger health problem or a public health problem. So I really take the moment here to really emphasize this point that we have to have this culture of biosafety during our daily routine procedures in the laboratory, either biomedical labs or researchers labs. And if something happens, we shouldn't be afraid to report it. And just to give you an example, when I was researching, I really had difficulty finding documented incidents in Africa. There are really few ones, and the Salmonella typhi was the most reported one. But I'm sure there are several ones, and there are multiple ones. They're just not reported and not documented properly. Thank you for your question again.
Go ahead, Professor Bedabi.
Thank you very much. I have just an opinion. I think in this post-dose One Health, above all, when we deal with zoonotic diseases, we are the weaker part. We are the weaker part for the reason that controlling pathogens, it's not that easy. Above all, that the mutation rate is very high. And above all, that the consequence of the mutation is unexpected and highly complex to dissect. What I mean by that, I mean, even if we use genomics and we use all, and we detect what is the part that gives this new emergence of the pathogens, we need really very sophisticated tools to control this, because we know either if we put our hand on the problems, how we make vaccines, it takes a lot of time, how we produce treatment, it's very complex. I think what need to be added to this approach, it's what Professor Belqadi mentioned partially in her presentation, maybe using new technologies like AI, like gene editing to manipulate the genes. I think we need more strong genomic tools rather than simple one of sequencing to be able to act. Yes, the approach is very good in detecting the problem, but acting on them quickly, I think we need a very strong arms, gene editing, AI, modeling, and all that could allow us to deal with the problems.
Thank you, Professor Bedawi, for your comment. I think we can take one more question from the online audience. Please, Salah. Waiting for the question. Is any question from the on-site audience?
Yeah, so this is a question for Dr. Shbihi. What low-cost community actions most effectively prevent everyday infections?
Well, when we talk about prevention, the first thing we think about is the importance of training and awareness and raising awareness. This is the thing that when you said low cost, well, this absolutely costs nothing. When we talk about preparedness and we talk about rising awareness amongst our community, this is personally, I think it's one of the most key lessons or the key elements that we should really hang on. There are lots of programs, there are lots of associations like the one Professor Belqadi talked about, and there are lots of initiatives in this area. And the more we raise awareness and the more we train people about the importance of these matters and the biorisk management, the more we're aware of the impact of it, and we manage it better. And thank you very much.
Thank you.
Thank you. I have a question for Dr. Taqi. You mentioned antimicrobial resistance. How does it connect to biorisk management?
Thank you very much again for your questions. That means that you're really interested in the topic, and that's actually an honor for me. When we talked about antimicrobial resistance, we said that it's a silent killer. Why? Because it's a biological, it concerns biological risks interface between the three sectors, and it's really challenging for through One Health framework. And for it to be more better managed, as we can say, we can prioritize the fact of ensuring safe handling of these resistant pathogens, especially in researcher labs, because safe handling is one of the main reasons, or the unsafe handling is one of the main reasons why these resistant pathogens can actually not only affect the lab personnel, but it's also affect the environment, especially if, for example, it's not eliminated properly. And that's why we have a strict waste management procedures that goes into the biosafety measures. And that's one of the reasons. There is also the fact that we should strengthen the infection and the IPC, the infection prevention and control, and to implement all the biosafety procedures that we talked about earlier, and raise awareness about the use of antibiotics. And also there is the policies and laws and national frameworks that should work on this area too, to limit the use of antibiotics, not only for humans or for treatments, but also for agriculture and veterinary use. But I think Professor Fasil Fihri knows a bit more about this aspect.
J'ai pas bien suivi, mais je pense que vous parlez de la résistance antimicrobienne. En fait, nous avons fait une recherche sur les laboratoires humains et les laboratoires de diagnostic animal. Et nous avons trouvé une liaison entre la résistance antimicrobienne. Alors, chez les animaux, en fait, il y avait surtout le poulet. C'est là où on a le plus travaillé sur l'antibiorésistance par rapport aux autres espèces, parce que le poulet a fait appel au laboratoire beaucoup plus que les autres espèces. Donc, la quantité de données obtenues, c'était la volaille. Donc, et on a remarqué sur 3 laboratoires privés à haut débit sur Rabat, donc les responsables des laboratoires ont bien voulu nous donner leur antibiogramme et leurs résultats d'antibiogramme, et nous avons trouvé une corrélation entre l'utilisation des antibiotiques chez la volaille et la résistance chez la volaille avec la résistance humaine. Donc, voilà, c'est un travail que nous avons fait l'année dernière avec une étudiante pour son PFE. Et bien sûr, il faut le développer davantage. Voilà, merci.
Yeah, I think we have some participants online that have some questions.
Okay, just I think we'll take one more question from the from the online audience, and then we'll close the session.
Yeah, so okay, I think we have 2 participants that have their hands up. So is there a chance they can just merge their question together? Samuel and Tobias, can you be very brief with your question for 30 seconds, please, each? Samuel, go first. Samuel Sahil Kebed. Okay, maybe
okay,
Tobias.
Thank you.
Okay, go ahead, Samuel. Yeah.
Okay, thank you all of presenters. It is nice presentation. I will have one question, which is on biosafety and biosecurity. As we are in Africa, biosafety and biosecurity issue is a major issue. So how we integrate biosafety as well as biosecurity in our health institutions? That means our hospitals as well as other health institutions, including regional and national laboratories.
Tobias, do you also want to just quickly ask your question so they can?
Okay, is that audible? Okay.
Yeah, so I think, Samuel, your question will be answered. Tobias, very briefly, please.
Okay, thank you.
Okay. Yeah, thank you. This Professor. What's happening? Can I come on? Do I go ahead?
Come on, please.
Okay. This is Professor Tobias Ezejo for Federal University of Technology, Owerri, Nigeria. Mine is actually a comment. Comment in the sense that two earlier presenters did this in French. I couldn't understand anything, but I think from their slides, it's great presentations. However, based on my understanding of the last two presenters on One Health, those lady presenters did great work. And I'm thinking, whether we cannot, even when we are talking about One Health, you find out that approaches are still compartmentalized in terms of environmental health, in terms of hospital, in terms of veterinary or animals. When we talk about One Health, we want to think whether there will be a way of building systems that will be of both diagnostic and the surveillance, competence or capacity, whereby some markers or some systems will be developed that will be unified in approach in monitoring diseases, both in the human, in the animal, and in the environmental sector. Thereby, particularly in low-income countries such as we find in most of African countries, where cost is always a problem. So we'll be thinking about a system whereby we develop a unified system of monitoring that becomes both useful in the three compartments under One Health consideration. That's my thinking. So that instead of cost being duplicated or being triplicated in the different sectors, we have a one-track approach of monitoring system that is effective both in the animal environment and in the human. Is there thinking? Maybe we'll take in that direction. Thank you very much.
Thank you, Tobias. Over to you, the on-site moderator, please, for the response to the questions.
I will take a moment to just respond to the second person quickly before responding to the first one. You talked about the understanding of the One Health framework and the French presentations. Well, we made sure that the second presentations, or the third and the fourth one, were absolutely understandable by the whole community, and we practically rephrased lots of concepts already spoken about in the first presentations, so that everyone has absolutely the same understanding and the same sharing of information. I will now just answer quickly the first question. Maybe there is a little lapse between the comprehension of the difference between the measures between biosafety and biosecurity. I will just redefine quickly the two procedures. The biosafety concerns all the measures implemented in a laboratory, for example, to avoid or to prevent accidental and unintentional exposure of the pathogens. So when we say unintentional or accidental, we use some key elements. We use some procedures, some strict procedures to prevent these exposure. We talked about the PPEs, or the personal protective equipment, the installation of infrastructures, and of course capacity building. The cabinets, the biosafety cabinets should be adapted to the type of the hazardous risk, and there are lots of things like waste management procedures, or in general standards operating procedures. Those are the measures or the ways to prevent these kind of accidental or unintentional exposure of the biological harm or the biological hazard. On the other hand, the biosecurity, on the other hand, concerns the measures that we use to prevent intentional the intentional exposure of the biological harm. And this is really the most key word to keep in mind, is the intentional use of these harmful elements. And of course, the measures we use to prevent these are like, for example, the physical control access in the laboratory of of course, the lab personnel. There is also a very important point about the staff reliability. The person that works with these infectious agents should be someone that we rely on and that has the conscience and the social, the community understanding and the awareness of the harm of these pathogens. And there is also the cybersecurity. We have to have robust cybersecurity for the data that's, of course, that we use. There are lots of elements that we, of course, make in place to prevent this kind of intentional use, like, for example, misuse or theft of these pathogens that can have a very serious implementation into the public health or the use of these, of course, harmful pathogens. And I wish I was clear, and thank you again for your question.
Thank you very much. I think it's time to end the session. Thank you very much to our distinguished speakers. Please applaud all of them. Thank you. Your sharing was insightful and complementary. Thank you. This session highlighted how genomics can advance, can help, and can serve as a powerful catalyst for advancing the One Health approach, connecting the human, animal, and environmental health, as was said by our speakers, and fostering innovation across Africa. I believe the key messages from this session is clear. Building resilient health systems require collaboration, knowledge sharing, capacity building, and also the integration for like genomics and new tools, as was said by Professor Badawi, into our strategies for prevention and response. Thank you very much for your presence. I think that we are having a coffee break, but before we'll take a photo group, please, if people can join us here. And if the audience online can turn on their camera so that we can see them and they can be in the picture to memorize this nice moment. Thank you very much. To see all the audience from online. Thank you for being and staying with us. Everyone will be in the picture. Please come on.
BGI Genomics is a leading global provider of a wide variety of BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since Illumina Biotech West Africa opened its door for the business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within IITA.
It feels just like yesterday. I joined InCaba in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for InCaba Biotech, our creation and existence has benefited considerably from experience and mentorship of Inkaba Biotech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At Inkaba Biotech Limited, our core values are quality, customer focus, integrity, teamwork, and others.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from PacBio West Africa Limited, Canada. My name is Yusuf, the manager for PacBio Ghana.
Good morning, I am Mamadou Dabo from Mali.
I extend our warmest congratulations on the 10th anniversary of PacBio Nigeria branch. Congratulations to our counterparts and our colleagues in PacBio Nigeria for their 10th anniversary.
Happy 10th anniversary year to all Inkaba Biotech staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such, we see the need for improvement. And in the future, we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA, as a company, Inkaba Biotech West Africa, has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary, Inkaba Biotech West Africa.
Long live Inkaba.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary, Inkaba Biotech West Africa. Cheers.
As we look ahead, Inkaba Biotech remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed the enterprise, including the setup of an own lab that provides summary sequencing services in Ibadan. And we look forward to add other genomic services like next generation sequencing using biosequencers like we do here in Pretoria. As Inkaba Biotech, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana, and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since AfricaBP West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within IITA.
It feels just like yesterday. I joined InCaba in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for InCaba Biotech, our creation and existence has benefited considerably from experience and mentorship of InCaba Biotech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At AfricaBP Biotech Limited, our core values are quality, customer focus, integrity, teamwork, and others.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from AfricaBP West.
from Mali.
I extend our warmest congratulations to the team and Ministry of AfricaBP Nigeria branch. Congratulations to our counterparts and our colleagues in AfricaBP Nigeria for their 10th anniversary.
Happy 10th anniversary year to all Inkaba Biotech staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such, we see the need for improvement. And in the future, we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA, as a company in BioInformatics West Africa, has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary INCABA BioTech West Africa.
Long live INCABA.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary INCABA BioTech West Africa. Cheers.
As we look ahead, INCABA BioTech remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed an enterprise, including the setup of an own lab that provides summer sequencing services in Badan. And we look forward to add other genomic services like next generation sequencing using biosequencers like we do here in Pretoria. As INCABA BioTech, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana, and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
This. So we're going to start session 2. I would like to ask you to please take your seat. And if you can, Salah, if you can call people outside if they want to join. Okay. So welcome to session 2, Biodiversity, Genomics for Conservation and One Health. Well, in this session, we explore how advances in genomics are transforming our understanding of biodiversity. So we are privileged to hear from 2 outstanding scientists who are leading global and national initiatives in this field. It is now my great pleasure to introduce our first speaker, Dr. Shane Murray. Dr. Murray holds a PhD in plant molecular biology from the University of Edinburgh and brings over 25 years of experience in research, teaching, and project management. Dr. Murray has held key positions at the Agricultural Research Council, the University of Cape Town, and the Center of Proteomic and Genomic Research in South Africa. In her current role in as a genomics coordinator at Diplomix, Dr. Murray leads the 1,000 Genome South Africa project, which is a national initiative focused on advancing genomics capacity and works in close collaboration with all partner laboratories of Diplomix network. So we are delighted to have her with us today to talk about this initiative. I'm just going to make sure that Dr. Murray is with us. Can you hear me, doctor?
I can hear you perfectly. Thank you.
Okay, so thank you. The floor is yours. I'm sorry.
Thank you very much.
Okay,
you can hear me fine.
Yeah.
Okay,
yeah,
thank you very much.
The floor is yours. Thank you.
Okay. Okay, I'm just going to switch off my camera while I'm talking. Thank you very much to the conference organizers today for inviting me to come and talk. So I'm reaching you from the most southern point of the continent, and so it's great to be interacting and to hearing about what is happening in the north of Africa. So my name is Shane Murray, and my role is Genomics Coordinator at Diplomix. Diplomix is a research infrastructure. It stands for Distributed Platform in Omics, based in South Africa. We are supported by the Department of Science, Technology and Innovation through a program called SARIA, the South African Research Infrastructure Roadmap. And we are hosted, we are hosted by a not-for-profit biotech company in Cape Town called the Center for Proteomic and Genomic Research. And so Diplomix has the mandate to enable access of students and researchers in South Africa to a world-class omics infrastructure, with omics representing both genomics, which is the field that I look after, and then also proteomics and metabolomics, and then also bioinformatics. And so we reach our mandate through... Apologies. I'm trying to just get back to that screen.
Yeah, I think, Shane, we can't see your screen moving.
Yeah, my screen has frozen. So I'm going to stop sharing and then reshare again.
So alternatively, you can convert it to PDF and then kind of share the PDF.
Okay, let me see if... Let me try one more time with the PowerPoint. Can you see it okay?
Yes, loading up. Now we can see the first slide.
Okay, great. And the second slide, all fine.
We can see this. Oh, yeah, we can see the second slide. Yeah.
Okay, great. So it's just going to take a few seconds. So I'll just bear that in mind. Okay, so we support genomics in South Africa through a number of different different initiatives. And one of them is what we call high visibility, high impact projects. And we've got 4 main aims there, is to build a community of users, to do a lot of training in that particular field, to develop methods that the service labs and our network can then use, and to incorporate open science into the initiative as well. And so 2 years ago, we set about designing a high visibility, high impact project around biodiversity sequencing. And we elected to use Oxford Nanopore sequences, as they were relatively affordable, and we were able to negotiate a bulk discount arrangement with ONT, which meant that we could buy flow cells on behalf of a distributed network. So we placed one P24 sequencer, which is the bigger sequencer in the Diplomics training lab, and then we placed the smaller P2 solos, which are about the size of a loaf of bread, at the University of the Western Cape, at Sengen, which is a small plant genomics company about 2 hours out of Cape Town, at the University of Pretoria, and at the South African Institute for Aquatic Biodiversity, which is in the Eastern Cape. And then our 6th partner on the project, the AMRC Genomics platform, already had a P2 solo, and they joined us. So why did we use Oxford Nanopore long-read sequencing? Why did we elect to choose this? Well, if you consider a long strand of DNA that passes through a nanopore, this is a very sort of high-level description of how Oxford Nanopore sequencing works. And as that long DNA strand passes through the pore, there's a change in the current, and that squiggle plot, the current intensity, can then be converted into a base sequence. If you consider that long piece of DNA as a big puzzle piece, it's much easier to build a puzzle with big puzzle pieces compared to small pieces, especially if you don't have a picture to copy. And there were other reasons too. As I said, it was more cost effective to go this route as well. So what is 1KSA? I've already mentioned it's a Diplomics project. It's focused on biodiversity sequencing in South Africa. So it's a proudly South African project. We're doing everything from sample collection, DNA extraction, library prep, sequencing, and all the downstream steps here in South Africa. It's a framework for teaching, for training, and for outreach. It uses a distributed infrastructure, which was important for us. There's a lot of method development that we share among the different labs. So we've created a community of labs that are able to do the sequencing. And we also use open science principles. We create draft genome assemblies, and we partner with the sample contributors who provide us with the samples for DNA extraction, and they can then use these draft genome assemblies in their further research. We also have a centralized collection of sequencing files, which are available for people to access under certain conditions. And then it's also an opportunity to collaborate with biobanks in South Africa. One of the long-term aims of 1KSA is to make sure that the genome sequences are linked up to accessions in biobanks, so that this combined biodiversity information can be incorporated into GBIF and other data repositories. So this is the landing page of the 1KSA project. We have a lot of information on our website. Please go and have a look at it to learn a little bit more about the project. But on our landing page, we update these key metrics once a month. And you'll notice here that we have sequenced more than 150 eukaryote samples, and we've just under 130 assembled genomes. And as of last week, we were able to publish 100 species cards, and I'll talk about that in a little bit further on in a few slides' time. We also have a lab at the University of the Western Cape who are focused on bacterial genome sequencing, and they have sequenced more than 500 bacterial isolates to date. So this slide describes the project as a whole. So scientists, we call them sample contributors, are able to go onto the website and can fill in a short form motivating why their species of interest should be included under 1KSA. And the criteria are that it has to be collected in South Africa, it has to be indigenous to South Africa, The sample contributor is responsible for collecting the sample, but also for getting permits and ethics, and is responsible for getting the DNA to us. The steering committee will evaluate the motivations on a monthly basis. And once a sample or species is approved, we use REDCap to do an online sample submission form to the sample contributor, and they then fill that in with all the metadata around the sample, where it was collected and so on, and all that information around the permits and the ethics. And then the DNA can be delivered to one of the 6 labs. And typically, the sample contributor will choose a lab that is geographically close to where they are based. So the 6 labs then carry out DNA quality control on the samples that are submitted, and then they carry out library prep, followed by whole genome sequencing using the PromethION. Information is collected at each step of the way using REDCap. So we use REDCap sort of as a LIMS system. The data is transferred from the sequences using Globus Connect to a national infrastructure, the CHPC, where the 1KSA pipeline is run. And after that, all the different data sets are then also transferred via Globus to DERISA, which is another national infrastructure for long-term storage. We have developed policies around sharing that data. I'll talk about that shortly. And we use Zenodo to create a community, and we publish our methods and our policies on our 1KSA community on Zenodo. A big part of 1KSA is raising visibility and awareness around the species that have been sequenced, and we use kind of a report card, which we call a species card. And here are 3 examples. The Bruder's whale, which is the only inshore whale found off South Africa's shores. This was sequenced by the University of Pretoria. The speckboom, which is an iconic succulent found in South Africa that was sequenced by Sengen. And then the galjoen, which is actually the national fish of South Africa that was sequenced by SciAp. We also produce stickers. You can see example of stickers for these 3 organisms here, which are normally very popular at various conferences that we attend. We also make videos about some of the key interesting species that we work with, and you can also find those videos with links from the website. So not only have we generated a community of users, so people who are able to do sequencing of biodiversity in South Africa, we've also generated or created a community of sample contributors. And to date, we've had more than 45 scientists come forward with samples for sequencing under 1KSA, and they belong to these various organizations that are shown here on the screen. So we're quite pleased that the impact of what we are doing is spreading beyond just the sequencing labs. The terms of use of the 1KSA project, these are listed on the website, but we are using the open science mantra as open as possible, as closed as necessary. So we have developed policies, as I said, around data access. So for the sample contributor, they receive the sequencing files back and also the draft genome assembly, and they are then free to use that data for up to 3 years. We chose that period of time because we wanted to give South African PhD students, which is typically a minimum time is 3 years, the opportunity to acquire the skills themselves so that they could analyze their data sets themselves. We do request that after the 3-year period, the data is then uploaded onto the European Nucleotide Archive. We have started to think about the 1KSA species as being different between either a conservation species or a bioeconomy species. In the case of a conservation species, it would be very important for that genome data to be released as soon as possible so that people can use it in order to design conservation strategies. But in the case of a bioeconomy species, where there might be a lot of interesting secondary metabolites being produced by the organism, we have started saying that it's possible then for the sample contributor to request a longer embargo so that they can fully evaluate any potential intellectual property that might arise from the genome. In terms of other scientists requesting access to data sets, We do have a form on the website as well that enables people to request access. We have a data access committee that will evaluate requests for data, and we have a data transfer agreement template that we've developed for this as well. We do also discuss data access requests with the sample contributor. Our policies around open science and acknowledgements for publications and reporting and publicity are also on the website. We do request that the sample contributor and the data user track any intellectual property that is generated through having the genome sequence available. And we would really like in future to develop a biodiversity genomics trust, which could then be used for benefit sharing as a benefit sharing mechanism. So a portion of intellectual property could be paid into that trust, and that trust would be used for the purposes of education, especially around the indigenous knowledge holders of particularly important bioeconomy species. So in terms of our method development, so if you remember back to one of my first slides, method development was a key part of why we set about designing 1KSA. So in the lab, there's 3 key aspects: DNA quality control, library prep, and sequencing. And we've developed a decision-making tree around the DNA that we receive. So if the DNA passes QC, it can then proceed to the next step. But if it fails, we've developed about 3 or 4 different cleanup strategies to try and improve that DNA, bearing in mind that very often this is a very precious sample and the DNA has been extracted from it, and we have to basically try and make it work with whatever DNA we receive. We then look at the average fragment size of the DNA. If it is not too high molecular weights, we can proceed with no fragmentation. But if it is longer than about 40 kb, it does require a little bit of fragmentation for the library prep kits that we are using. The next step is to then look at the genome size. If the genome size is above 1 gigabase in size, the expected genome size, we would then use the ligation kit. And if the genome size was less than 3 kb, we would then go on to use a single flow cell, which would generate about 30x coverage. If the genome size is much bigger than 3 gigabases, then we're going to have to use multiple flow cells. So that's more about sort of a stock management decision as well. We also have 2 different DNA fragmentation buffers that we can use, a long fragment buffer and a short fragment buffer. And in the early days, we tended to use the short fragment buffer more often, but as the DNA quality is improving over time, we are increasingly only using the long fragment buffer in the ligation step. If the genome is small, so it's less than 1 gigabase in size, we have the option of potentially multiplexing that species with other species. And we can proceed with that approach if we have more than 2 micrograms of DNA. But if we only have a relatively small amount of DNA, then we still use the ligation kit. So yeah, so this is an ongoing process, and as we learn more about the samples and the DNA quality, this is likely to change. On the data analysis side, we've also done a lot of method development, and initially we set up a workflow in NextFlow using Fly as the assembler. This was on the advice of Oxford Nanopore, but we quickly saw that under the conditions on the CHPC, when we had big genomes, we were running into wall time issues. So we could assemble genomes that were up to about 5 gigabases in size using Fly. But anything more than that, we just were really starting to have problems, and we were starting to get worried about that. But luckily, at the beginning of this year, Oxford Nanopore announced that HiFiism, a PacBio assembler, could now be used for Oxford Nanopore sequencing reads. And we've tested this in our own hands, and it's giving us really good results. So we've also developed a decision-making tree for doing draft genome assemblies. So the first QC, one of the first QC steps that we use is looking for a KMA peak, so doing a KMA count to see if there's sufficient DNA for a genome assembly. If the answer is no, that there's no KMA peak, then we've got a couple of tips and tricks around filtering and trimming reads. If the KMA peak is there observed, we then proceed to the genome assembly step. But if we still don't see a KMA peak, then we go back to the lab to do more sequencing. And this is in particular where the estimated genome size doesn't quite correlate to the final genome assembly size. So once we have that KMA peak, we then ask, is the genome bigger than 3 gigabases? If it's no, we can still use fly with RECON, which is the polishing step. If it is bigger than 3 gigabases, then we go straight to HiFiSM. The recommendation from Oxford Nanopore is that we needed a sequencing N50 greater than 10 kb. So we can proceed if that's the case. However, if it's less than 10 kb, we can also filter and trim the reads to try and improve that. But we have also been finding more recently that we still get really good assemblies using HiFiSM if the sequencing N50 is less than 10 kb. So most likely this decision-making tree is going to change at some point in the future, but for now, this is the current version that we're working off. So just in terms of some results, so there are a couple of key metrics that we look at. So the amount of data produced from the sequencer, so the read N50. So the read N50 being defined as the size of the small read where it and all of the larger reads make up 50% of all the reads. We also look at contiguity of the draft genome assembly, and we do this by looking at the conteg N50 with the length of that conteg, where all the contegs of that length and longer cover at least half of the assembly. And we're looking at a minimum and hope at about 30 kb there. And then the final metric is completeness, where we use the benchmarking universal single copy orthologs, the BUSCO tool, where we're also looking at a minimum of about 95%. So this is the first 100 genomes for which the species cards have now been produced. So on the left-hand side is the count of the sequencing N50s, and you'll see that there's a big distribution. But all the way up to 25 kb, we're getting quite a few in that bigger category over there. And then in terms of the draft assemblies, the conteg N50, most of our genomes do have a lower conteg N50, so less than a megabase in size. But we do have some with really good conteg N50s, around 90 megabases in size. And this is, I think, what we're seeing this is as a consequence of being able to use HiFiSM for the draft genome assembly. And just to show you an example of one of those, a species card of one of the plants with an excellent, really long assembly N50. This is bush. It's called kabok bossie in Afrikaans, or snowbush in English. It grows in the Karoo, which is the big internal dry region in South Africa. And it's one of the most recent plants that have been assembled, and it showed an assembly N50 just under 90 megabases. In terms of the BUSCO score, so this is just a breakdown of the different groups that we have analyzed. And you'll see that in most cases, we've got way over 95% single and duplicated. If you combine them together, a BUSCO score above 95%. In the case of the only amphibian we've done today, which is the western leopard toad, which is indigenous to Cape Town, it's severely threatened due to lost habitat. We have struggled quite a bit to get a good assembly for this, but it's our first amphibian, so I'm sure we're going to get better at doing this. And just to show you what it looked like, there's our sticker image of the western leopard toad. So training is a big part of 1KSA, and so we initially set out with a train the trainer workshop to train all of the heads of our labs in library prep and sequencing. That was in 2023. And these members have gone on to run their own workshops across South Africa. So to date, that's been 15 workshops in the last 2 years, and this has reached more than 140 participants. We've supported the AfricaBP Open Institute, the South African workshops, for the last 3 years, with library prep and sequencing workshop. The first 2 at the University of Pretoria, and the one this year was at the Diplomics Training Lab here in Cape Town. This year, we've also started genome assembly workshops, and these are some images here from the first assembly workshop that was run in May, and we ran this again in September as part of the Southern African Open Institute workshop. So a few sort of comments about our open science policies. So employing as open as possible, as closed as necessary. So these are some of the guidelines from UNESCO in terms of applying open science and open access. So we use open method through indicating the methods that we use through the 1KSA website. We keep careful records on REDCap. We note the protocols that we use through our 1KSA Zenodo community, which is open to anyone. The open resources, we enable access to sequencing across South Africa through the 6 different labs. We enable access to the national infrastructure, the CHPC and DERISA. We have a data management plan, which is on Zenodo. So that's openly available for anyone to look at. And we also promote training. In terms of open source, the 1KSA workflows on GitHub, and the link is available from the 1KSA website. Into open data, we have, as I said, the 1KSA data management plan. We have a data access committee to evaluate requests for access to data. We create visibility through species cards and videos, and we request that the data is uploaded onto ENA after 3 years. We also promote open access, so publication in open access journals. Okay, so that's it. I just wanted to finish off, though, by thanking the 6 different labs who have contributed hugely to this project. They're the people that have done all the hard work and continue to do the hard work, and I would just like to thank them here. So thank you very much.
Well, thank you, Dr. Jane Murray, for having some time in your busy schedule. We wish that you stay with us until the Q&A session in a few minutes after the next presentation. Well, now I'm going to be presenting the next speaker, Dr. Bushra Al-Amiri. She's a researcher at the National Institute of Agricultural Research and the Regional Center of Stad. She graduated from the Hassan II Institute of Agronomy and Veterinary Sciences, and then she did both her master's and PhD degree in University of Liège in Belgium. Well, I would like you to welcome with me Dr. Bushra.
Hello, everybody. Thank you for the invitation. I'm very honored to. Didn't hear me well.
Yeah, we can hear you.
I think it's, okay. Thank you. Then I'm very honored to be among with you this morning or this afternoon to share with you a talk about one for me a nice topic for Morocco at least. I will share with you this talk about with the title Beyond Abscesses. Cases lampadinitis in sheep. My talk will start with the context, then the understanding of this disease. I will present an overview from 2 studies that we performed so far I will call it CL for an easy use for this disease within One Health concept. And I will finish with some take-home messages. First of all, could you please? I'm sorry. For those who don't really have some ideas about sheep in Morocco, I have just to underline that the complex topography of Morocco offers diversified ecological conditions and climate types, which influence notably sheep breeds distributions. Successful adaptation to a particular set of environmental conditions allow acquiring specific. Okay. The successful adaptation of particular sets of environmental conditions allow acquiring specific characteristics that vary from breed to breed. From the consensus of 2025, we learned that Morocco have about 33 million heads of livestock population. The sheep represent 23 heads, including 16 million females. And it's the largest population comparing to other species. Among this sheep, we have more than 10 sheep breeds that have been reported so far. The main breeds are Sardi, Boujak, Timhdit, Benigil, and Daman. This population of sheep provides about 35 of red meat in all regions of the country. Sometimes it constitutes the only source of income for farmers, allows the use for pasture and marginal lands or marginal areas, contribute to fertilization of cultivated lands by producing manure, supply industry and handcrafts workers with wool and skin, and wool and milk production are not too important except for some breeds. Sheep are mainly raised for meat production. Sardi breed, coming back now to Sardi breed, the Sardi breed with a population of 2.5 million heads is one of the main local Moroccan sheep breeds, represent 30% of the national flocks. It produced the sacrificial sheep for religious feast, Eid al-Adha, considered among the best breed, and it is the most preferred animal by all Moroccans for this event. Challenges for this Sardi breed, we can find the drought reared in semi-arid areas. We found the nutritional stress, the water stress, the heat stress, and others. Fluorisis, because this sheep, main of these sheep are lived in the area with a high concentration of fluoride. And the last, but not the last one, but not the least, is the Caseus lampadinatus, one of the dangerous and disease. This disease, which I will call it CL, is caused by a Corynebacterium pseudotuberculosis, creating suppurative necrotic inflammation in lymph nodes and organs. It manifests a superficial abscess in external lymph nodes or internal lesion affecting vital organs. In Morocco, CL causes significant losses due to saleability of affected sheep, especially during religious celebrations, The disease impacts meat yield, wool production, leather quality, and reproduction efficiency. The clinical forms of this disease, superficial external lymph nodes are affected, parotid, prescapsular, and prefemoral, but the real dangerous one is the visceral internal organs when it affects lungs, liver, and spleen. We conducted two main studies. The first one called the rearing management, entitled rearing management and its impact in caseous lymphadenitis in sheep. And the second one, longitudinal follow-up of clinical superficial ovine caseous lymphadenitis. And we learned a lot from this, the economic burden in Morocco. It represents 95% of flock that are affected by this disease. 35% are clinical as a clinical prevalence, and 185 are new cases. Among the animal monitored, the animal monitor was 274. Significant losses due to low saleability and reduced prices of sheep with visible lesion, especially during religion celebration like Eid al-Adha celebrations. Economic losses are about 1,500 dirhams and can go to 4,000 dirhams, which means the total price of the animals. And this will depend on the location of these abscesses. We learned also from these studies that there is some practices or management implications, target spring and summer, because we have to focus mainly on preventive measures on April to September, when indoor housing and fattening creates peak transmission conditions. Prioritize young animals because lambs and young sheep require intensive monitoring and improve hygiene during indoor fattening periods. Improve housing, enhance ventilation, reduce density, and proper drainage reduce bacterial survival indoor environments. Vaccination strategy, persistent vaccination prevents infection and limit disease spread in endemic flocks. Critical findings, intensive indoor rearing management during fattening periods is the primary driver of high CL incidence in young sheep. Semi-extensive systems show significantly lower infection rates. The risk factors and transmission, indoor housing, poor ventilation, and high animal density increase transmission risk. Sharing practices, contamination equipment, and a brighter skin provide infection routes. Environmental survival, this bacteria survive 8 months and more in the soil with organic material. Abscess location and patterns, we found that 50% of these abscesses are located in parotid lymph nodes, while only 20% are located in caudal location, and prescapsular nodes are about 23%. Proximal lymph nodes comprise 90% of all abscesses, while caudal transmission in old use likely occurs during lactation, during the contact with infected lambs. High incidence rates, 67% overall incidence. Total abscesses were 335, while the seasonal risk significantly higher between April and September pair to Generi and match. Here, just this plot showed you this distribution among the seasons. And the age, we found that also the age as a critical risk factor. Lambs less than 6 months showed a high risk group. Young sheep between 6 and 24 months, moderate risk, while old sheep more than 25 months showed low risk. Why age matter? Young animals lack immune experience with the Corynebacterium pseudotuberculosis and are subjected to intensive indoor rearing during critical fattening period. Old sheep benefit from semi-extensive grazing, sun exposure, and acquired immunity from previous exposures. What is then the link between this disease and the One Health concept? We should know that CL can infect humans, particularly farmers, veterinarians, and shear workers in close contact with infected animals. Cases of human have been documented. The disease affects small remnants worldwide. It's not limited in Morocco, with a high prevalence in endemic areas. It spread through contaminated equipment, ruptured abscesses, and environmental contamination. The CL survived. The bacteria survives 80 to 210 days in soil with organic material, creating ongoing environmental reservoir that perpetuates transmission cycles between animals and humans. Then for this, really, the link between then this bacteria or this disease and the One Health concept, we should know that there is a big connection between them, because the farmers, the veterinarian, and shear workers in close contacts with infected animals faced infection risk through skin wounds and contaminated the equipment. This information has already said. Then we are really award to improve rearing conditions, enhanced ventilation, reduced density, and better hygienic indoor facilities. Target young animals, focus preventive measures on lambs and young sheep during spring, summer months, and vaccinations program prevents infection in animals to limit this disease spread. For biosecurity protocols, protect workers through proper equipment and handling procedure. Some take-home messages: connected health. CL demonstrates how animal disease affects human health and economic sustainability through One Health challenge. Prevention focus targets young animals during high-risk seasons, April to September, with improved management and vaccination. Collaborative action. Farmers, veterinarians, and public health officials must work together to control CL and protect all stakeholders. With this, I will finish my talk, but I would like to emphasize that recently we really developed a project. You see the number of institutions. We are collaborating with different institutions really to combat this disease, and really try to find a solution. We are universities, research institutes, industrials. We collaborate with one industrial society company, and farmers represented by ANOC, which is the Association for Breeders. and for the animals. Then we hope then with this group we can really combat and succeed to find a solution, vaccine or new threats to really develop new varieties or new vaccines. Thank you for your attention. And before to close, I would like to really acknowledge all the contributors to these studies. We continue to study this topic, and you are welcome if you would like to join this team. Thank you very much.
Thank you so much, Dr. Al-Amiri. Can you please join me here for the Q&A session? I will just check a second time if Dr. Murray is always with us.
Hi, I am here, but I need to go in 2 minutes' time, unfortunately.
Okay, we're so sorry for the delay.
At half past, yeah. Okay.
So if there is only just one question for Dr. Murray in the audience or online, so yeah.
Thank you. Thank you very much, Dr. Shane, for the presentation. And first, I would like to congratulate you as citizens of South Africa for 1,000 genomes for South Africa. I think it's the first example in Africa. Here in Morocco, we are preparing and fighting to get this too, so we can start the 1,000 Moroccan genomes. And I have so I would like your feedbacks about some points, as you are, you are there before us. So maybe there are some lessons to learn from you. And my first questions is, of course, South Africa is very rich in terms of species comparative to Morocco. But I think the choice of species to be sequenced from you, it was mainly for conservation. Here in Morocco, we are insisting on species with economic impact, and they would like to know what is your feedback about this choice. Second point, of course, maybe the work would come later, but if we do sequencing, maybe we hope that the information we get from sequencing should guide us for decision making. And sequencing one species, of course, it's having a reference genome, it's sustainable and very, very good information. But maybe with that, we can sequence at a low level of coverage many other individuals from the same species, so we can have more information using population genetics. What is your point of view about that? And another point I would like to get from you is, of course, we are working in genomics, and we all believe that having the sequences could give us some information, but about other genomic information concerning gene expression, epigenetics, and all. I mean, what we are losing by focusing all our effort on the sequences rather than the other information we can get from other omics techniques. Thank you very much.
Sure. Okay. Hopefully I can remember all of that. But to start off with, in terms of the choice of species, so one of the key things that we said about putting into play for 1KSA at the beginning was that if a genome had been sequenced elsewhere, that we would evaluate the genome assembly. In a few instances, elephant is one example, where there was a short read genome assembly, but it wasn't a very good one. We agreed to allow that sample to be sequenced again under 1KSA. But in most cases, we're trying to sequence new species that don't have a reference genome yet. There are economically important species that maybe aren't necessarily indigenous to South Africa as well. So that's been, our steering committee sits on a monthly basis and will evaluate those nominations on a case-by-case basis. But we also don't want to duplicate efforts. So, for example, there's a big livestock sequencing program out of UNISA, and so we don't want to duplicate what they're doing. but we want to work together as well. So in many cases, I'm having an Oxford Nanopore long-read sequencing dataset and a PacBio sequencing dataset could be advantageous, for example. But in generally, we're trying to choose species where there's a conservation need, where it's linked to training of a PhD student as well. So where the data is going to be used immediately. It's not just going to sit in a repository and maybe be used in a few years' time. I hope I've answered that question sufficiently. And then the second question was around low pass coverage, I think. So one of the impacts, one of the spin-offs of what we've done with OneCaseA is that it's enabled the 6 labs to now offer long-read sequencing as a service. And so in many instances, we've seen it in many cases that the scientists who put forward the genome for sequencing under OneCaseA actually does have an interest in population genomics. And they typically have a grant, and so they can then work with the lab that did that initial sequence run to do more sequencing. And so one of the method development initiatives that we're also considering supporting is trying to figure out what is a good low pass sequencing sort of coverage for a population genomics project. There is going to be a conservation genomics course in December at the University of Pretoria, ConGen, with the University of Montana. And I think, a lot of scientists are hoping to go to that and start learning techniques for population genomics. So they're looking forward to the next step of how they can use the reference genome. And then the third question about RNA-seq and other data sets, other omics data sets. So for the POD5 files, which are the raw files from the genome sequencing run, we store those because we also get methylation information out of those files. And so that is available to be mined should somebody be interested in epigenetics for the species of interest. We also store those POD files just incidentally in case the improvements in base calling algorithms in future, and then that analysis can also be rerun. We are also starting to think about a method development project around RNA-seq for genome annotation, but linking that to doing ab initio gene annotation using AI tools. So yeah, there's a lot in the space. We have to be quite conscious in OneCaseA because we supported by Diplomics that we must create a research infrastructure. We can't do the research ourselves, but we can create an environment in which scientists can apply for grants to take some of the analysis further. Yeah, hopefully I've covered all your questions. Was there anything else?
No, that's okay. Thank you very much. Thank you very much.
Okay.
Thank you, Dr. Murray. So now I'm going to ask the audience if you have another question for Dr. Lamiri.
I'm going to leave if that's okay.
Yeah,
Thank you very much for the session. Okay. Thank you. Okay, bye.
So if there is no question, I will ask a question. Okay. So in the context of genomics and omic data, so how do you think that genomic can help actually either improve the production and reproduction of sheep, even with the disease, or how to prevent this disease and stop this bacteria spreading in human and sheep?
Thank you very much, Dr. Nargis. Like I showed it last slide, we are gathering a lot of institutions, universities, and Professor Bouaouid, he's with us in this project. We hope to really found specific strains for each part in Morocco because we are convinced that, for example, for the cold regions like Ifran, it's not the same, for example, in Sardi where it's very hot. I'm sure that there is many strains for this bacteria, and we would like to target for each part of Morocco, produce a new vaccine targeting specific strain, not having the same vaccine like it is happening now. having the same vaccine that we use over Morocco. Then this is what we will use these tools really to. We are using many tools. We are using, for example, targeting nutrition to really develop a good immunity. We are adding some additives to the animal nutrition. We are targeting vaccines. We are making antibiotics. We are using some plants also, phytotherapy, natural therapy. We are really using all to combat this bacteria, because really it is causing a lot high losses for the breeders. You showed that I emphasized that the losses can, for the salability, can vary from 1,050, 1,500 dirhams to 4,000 dirhams. 4,000 dirhams, really, c'est la valeur totale de l'animal. Then it's really pity for these farmers who are struggling they're working for the fattening period. Pour l'engraissement, they are really struggling to get better and better productivity, money also, the income, to improve their income. But unfortunately, with this disease, because je vais peut-être parler en français, c'est plus facile pour moi. En tant qu'acheteur, vous pouvez acheter l'animal, mais le lendemain, vous le laissez chez l'éleveur. Le lendemain, l'éleveur vous appelle, je viens de voir un abcès qui apparaît, donc vous allez lui laisser cet animal, d'accord? Donc, il est vraiment perdant. Donc, ça cause vraiment une énorme perte économique donc, pour les éleveurs. Donc, c'est pour ça on s'est dit donc on va se réunir donc, pour lutter et combattre cette maladie. C'est l'une, vraiment, parce que toutes les collègues ce matin ont bien présenté de belles présentations sur le One Health Concept. Malheureusement, cette bactérie, elle peut atteindre aussi les organes internes. Et ça c'est vraiment un grand problème. D'accord? Dans les abattoirs, vous allez rencontrer beaucoup d'animaux qui sont atteints de cette maladie, mais ni l'éleveur ne le croit, ni les policy makers. Mais si tu le dis, par exemple, pour les décideurs, ils ne vont pas te croire. Donc, ils vont mieux vraiment montrer avec chiffres à l'appui les nombres de cas qu'on peut rencontrer aux abattoirs, parce qu'on travaille aussi aux abattoirs. Et les éleveurs eux-mêmes, quand vous voulez les convaincre que la bactérie peut vraiment atteindre l'homme, ils ne vont jamais vous croire. D'accord? Alors qu'il y avait des cas vraiment de gens qui étaient infectés par cette maladie. D'accord? Et le problème devient trop grave. Quand c'est vraiment sous-maxillaire, ça diminue la production. Quand c'est interne, ça peut vraiment aller jusqu'à la mortalité de l'animal. D'accord? Donc, c'est vraiment une perte économique, mais qui est silencieuse. Vraiment, pendant la fête de l'Eid al-Adha, nous, on ne sent pas cette peine que l'éleveur elle endure pour nous livrer un mouton. Nous, on sait juste négocier le prix, il faut diminuer les prix, alors qu'il y a beaucoup de choses derrière. Merci.
Thank you so much, Dr. Lamiri. Is there any question? Is there any question online, please?
Yeah, I don't think we have a question. It's more of a comment. What's doctor's comment on the phylogenesis of Corynebacterium, especially in the sense of its endemic? Does it have any dynamics in terms of how it is or may be spread from Moroccan region to the other? Just one question here on Zoom.
I'm sorry, it's not just only for Morocco. It's spread over the world. We can find it in Australia, in Europe. Really, it's perhaps the strains are not the same, but the disease is really over the world. They have no limits, and the solutions are till now limited, for example, to some vaccines that are good or not good. Then in Morocco, we just have one we are trying also in these projects, we are really targeting to try this vaccine also and to develop others, really.
Well, thank you so much, and thank you to our speakers for their insightful and engaging presentation. So we begin with the One KSA project, which showcased the remarkable efforts of diplomats in decoding biodiversity and building genomic resources and capacity in South Africa. And we're hoping that we have the same in Morocco. And then we saw the case study of this disease that is impacting sheep, but also human. So this is a real case study where we're needing these genomic and omics technology to prevent this disease and improving also livestock health, but also human. So thank you all for being here. And we're having the lunch break for 1 hour. Yeah, so we're having a 1 hour lunch break, and then we're gonna meet you at the session 3 of this day. Thank you so much.
BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
I can't believe it's been 10 years since Illumina Biotech West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within high IT.
It feels just like yesterday. I joined INCABA in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for INCABA BioTech, our creation and existence has benefited considerably from experience and mentorship of INCABA BioTech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At INCABA BioTech Limited, our core values are quality, customer focus, integrity, teamwork, and others.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from PacBio West Africa Limited, Ghana. My name is the manager for PacBio Ghana.
Good morning, I am Momodu Dabo from Mali.
I extend our warmest congratulations to the 10th anniversary of PacBio Nigeria branch. Congratulations to our counterparts and our colleagues in PacBio Biotech Nigeria for their 10th anniversary.
Happy 10th anniversary year to all INCABA Biotech staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such, we see the need for improvement. And in the future, we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA, as a company, INCABA BioTech West Africa, has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary, INCABA BioTech West Africa.
Long live INCABA.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary, INCABA BioTech West Africa. Cheers.
As we look ahead, INCABA BioTech remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed the enterprise, including the setup of a genome lab that provides summer sequencing services in Ibadan. And we look forward to add other genomic services like next generation sequencing using biosequencers like we do here in Pretoria. As INCABA BioTech, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana, and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
MGI-Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since Illumina BioTech West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within IITA.
It feels just like yesterday. I joined InCaba in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for InCaba BioTech, our creation and existence has benefited considerably from experience and mentorship of InCaba BioTech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At AfricaBP Biotech Limited, our core values are quality, customer focus, integrity, teamwork, and others.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from PacBio West African Limited, Ghana. My name is Yusuf, the manager for PacBio Ghana.
Good morning, I am Mamadou Dabo from Mali.
I extend our warmest congratulations to the team and Ministry of PacBio Nigeria branch. Congratulations to our counterparts and our colleagues in PacBio Nigeria for their 10th anniversary.
Happy 10th anniversary year to all in BioTech staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such, we see the need for improvement. And in the future, we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA, as a company in BioInformatics West Africa, has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary in BioInformatics West Africa.
Long live in Kaba.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary in BioInformatics West Africa. Cheers.
As we look ahead, BioInformatics remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed the enterprise, including the setup of a genome lab that provides summer sequencing services in Badan. And we look forward to add other genomic services like next generation sequencing using biosequencers like we do here in Pretoria. As in AfricaBP, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana, and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since Illumina BioTech West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within high IT.
It feels just like yesterday. I joined INCABA in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for INCABA BioTech, our creation and existence has benefited considerably from experience and mentorship of INCABA BioTech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At INCABA BioTech Limited, our core values are quality, customer focus, integrity, teamwork, and others.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth, while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from PacBio West African Limited, Ghana. My name is Yusuf, the manager for PacBio Ghana.
Good morning, I am Mamadou Dabo from Mali.
I extend our warmest congratulations to the 10th anniversary of PacBio Nigeria branch. Congratulations to our counterparts and our colleagues in PacBio Nigeria for their 10th anniversary.
Happy 10th anniversary year to all Inkaba Biotech staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such, we see the need for improvement. And in the future, we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA, as a company in PacBio West Africa, has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary Inkaba Biotech West Africa.
Long live Inkaba.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary Inkaba Biotech West Africa. Cheers.
As we look ahead, Inkaba Biotech remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed an enterprise, including the setup of an own lab that provides summary sequencing services in Badan. And we look forward to add other genomic services like next generation sequencing using biosequencers like we do here in Pretoria. As Inkaba Biotech, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana, and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
Some cities across the world are sinking. These pictures give you an idea of what's happening. There are both natural and human reasons for this. Climate change, too much
BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since Illumina Biotech West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within high IT.
It feels just like yesterday. I joined InCaba in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for InCaba Biotech, our creation and existence has benefited considerably from experience and mentorship of InCaba Biotech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At InCaba Biotech Limited, our core values are quality, customer focus, integrity, teamwork, and all that.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from PacBio West African Limited, Ghana. My name is Yusuf, the manager for PacBio Ghana.
Good morning, I am Mamadou Dabo from Mali.
I extend our warmest congratulations to the team and Ministry of PacBio Nigeria branch. Congratulations to our counterparts and our colleagues in PacBio Nigeria for their 10th anniversary.
Happy 10th anniversary year to all Inkaba Biotech staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such, we see the need for improvement. And in the future, we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA, as a company in biotech West Africa, has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary Inkaba Biotech West Africa.
Long live Inkaba.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary Inkaba Biotech West Africa. Cheers.
As we look ahead, Inkaba Biotech remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed the enterprise, including the setup of an own lab that provides summary sequencing services in Badan. And we look forward to add other genomic services like next generation sequencing using biosequences like we do here in Pretoria. As Inkaba Biotech, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since AfricaBP BioTech West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within high ITA.
It feels just like yesterday. I joined InCaba in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for InCaba BioTech, our creation and existence has benefited considerably from experience and mentorship of InCaba BioTech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At InCaba BioTech Limited, our core values are quality, customer focus, integrity, teamwork, and others.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth, while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from PacBio West African Limited, Ghana. My name is Yusuf, the manager for PacBio Ghana.
Good morning, I am Mamadou Dabo from Mali.
I extend our warmest congratulations to the 10th anniversary of AfricaBP Nigeria branch. Congratulations to our counterparts and our colleagues in PacBio Nigeria for their 10th anniversary.
Happy 10th anniversary year to all AfricaBP staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such, we see the need for improvement. And in the future, we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA, as a company in PacBio West Africa, has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary in PacBio West Africa.
Long live in Kaba.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary in PacBio West Africa. Cheers.
As we look ahead, PacBio remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed the enterprise, including the setup of an own lab that provides summary sequencing services in Badan. And we look forward to add other genomic services like next generation sequencing using biosequencers like we do here in Pretoria. As AfricaBP, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana, and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since Illumina Biotech West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within high IT.
It feels just like yesterday. I joined INCABA in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for INCABA Biotech, our creation and existence has benefited considerably from experience and mentorship of INCABA Biotech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity,
Now we will
excellent
start
leadership, and help from
third
God.
session
At INCABA
of this
Biotech
first
Limited,
day
our
of
core values
workshop.
are quality, customer
And the third session will be on bioinformatics and genomics technology for agri-environment. We will have during this session 3 speakers, and we will start directly with Prof. Zainab Rashid, that will be talking about empowering Africa, the role of genomics core labs in fostering scientific sovereignty and sustainable development. So Professor Zainab Rashid is the lead of the bioscience platform at UMCXP Core Lab. She obtained her PhD in 2016 from Coast University in Saudi Arabia in genomics, and she's now the lead of the core lab in UMCXP. So now we'll start first talk.
Thank you very much. Hello, everyone. Assalamu alaikum. I'm Zainab Rashid. I'm the lead of the Biosciences Core Lab at UMCXP, and it's a pleasure to be amongst you today. I'm not here today to talk about scientific results, but instead I'm here to preach for the importance of core labs in Africa. And my presentation is entitled The Role of Genomics Core Labs in Fostering Scientific Sovereignty and Sustainable Development. Well, I will start my presentation by some shocking stats and a reality that deserves our attention and, in my opinion, our action as well. A recent evaluation in 2024 revealed that data included in the National Human Genome Research Institute EBI-GWAS catalog found that only 2.4% of the individuals included in the catalog were of African ancestry. And this despite of the immense and important genomic diversity in Africa, and despite Africa being an important point in human genomic evolution. And probably we'll ask the question, is this trend improving over time? That's not true. The proportion of the genomic studies that were conducted in Africa was 3% in 2016, and now it's 1.1%. So we're not improving, quite the contrary. And on the other hand, we have an increase in genomic studies that have been conducted in individuals of European descent, which was 86% by June 2021, and which is increasing from 81% in 2016. And this suggests that in spite of calls for more diversity in genomic studies, the gap continues to widen. Now, this points towards the widening inequality in who benefits from genomic science, who defines its priorities, and who controls its output. And if you do a simple exercise and you ask your AI bot what's the percentage of genomic research from Africa in 2025, the answer is not going to be any different. It's around 2%. But what's interesting is that the answer includes the following information. This underrepresentation reflects limited funding, infrastructure constraints, and data sovereignty challenges. And this despite the fact that we are around 18% of the world's population, and we generate only around 2% of the genomic research. Again, this raises a red flag. We are severely underrepresented. Now, this underrepresentation comes at a cost, multiple costs, in fact. And most importantly, a decrease in visibility for African researchers and increasing disparities in access to technologies, genomic technologies, especially in fields that disproportionately affect the underrepresented populations. And in this case, maybe for Africa, we can cite neglected tropical diseases and infectious diseases. A good example can be the leading causes of death in Africa. Around more than 30 years ago, these were maternal, neonatal, and communicable diseases and nutritional diseases. Now, 30 years later, we are still dying from the same kinds of diseases, whereas in high-income countries, the leading causes of death are quite different, like cardiovascular diseases and neoplasms. So this points towards different interests and areas of need for the use of and application fields of genomics. Genomics core facilities can help tackle health problems that disproportionately affect Africa, such as the neglected tropical diseases, etc. And research focused on African populations leads to the identification of unique genetic markers that are ubiquitous to our populations, enabling health equity and access to precision medicine. And here we're citing some problems related to health and disease, but the same is true for agriculture and the problems that disproportionately affect Africa in the fields of agriculture. Here we can cite, for instance, drought and climate change problems, soil salinity, soil degradation, post-harvest problems, and indigenous species of Africa. Now, the unique challenges of the continent need adapted solution, and I advance that these solutions need to include the following: adapted equipment installations in specialized research centers and infrastructures, proximity hubs for preparedness, and we cannot have a better example than the COVID-19 when this happened a couple of years ago. The development of comprehensive training programs for capacity building and interactive workshops like the one we are in today on relevant African challenges for continued learning. However, often researchers are obliged to send their samples abroad. And here I say obliged because we wouldn't send samples if we do have the adequate infrastructure to treat our samples ourselves. With sending data abroad comes another problem, that of metadata and data use. And here I'm not talking about the outreach of selling identifiable data, but instead I'm talking about sharing aggregated, de-identified genomic data for research purposes, including drug discovery. Even that can be a problem because we sign, sometimes the patient signs ambiguous consent forms that we do not correctly understand. There are sometimes problems in permitting access by for-profit entities. We do not know how the data and the samples are going to be used later on, and transparency in how the data is going to be utilized later. So now that I presented and I preached, as I promised, for the importance of core facilities, let's have a look at the opportunity gap that we have now. What can core labs and core facilities enable us to do in Africa? First and foremost, the scientific sovereignty. Reducing dependency is a very, very important advantage of having our own technologies and producing our data ourselves, allowing the ownership of locally generated data and metadata, influencing the creation of new data policies, because we need to understand what we have circulating in order to adapt our policies, and reducing risks of biospiracy. This is a danger that we cannot oversee. The capacity building for young African scientists, for developing local expertise, retaining the talent, and mitigating brain drain. There is also a great economic growth potential by the rising demand for genomic data and the increased genomic research in precision medicine today. There is also a very important economical point, which is that of reducing the currency hemorrhage through local infrastructure. So this way we'll be retaining our own wealth, what we call like building internal wealth loops, in addition to attracting regional collaborations and retaining our funds. If we fund our research, we should be able to retain those funds as much as possible within the continent. There is also a strategic value by addressing health disparities and promoting health equity and driving national genome projects, like for instance, species conservation and precision medicine and public health strategies. In addition, of course, to the Sustainable Development Goals 3, 2, and 9. So what is a genomic core laboratory? So instead of asking this question, I think that the question that should be asked is, what is a good genomic core facility? A good core lab is a well-equipped, professionally managed facility that delivers standardized, reproducible, and high-quality scientific analyses and services, and it should be a pillar for the biomedical and research ecosystems. Now, the types can be like either research-focused, clinical grade, or hybrid mode, and we, especially in Morocco, we need both. We need to lay on a few pillars, such as technical excellence, standardization, having expert staff, training and education, continuous training and education, and having the newest multidisciplinary technologies possible. In genomics, one size does not fit all, and a very good example is that of weapons. Depending on what we need to tackle, what we need to do, we may need a knife, as we may need a missile. And for that, we at the UM6 Collab got equipped with a high range, a very wide range of sequencers, depending on the type of sequencing that needs to be conducted and the size of the samples in the project. Our mission depends, and I believe that this should be the same for other collabs across Africa as well. It should be first and foremost promoting sovereignty and enabling sovereignty, because by producing our data, by having our own results and keeping our currencies as well, hard currency as well, we keep our sovereignty and we're capable of protecting it. And also positioning our universities and empowering our continent and ensuring capacity building as well. So, of course, the main reason behind the existence of the core lab is to support research, to generate revenue, and to enable capacity building within the university at least. But with that, we need to maximize our impact. And this is something that, again, I'm preaching for, and we also need to promote as much as possible. The first thing is the knowledge transfer. It's very important to share protocols and project and co-develop projects together. This is something that goes beyond the individual interest of a researcher or the interest of a university, but goes for public intelligence and having some themes of research that will benefit at the same time the researcher, the university, but also the whole continent. There's also the importance of impact-driven solutions. There are some solutions that we need specifically for Africa, and these can be identified and tackled only if we can discuss them openly in areas like research, health, and agriculture. These will be the priorities that the core labs need to tackle. There's also the importance of training human resources. This is another pillar that should be always at the forefront of the interests of any core facility. And a very good example is the training sessions that will be held after this workshop. This is something that we at the M6 Collab are working with Illumina, but working with other providers, we should discuss other sessions and make sure that we train youngsters in across Africa to make proper use of these technologies and understand the ins and outs of each of their technologies. And finally, the servicing is something that needs to keep competitive pricing and favorable to AT. I think that needs to be implemented, kept, and we have to continuously develop it. So as I started with some shocking stats, I would like to end with a shocking story. And this is about preparedness before it's too late. In the aftermath of the devastating Nepal earthquake in 2015, there has been around 9,000 lives that were lost. Nepal's forensic infrastructure was not adequately prepared and equipped to handle large-scale DNA analysis, despite the importance of molecular biology and sequencing to properly handle such cases and identify victims. So the country faced limitations in laboratory facilities and the trained personnel as well, and the necessary equipment. And hence, in order to identify the corpses, alternative identification methods were used, like forensic odontology, personal belongings, or even just visual identification. This really points towards the importance of having already established laboratories and being prepared before an event happens. So enhanced forensic infrastructure, but also scientific infrastructures can help in such cases, and investing in forensic laboratories or genomic laboratories and training personnel, acquiring necessary equipment can bolster a country's capacity to conduct DNA analysis during disasters, just as an example. So in conclusion, again, I'm preaching for the importance of genomics core labs in Africa, and I just wanted we really need to have multiple core labs in Africa for our own sovereignty. These are more than just scientific facilities. They are key drivers of sovereignty, economic stability, and empowerment. And by investing in core labs, Africa can reduce dependency on foreign services, retain its wealth, and build the scientific capacity needed for a sustainable future. And with that, I come to the end of my presentation, and thank you for your attention.
Thank you, Professor Rashid, for this nice presentation about core labs. Really, very good infrastructure for genomics. And through this presentation, we can say that the genomics in Africa is in safe hands in the future. We have really exciting infrastructure. Thank you. And now we'll go to the next presentation by Professor Walid Fouad. Yeah. So Professor Walid Fouad will present a talk about advanced large identification, the role of sequencing tools, databases, and culture strategies. So Professor Walid Fouad is an associate professor of plant biotechnology and biology department and biotechnology program at the American University in Cairo. So he holds a bachelor in horticulture science and master in vegetable crops in genetics from Cairo University, as well as PhD in horticulture science, specializing in plant molecular and cellular biology from the University of Florida. So the floor is yours.
All right. Thank you so much. First of all, I would like to thank the organizers for the invitation. It's a very good opportunity to know, to be exposed to the research that's being conducted in Africa and in Morocco, indeed. So my talk today a little bit on algae diversity. So Egypt, we have like the Nile River, which is a large body of fresh water in Egypt, as well as the Red Sea and the Mediterranean Sea. So we have a lot of water resources that can be explored, and they provide a lot of diversity that we can learn from. So today's talk is mainly about, it will be mainly divided, I'll give you a brief introduction about the talk, about what I'm going to talk about. This one is better. All right. Okay. About the algae and the importance of algae. Then I will I'll talk mostly about the fresh water, microalgae diversity. We'll be covering some of the tools that are being used to understand the diversity of algae in the fresh water. There's one of the approaches that involve an enrichment of the agricultural diversity by media. So what is the impact of the enrichment on the diversity? Following by the reason that's being used for identification using the molecular tools and the database that are actually lasted. Then I will give you a glimpse on some of the ongoing work related to the Red Sea marine algae diversity, which is still ongoing work. So why diversity of algae? Algae, as we know, is a primary producer in the freshwater habitats. They fix carbon dioxide, produce organic molecules, and they live in a very close harmony with bacteria. In most of the time, they do have some symbiosis with bacteria. And in return, they provide the bacteria with organic molecules, different types of organic molecules. And in return, some bacteria provide algae with vitamins. Sometimes it's very challenging to isolate agrobacterium because of their dependence on some of the, so it's difficult to isolate algae because of their dependence on bacteria. Algae also serve as a primary producer for a much more complex web chain, including the phytoplankton, the zooplankton, and fishes and birds, to provide an example. Therefore, the algae diversity and the balance of the diversity within the ecosystem, if it's altered either by shifting toward one species or another, or reducing its diversity, can have a huge impact on both bacterium diversity within the ecosystem as well as the whole food chain. So it is important to understand algae diversity. But another important aspect that you can look at is the isolation of algae species. And that comes for the potential of algae species for multiple downstream application. Whether we're talking about microalgae or macroalgae, they can be used, their biomass can be used as to produce a lot of byproduct, including vitamins, fatty acids, pigments, antioxidants, which is really becoming popular. They can be used for biofuel production. There are a lot of work ongoing for the microalgae biofuel production. And indeed, they serve as several types of industries, including pharmaceuticals, food chain, and as well as animal feed and so on. So it's important to identify species to understand their potential. How do we identify algae? There are different approaches to identify the algae species. You can go for their biochemical and chemical composition, and that's usually looking into the pattern of the carbohydrates they have, the pattern of the pigments, what type of pigments or protein and lipids. So that's one of the mechanisms that we can identify algae. We can directly identify algae from the environmental samples by visual assessment under the microscope, following some identification key relative to their structures. We can also use molecular tools for identification. So any environmental sample can be processed in these three approaches. Molecular tools involve different DNA barcodes, ribosomal RNA, whether we're talking about 16S or 18S, 23S, or gene-specific tools, so like RPCL or the CUI. So all of this has been used to identify algae. Another approach which we were really interested in, which is actually must if you want to isolate species. You do an enrichment for the algae population within the environmental sample, and the enrichment usually conducted on some sort of media. The two most popular media for algae growth is the PG11 and the PPM. Following the enrichment for whatever period with whatever media composition, you can identify the algae later on by either by chemical, molecular, or under the microscope. So this is the focus of my talk about the freshwater algae. So when it comes to the freshwater algae, what we did, we collected water samples from the River Nile, and we collected all the water mainly from the greater area region. So there is small difference on the location when it comes to their differences. They are very close proximity. So we maintained we inoculated the culture with four different media, the PG11, PPM, and modified media. We looked into the composition of both media, and we found that there is a big shift for some of them having high nitrogen compared to others, and so on. So we formulated another media. We call this modified media. Then we used also another type of media called MS media, which is very popular for plant tissue culture, which is rich in nitrogen. So we maintained the algae under this with this media separately. Then we identified them morphologically during the we were looking at the population dynamic throughout the 14 days, and we did DNA extraction at the end and sequenced it for 3 regions, 2 regions within the 16S and 1 18S V4 region. Then it was the data processed through 3 references database. We used Selva, which is very popular. We used also Green Gene and the PR2, which is known to have very good information, the database about the eukaryotic algae. So if you look at the differences between the species, the 3 databases, you can see Selva has both 16S ribosomal RNA and 18S ribosomal RNA, while Green Bean and PR2, both of them have only the 16S ribosomal RNA. So when we identified in Selva, as I said, Selva is mainly eukaryotic database. So we used the chloroplast 16S ribosomal RNA for the identification of eukaryotic algae in the Selva. So if you look at the differences between morphological versus molecular identification, we were able to identify around 37 species or genus using the microscopic, just direct, typical, traditional approach. 20 of this genera have been identified also by the molecular approach. So on top of this 20, the molecular DNA barcode identified 67 genera. So that revealed the power of the DNA identification or the molecular tool for identification. Interestingly, I mean, you will expect to see whatever genera been identified by the molecular to be present morphologically. It's easy to, but however, that was not the case. So, for example, some of the genuses, for example, the Bistrems or the Tetrodron, both of them, and all of the species, very easy to identify morphologically, yet they were missing from the molecular identification. On the other hand, you have some other species that were easy to identify, what was actually very powerful to identify using the molecular tool, like many of the diatoms and many chlorellas and so on. So that shows us that both of them are very important, morphological important and molecular important. Then we wanted to see the difference of the media. I mean, would some media enrich certain species over another or not? And that was really actually obvious. You see, the most common common, let's see, can I have a point? No, no pointer. No pointer? No pointer, okay. Anyways, so if you look at the panel A, you see that the PG11 and the PPM, both of them are the most common species media we used, and both of them they failed to maintain or to enrich for at least, I would say, 13% of the algae that were identified by the other media. So integrating multiple media for the enrichment was actually a very successful approach to give better spectrum of the species diversity. At the molecular level, you can see more or less the same percentage. 13% of the species would have been lost if we only used one or even the two popular media together. After that, we moved to, and you can sell that when you do this is the box plot that shows you the impact of the media on the enrichment. This is mainly using eukaryotic bacteria. And you can see that in spite of the source of the DNA, source of the water, which is represent the diversity within the environmental sample, you still have the media has a bigger impact on the selection of certain species over another. Then we move to the database. As I said before, the PR2 and the green gene, both of them have the prokaryotic 16S database. So here, when we use the V1, V3 region to identify the cyanobacteria, for example, and the three databases, we did not really miss much. I mean, both regions, in other words, both regions were actually successful in covering the good spectrum of the diversity. However, each database was to some extent giving us different one or two species. There is no single database that kind of give us the whole spectrum. You can see they complement each other. We have, for example, around maybe 18% of the species would have been missed if you only used Silva. However, adding green gene and the PR2, both of them give us a better picture about the diversity of the algae. When it comes to the molecular level, though, or when it comes to the eukaryotic level, though, the picture was completely different. So here we used to identify the molecular by two approaches. Number one, either the 18S in the Salvia database, and also we identified with the 16S representing the chloroplast genome within the eukaryotic bacteria. Within the eukaryotic algae. And you can tell here that we have, like, if you only use the Salvia for eukaryotic 18S, you would have lost something like maybe 30% of the species. By, however, integrating the two different regions was also complementing each other. If you only used one of the 16S region with one of the 18S region, you would have lost around 10 to 11% of the species. So the take-home message here is literally you have to integrate multiple databases. So another important thing is which version of the database, and that's actually was something that we actually we learned early on. Remember when I tell you about pedestrians that were not identified by molecular, however it was identified morphologically? That was when we done the work with the older version, and this is the one that was available back then, which is the Salvia V version 132. Now the 138 is being released. So when we run the same data again on the newer version, we were able to identify one of the species that were not identified before. However, when you look at the dynamic of the database and the reannotation of the species and the genus, and sometimes some ambiguous genus are not really grouped into the not really labeled at the genus level. So the dynamic of the database is something that also has to be considered. Which database, which version of the database, what is the power of each database, and how much can it actually resolve. So the previous results give us on the Salva, the older version, 27 genus. Right now, we only have 8 genus. Some of the genuses that were actually present, genera that were present before, will actually move to another classes. So that actually generate kind of something that everybody should be aware of when it comes to the database search. So, what is the lesson learned from the freshwater algae? Number one, enrichment has a big impact on the diversity. If you take the approach of the enrichment, you have to use multiple media. If you know a specific algae that you would like to isolate, so maybe you can go for a specific media condition that enrich for this particular algae. Then which region to amplify, to sequence? Are we talking about 16S? Maybe the best approach would be using the multiple locus sequence type, because that gives you high level of confidence on the on the identified algae. And the same thing is the database. Which database to use? Don't stick to one database. You have to based on what our experience, using multiple databases is actually beneficial because that gives you better resolution for the diversity. I have to mention that Green Gene 2 is right now provide the new taxonomical framework, and that with very important, with very consistent lineages of the species. So this is for the freshwater. Marine algae is another work that another research that we're working on in the Red Sea. So if you look at the Red Sea, we selected 5 locations within the Red Sea, and we wanted to see the location differences versus seasonal differences of the marine algae. We'll be focusing mostly on the macroalgae. So we collected, we covered so far 2 full seasons. We don't have data for all of them, but I'm going to give you data about 1 season only, which is the summer 2023. So basically, we collect the data, we identify morphologically. Macroalgae is relatively easier than the microalgae when it comes to the identification. Do DNA extraction, sequencing, and the data analysis. This picture this is heat map that shows you the relative abundance of different species across different locations. So forget about it, you don't have to get into it. But in total, we have around 41 species that have been identified morphologically. When we did the molecular identification, actually we discovered another 31 species that were overlooked. It's important to mention that most of the molecular identified species are mostly endophytes, so it will be very hard to identify morphologically. These are some species that living within another macro species. It was very obvious to see differences between the location, even though they are not very far away from each other. If you look at the map, so except for Zaffarana was very far north. The other species, the other location, relatively close to each other. However, we found big differences. We found, for example, 41 species in Zaffarana compared to 30 in the range of 30 of the other regions. Interestingly, when we compare the relative abundance of the species across location, for example, start with Hurghada that has 20 genera. Out of this 20 genera, 10 were actually only identified on Hurghada. If you look at Makadi, we have 17 genera species been identified in Makadi. 6 of them are unique. There are some species that you can find them across the 5 locations. Some species are shared between 2 locations and so on. This pattern we've seen it with the red algae. We've also seen it with the brown algae and the green algae. When you see, you also have some species that are unique to a particular location, while others are actually shared with 1 or 2 or sometimes the 5. We've seen also a lot of seasonal variation. I don't have data for that presented with me today, but there's a significant dynamic of the algae diversity, red, brown, green, within the same location from one season to another. So hopefully this will be finalized, and maybe we can share this in the future. This is some of the species that's been identified, and this is some of the species that abundant among all the locations. And this is some species that are unique to some location. For example, the champia. The champia is specific mainly in Makadi, present all throughout the whole seasons, and so on. Interestingly as well, we have the first report of the Pradia, which has never been reported in the Red Sea before. So that was reported or identified in June 2025. And hopefully we'll discover more dynamic about the algae, macroalgae in the Red Sea. With that, I'm going to wrap up my presentation. So it's important to continuously monitor the macroalgae and diversity in the Red Sea. We've seen big shifts. We've seen some species that never been reported there. We've seen shift from some species that used to be mainly in the southern part of the Mediterranean of the Red Sea, now reported in the northern part of the of the Red Sea. So you see a lot of dynamic and shift in the diversity. So it is important to monitor on a regular basis. As a molecular tool is very powerful when in identifying the endophytes. However, morphological identification is still very, very important. So to wrap it all together, it's very important to create multiple approaches to accurately support the discovery or the identification of algae, and relatively to be able to assess the biodiversity and make an informed decision about the conservation later on. That's it. I would like to acknowledge the funding. All the research was done mainly by internal fund from the AUC for graduate students. So both of the work that are represented actually work of graduate students. The freshwater work was conducted by Amal Badr, is the second from the left. She finished already, and now she's doing a PhD in sorghum. She shifted from algae to plant. The Red Sea work was conducted by the second person from the right, which is Iman. Iman is really, she is passionate about going to the Red Sea and collecting, and she's really passionate about this work. She's the one who's introduced it to the lab, and she's doing a PhD, and hopefully she will be done soon. Other members of the lab working on different projects, including sorghum, millet, tissue culture transformation, physiology, and so on. And with that, I would like to conclude my talk.
Thank you very much, Professor Walid, for this exciting presentation about algal biotechnology, focusing especially on algal species identification using databases and especially enrichment using some media. And that shows again the concept of One Health. Every living organism on the earth is important for conservation of the concept of One Health. So thank you very much again.
Thank you.
And now we'll go to the next presenter, who is, I think, online. So Professor Ashraf Allali will give us a talk about genomic language models and their role in decoding genomic signals. So I will give just a brief introduction about Professor Ashraf Allali. He is an associate professor at the College of Computing at UM6P at Ben Gurion, where he leads the bioinformatics laboratory. So a laboratory that aimed at advancing UM6P research goals in agriculture, environment, and health. His research involves integrating experimental data with computational techniques, primarily focused on computational genomics and metagenomics and identification of genetic markers. So the floor is yours, Professor Allali.
Thank you. Thank you, Sisman. Can you hear me?
Yeah.
Let's see if I can share my screen successfully. Can you see the screen?
Good.
Okay. Assalamu alaikum. Good afternoon, everyone. And thank you for being here. Would have loved to be there with you, but unfortunately, I had some circumstances today that prohibited me from making the trip. But nevertheless, I am here. My name is, for those who don't know me, Ashraf Allali. And today I will be sharing a talk titled Genomic Language Models and Their Role in Decoding Genomic Signals. This presentation will explore how artificial intelligence, specifically transformer-based models, similar to those behind ChatGPT, for example, can be used to read and interpret the language of life, which is DNA. So over the next few minutes, we'll talk about the concepts of treating DNA as a language, how genomic language models work, and what they can tell us about biology and disease in general. So let's start with the basic question. Why would we treat DNA as a language? So the genome contains far more information than just the sequence itself that makes the proteins. It also encodes regulatory signals, structural patterns, and evolutionary conservation, all hidden in the long strings of what we all know, the A, C, G, and T. So traditional bioinformatics models have captured the short range of dependencies, with maybe 20 to 30 bases long, but they often miss the bigger picture. This is where language models come in. By contrast, language models, they excel at capturing the context, meaning they can learn that the meaning of a word, or in this case, the meaning of the base, which depends on many other faraway bases in the sequence. So by viewing DNA as a structured language, we can start uncovering its grammar and syntax, the rule that govern how life's code operates. A genomic language models, or short GLM, works much like a text model in language processing models, such as the well-known ChatGPT, for example. Instead of words or sentences, it reads sequences of nucleotides, A, C, G, and G. So these models are trained using next token predictions or by masked language model, which I'll describe in a little. And so either these two approaches, and the idea is the model will try to predict the missing bases. It will try to predict the missing bases on the surrounding based on the surrounding context. So from that process, the model learns what's called embeddings. These are numerical representations that capture the biological meanings and context of a sequence. So in short, a genomic language model translates DNA into a mathematical language that computers can use to reason, or they can use in their reasoning. Now that we have these models, what do they actually learn? What do these models learn? They actually learn quite a lot. They pick up on motifs, repeated patterns that correspond to biological signals like transcription factor binding sites, for example. They also learn dependencies between distant regions. For instance, how a promoter affects an enhancer that's thousands of bases away. They can pick up on those as well. When we visualize their attention maps, we can see that the models focused align with known functional gene regions, meaning it's actually learning real biology, not just memorizing the sequence. So let's look at some landmark models in the field. The first that came out was called DNA-BERT, which adopted the BERT architecture that is used by ChatGPT to DNA and showed that sequence context can be learned effectively. So this was our first approach into doing this. Then, of course, from that came a lot more. APG-PT came afterwards. It integrates epigenomic signals, such as chromatin accessibility and histone marks, for richer biological understanding. Another one called gene narrator, like a generator. So this pushed the idea even further with long context modeling. And then came Janus DNA, which introduced a hybrid training approach combining autoregressive and masked learning. The latest one actually is EVO, which focuses on integrating evolutionary conservation signals across genomes by enhancing the model's ability to detect functional and regulatory elements conserved through evolution. These innovations show how quickly this field is evolving, blending the ideas from natural language processing and systems biology together. So what makes GLMs, or genomic language models, powerful is their ability to decode regulatory logic. I've said that, but it's kind of like the basic idea of what they have were able to give us. They can identify promoters and enhancers, which control the switches of genes, and recognize transcription factor binding motifs. Beyond that, they can infer higher order patterns like chromatin organization or 3D structure, genome structure. Essentially, we're given the model, the genome, and it's learning to interpret how different regions talk to each other within this genome. So that's the training. It's as much of like, you know, an NLP and a natural language processing model would actually learn the language of speaking. It's actually learning not only the language of DNA, but how do you use it to communicate. Another key application of genomic language models is variant effect prediction. So by comparing embeddings of a wild type sequence, for example, with a mutated version, we can estimate how much change might affect gene regulation or splicing. This is vital for clinical genomics, of course. For example, prioritizing which mutations might cause the disease. If anybody's worked on this, you know that this is a very, prioritizing which mutations to filter, because you'll be looking at thousands and hundreds of thousands, is a big help for further downstream analysis. However, it's not without challenges, of course. Rare mutations, tissue-specific expression, and the need for more diverse data sets is also, remains in active research areas that will help this part be, even more applied. Another aspect beyond analysis, genomic language models are now being used in design. So we can actually use them to generate entirely new DNA sequences that can perform specific functions, kind of like optimized promoters or enhancers or codon efficient genes. So much like we're using ChatGPT to generate, text, we can actually use them to generate sequencing. Of course, these predictions must be validated experimentally, but this computational to experimental loop is accelerating synthetic biology like never before. So DNA, of course, doesn't act alone. It operates within a complex 3D and biochemical structure environment. So factors like chromatin structure, transcription factor bindings, and DNA methylation all influence gene regulation. That's why models such as APGEPT integrate these multi-omic layers alongside DNA sequence, allowing a more biologically realistic prediction. So it's not just about reading the letters of DNA, it's more about understanding how they behave in their actual native context. Okay. Another exciting outcome is improved gene annotation, and I'm sure this would be of interest to the audience here, working on genomics and genomic sequencing and genomic analysis. So genomic language models can distinguish between coding and non-coding regions more accurately than traditional rule-based methods. They also refine predictions of the translation sites, which is very important in the case of microbial genomes, and help identify regulatory sequences. So this improves our ability to annotate newly sequenced genome, especially when we are dealing with sequences that lack prior knowledge. Those of you who have done this or know about this, you know that in order to annotate genomes or even predict genes, you must use related genomes. So genomic language models are getting us closer to being able to do this without, more and more in the areas where we still lack prior knowledge about the particular genome we're analyzing. Classical tools like Prodigal, GeneMark, Glimmer, they all use statistical or homology-based methods. While effective, they often misidentify coding sequences or start sites, especially in high GC genomes. This is why I want to use this as a case study, because not only it's very important, but it's something that we've already published as our first addition to these genomic languages. So with these ideas in action, let's look at microbial genomes, where gene prediction is very crucial for studying things like metabolisms and evolution. All right, so as we all know, bacteria store their genetic information DNA, which made out of the 4 bases. Each unique sequence of these bases form the genes that control different cellular function. Within this gene, there are two major parts: the codon itself, which we refer to CDS, codon regions, and the translation initiation sites, TIS. So for those who deal with eukaryotes, this may not seem as a problem because it's a single codon, and then you have less to choose from. But anybody who's done this for bacterial genome know that you have many to choose from. And so how does the ribosome pick the right TIS? This still remains a mystery. Now genomic language models have really got us much closer than before as to picking the right TIS, as I will present in the next few slides. So the CDS determines the actual structure of the protein that will be made, while the TIS marks the correct starting point where the translation happens. So here is, we can see with the start and the stop, you end up with the CDS. And then from that, once we have the CDS, then we have to predict for that particular CDS, which is the correct start point. And then we end up with the correct open reading frame that codes for gene. Now, let's look at, okay. So the idea here is our genomic language model, GeneLM, approaches this a bit differently by treating bacterial DNA as a language, okay, instead of the traditional what Prodigal and GeneMark and all the existing tools use. We are obviously using genomic language models or leveraging them. For this purpose, we learned the contextual rule that defines the coding sequences. What makes this a coding sequence, and also what makes the translation initiation sites, leading to a much higher accuracy across species, as we will see in the results. This shows that the real-world power of treating biology through the lens of AI is very important, and it's the future to come. Here is a quick overview of the framework. We conduct two main classifications, two separate ones, one for codon regions and one for the TIS. This serves us as to reducing, first selecting the correct open reading frames, and out of there, then we can select which TIS. So there are two different classifiers, all based on genomic language models. So there are two different models, one for each step. The model is trained on large experimentally verified bacterial data sets that we have collected and evaluated on unseen genomes. This workflow really mirrors the natural language processing pipelines. The BERT architecture, for example, you have the chickenization, the masking, the embedding visualization, but here we're applying it to DNA. And I'll explain the core aspect in a minute. So this is the core aspect of that big framework. So I'll just give a little detail just for those who are interested to just know a little bit how this happens or how do we train these models. So let's take a look to understand how a transformer-based model process DNA sequence, specifically through the self-attention mechanism. On this slide, we can see how each DNA fragment or chimer, like ATCGA or TCGA-CAT, instead is treated now as a token, similar to a word in a sentence. The model learns to represent each of these as a vector. We call them embeddings, if you remember me say that before. On the left, we start with the query matrix, shown as an N by D vector, where N is the number of tokens and D is the embedding dimension. At the top, we have the key matrix transposed so that it becomes a D by N vector. When we multiply the query by the key transpose, we get an N by N matrix. This represents the attention score between all token pairs. Okay, and these scores tell the model how much one part of the sequence should pay attention to another part of the sequence elsewhere, anywhere within your training. For instance, regulatory motif near the start of the sequence might influence how a site at the end behaves. Okay, so this is how we end up learning that. Next, we apply what's called a soft max function to normalize these scores into probabilities. Shown here, for example, in the middle as the yellow boxes with weights like 0.4, 0.2, 0.05, and so on. These probabilities are then used to weigh the value matrix shown on the right. Each token's final embedding, called the output embedding, is a weighted sum of all the other tokens' value vectors. In simple terms, for those who are not following me too much, this mechanism allows the model to learn relationships and dependencies across entire sequence, no matter how far apart these bases are. So that's how transformers can capture the contextual grammar of DNA, just as language models understand the relationship between words in a sentence. So our experimental setup for our particular case of gene prediction or for gene LM, again, we built two different models. For CDS, we classified it on Tubqaz supercomputing using two of the NVIDIA A100s with a batch size of 512. Using a distributed training system, it took 37 hours for 2 epochs. For the TIS, the data is a little bit less, so we were able to just run that directly on our bioinformatics lab cluster in our lab. On one node training using a single A5000 GPU of a batch size 768 base pairs, 3 epochs took about 9 hours. Now let's look at the benchmark. When we benchmarked against traditional tools, we can see that GeneLM achieved consistently higher precision and F1 score. What I am showing here is the comparison on verified annotation, means that these annotations were verified in the lab. Okay, we are not comparing. We have other comparisons. You can see the paper, but what we are showing here is comparison with things that are verified in lab, because it would make no sense comparing with what's annotated, what's on the NCBI, for example, because all of that is built or done by software, by GeneLM, by GeneMark or by Prodigal and so forth. What we are actually comparing here is how GeneLM compares to all of these well-known tools on verified genes that had their TIS verified in wet lab. It's generalized. We could see that our model generalized well to completely new microbial species, demonstrating robustness beyond the training data, and that's a significant step towards genome annotation performance. If we have a better gene prediction, of course, our annotation would be much better. So Evo is one of the well-known language models out there recently, because this is again these models come up every few weeks. So we had to really, as we are publishing our paper, we were forced to compare to these guys who only had a preprint. But they happen to actually have one of the best models. They've used the largest amount of data available to train. And what I'm showing you here is a comparison. So the top lines are what the activations of, or the feature activations of Evo, and the smaller lines are actually our gene prediction of GeneLM. Of course, Evo doesn't predict genes, but we're all learning. It learns the signals. Okay, so we gave it the genomes that we're testing, and we see, give us your activations, and we could see that their activations with our activations of models align. Okay, so that's just gave us a confidence in our in GeneLM. Of course, we still made GeneLM because Evo is not a gene prediction language model. It's a generalized language model, but we're just viewing their activation just to compare to see are we also learning similar patterns as they are. And for our particular for this particular example I'm showing here for E. coli, you can see that we are aligning The first line is the full genome, and then we'll zoom in to different windows in the next two lines. And you can see when there is a gap, we have a gap. So we are actually matching the activations. So we're learning the same way. This interpretability is often challenging in deep learning. So it's not just giving me the model, but what's most important is learning. So we use the attention-based visualization to peek inside this model to see, are we really learning something? So what we did here is we're plotting for TIS, for the TIS model. We are plotting the activation, the attention activations. And you can see that the red lines are the red vertical lines are the possible TIS, and you can see before the correct TIS, there's always some signal, which is what the bottom at the very bottom. You see that there is some signal there about a certain number of nucleotides before. This could be indicative of any of the motifs, Chandel-Garno or any other motif. Tata box, different motifs. This is why traditional language model, traditional tools don't always give us a good TIS, because these motifs are not exactly the same among all the species. But for a genomic language model like GeneLM, we were able to pick up on these signals and be able to generalize this. So these motifs align with known biological patterns. In order to see whether we learned this really or this happened just by chance, we did something else. We did what's called a disruption. So we artificially disrupted the motifs. So we went to these motifs and we disrupted them. We changed nucleotide or 2 or 3, and then we said, or changed the location, move it from a 15 base pair to 20 or something like that, and we wanted to confirm that it wasn't. Our model wasn't just guessing randomly, but it was actually had truly learned the biological relevant signals. And then you can see that the accuracy dropped dramatically when we introduced these disruptions. So GeneLM, the models in GeneLM actually learned the biological relevant signals related to the TIS prediction problem. To wrap up, genomic language models are a powerful bridge between raw DNA sequence and biological function. They enable us to interpret, predict, even design DNAs with unprecedented precision. Of course, challenges remain, including interpretability, computational scalability. So these are very computationally powerful, require a lot of computation, and a bias of the genomic data. So we all, especially in the case of disease, because we have more data for diseased patients than we do non-diseased people. So that bias is going to be represented in these models. So this field thrives on collaboration between computer scientists, bioinformaticians, and biologists. And it's an exciting time to be working at this intersection. Very happy that I am there with many others of our colleagues and students. Thank you very much for your attention. If you'd like to learn more or discuss, don't feel free to reach out. You can also check out our website to see what else we're working on. Thank you.
Thank you very much, Professor Al-Ali, for this nice presentation about GLM and the importance of using AI in genomics. This is an emerging field that could bring several solutions to protect the One Health approach. So the simplest example is here. If we use your GLM model to annotate pathogens well, so we will contribute to identifying and contributing in preserving the One Health approach. So thank you very much, and I hope that you will stay with us for the QA session.
Sure.
So now we will take questions for our esteemed speakers. If you have any question, and also for remote audience, if you have any question, feel free to ask.
Oh, we have a question online. Shall we go ahead, Slamin?
Yeah.
Okay, yeah. So we have a question from Othman. He's saying that very interesting presentation. I think this is more to Professor Ashraf. He said he may not be so familiar with LLMs, but he has a question. When dealing with underexplored species, he had wondered if these GLMs must be customized to consider specific information, just in the case, for example, of plant species with highly repetitive content. Ashraf, do you want to go for this?
Sure. Well, GeneLM is actually for bacterial genome, but we have proven the generalization of this. We have even shown that it actually learns the proper signals. Now we are not just us and many other labs are working on different models for different aspects, different species, different genera, and so forth, and different problem, like the ones I've shown prior at the beginning. So yes, they will generalize. We have, just like they pick up in natural language processing, they don't necessarily memorize a particular thing versus the other. We've seen a similar approach for DNA sequences. They do not. They're able to pick up the signals instead of memorizing specific small patterns that are underrepresented.
Yeah, thank you, Professor Ashraf. So I think that's it from the on-site participant. Over to you, Slamin.
I actually have a question for Dr. Alali also. I would love to inquire about the eventual objective of such genomic learning language models, what eventually can be proven or what can eventually be achieved through the exhaustive training of such models. Thank you.
Yeah, so what we'd all like to know is the code of life. We know a lot of, we know actually, for example, let me just pick on GeneLM. We know how the gene, the transcription and translation happens. The ribosome comes in and we know what the signals are, but we don't know in all the cases. We don't have a general understanding of a single mechanism or set of mechanisms. This is the only way it happens. There's always different things that we don't know, that are still under being found, that we're still finding out. Same goes for a lot of other fields. So genomic language models would actually, the more data we feed it, we're going to be able to decode, how DNA actually works, how does the cell use the genome in general, all the aspects. For us particularly, we are trying to build a one-stop place where especially for those who are in the environmental, for metagenomics, we are building a one-stop place where you will be talking to this language model in pure English, giving it all your inputs and what you want to do, and it's able to process everything for you. And we're making some big head starts here with my students and my colleagues.
I have a question for you, Professor Alali. I want to use your model.
Yes, it's the model is available on GitHub. Also, we are working because it uses GPUs, so we can't really share it like we share our other models online. All of our tools are available online, and you can run it on our cluster here in our lab. This one is particularly expensive when it comes to time consuming. We are going to make it available soon as we set up how we're going to work out the logistics, but you can download it. It takes 5 minutes to set up and run.
Yeah, I can use it in my laptop.
Of course, yes. If you have like a GPU that comes with your even on a 8 gig GPU, 10 gig GPU should be, which is graphic cards have that. You should be able to use it.
Okay, thank you.
It'll just take longer.
Any other question? I have a question for Professor Rashid. I didn't see in your equipment long read except INT. So how to to tackle big genomes using ONT is all we know that ONT cannot really give high quality sequences.
Well, thank you. You're very right. But again, this is something. What to answer you right out, we are investing in another third generation technology. However, the way we want to equip the lab is to invest in capital equipment that's going to be used between multiple users. And as you know, for third generation sequencing, it's mostly de novo. I mean, yes, you can use it also for analysis of specific diseases, especially that it has been shown to give higher results compared to next generation sequencing. But in order to equip our laboratory with a sequencer or any other kind of equipment, we have to make sure that it is going to be used by a large number of users, and hence it has to be multidisciplinary. Until now, we did not receive a lot of demand for de novo sequencing because, as you know, you use it from time to time for specific species and then it fades away. So that's the reason that has been delaying us from purchasing the third generation sequencer. But that's definitely coming.
I have another question for you again. The problem of Africa is not the equipment. The problem of Africa is the consumables and the delay of delivering consumables. So how you will manage this for you and for other core labs who are in Africa?
Well, I disagree. It's both. It's also the equipment. Now, if you check, for instance, how many NovaSeq X Pluses we have in the whole continent, you will find that we have only a few. Very few, and the core lab was the second in the continent to purchase a NovaSeq X Plus. And if you check what's possible based on the grants and the capacity to perform any population genomics, whether it's related to a plant or to a species, animal species, or to human precision medicine, you will always find that you need a population genomics equipment, which is a NovaSeq X Plus. We were the second to buy it in Africa, and until now there are only a few pieces in Africa. So it is the equipment, but it's also the infrastructure that should come with the equipment. It's the hands, the trained hands, it's the fence as well. So it's multiple pieces of the puzzle that should all come together. For the procurement of the consumables, this is something that goes beyond us. We do what we can. We deal directly with the providers to cut the prices as much as possible. We did the registration and the importation and all that we do it ourselves to do what we have to do and to solve our part of the equation. But that goes beyond. There is a regulatory framework that has to be worked, not at the level of the core labs, but instead at the level of the countries, African countries, that should probably just realize the importance of simplifying procedures for importation, specifically for science, because it's very different from everything else that is related to trade.
Yeah, sorry, Sulaiman, I have a question. Can I jump in?
Yeah, go ahead.
Yeah, sorry. My name is Tango Ebenezer, and I think thank you, Professor Rashid. While you were talking, I begin to see the passion you have for this as well, and so I think I'm getting multiple screen. I'm not sure why it's happening. So my question is, there is also another puzzle, which is I think Sulaiman mentioned about the problem not being equipment, and I think you also did mention that it's both equipment and the consumables. But there's a third puzzle or element, which is how do we provoke assimilation and uptake, or actually provoke people to use the infrastructure? And I think I've seen that a lot in Africa, where even though the infrastructure is there, the consumable processes are simplified. How do we enable the population to still use the facilities there? And instead of still sending it outside the country. And I think that is also one thing I would like to hear from you on how that could, or in terms of the activities. For instance, one way we are doing it in AfricaBP is to kind of have our mantra is sequence it locally. In fact, we also encourage sequencing it in the country where the sample is gotten from. That way, we're able to maximize the infrastructure there and not send it out of the country. So that's one. I would like to hear from you how that is, your thought process on that. Then the second thing is, in addition to short read and also the ONT, there's also another technique we do, which is called the high C, which you may already know. There's also the Omni-C or the, on other aspects, like for instance, a POC. So is this a technique as well that the core lab has considered? Because in order to get the full-scale telemo to telemo, T to T genome assembly, or even high chromosome scale assembly, we often have, for instance, the short read, the long read, and ultimately, of course, doing like the high C. So is this also a technique that the core labs also integrate as part of its process?
Right, I'll answer thank you for very, very interesting questions. Well, in answer to your first question, there are three things that we need to do. Training, and then training. This is all we need to do to make sure that the equipment is used. We have invested in, I can name in the core lab, 3 equipment at the biosciences core lab, not the others, that are underused, mainly because the students do not know exactly how to use them. Now there are of course the professors, but then the students are really the power of use behind the experiments. So what we need to do, and this is something that probably we should work on together to make sure that students are perfectly trained to. Now, for sequencing, yes, students know, even if they don't know how to sequence, they know what sequencing is used for. But sometimes, if you compare the use of a digital PCR with the use of conventional PCR or QRT PCR, we find that students always know how to use QRT PCR, but they will be very reluctant from using digital PCR. So we have to train the students first. Other levels of researchers, but students, in my opinion, is the key. Training students is the key. In answer to your second question, yes, we are injecting other techniques, not only high-C or Omni-C, we're also injecting other techniques. We have just started the inauguration. Official inauguration did not take place yet. It's for the 4th of December, and maybe a few people here will be officially invited. But we have started very recently, and now we're building the core first. We want to be able to streamline and continuously improve over time. But we are definitely including the Omni-C, high-C, and other technologies like single cell and spatial transcriptomics very soon within our portfolio.
Thank you, Professor Rashid. Is there any question? Yeah. Go ahead, Professor Bobby.
Yeah. I have 2 small questions, one for Professor Walid. Professor Walid, I would like to ask you for some strains of the algae. You found that it was possible to identify the algae using genetics, DNA barcode, for your 3 markers. For others, at the opposite, it was possible to identify them phenotypically, but not genetically. How you explain that we can identify something phenotypically, but not genetically? It's a problem of the laboratory molecular biology amplifying, or it's another problem?
I would assume it has to do with the database. So, I mean, we did actually manual search for some of the species that were not identified in the fresh algae, for pedestrians, for example. This is something that's obvious. You cannot mistake it by the microscope. You cannot mistake it. So we it did not match. We did not find when we did any of the 3 markers. That's, I think, the limitation of the database. Even though we found some entry with the same genus, but it did not pick. We proposed different reasons. Could be the similarity between the variant that we have versus the variant that in the database. That could be some sort of a limitation. Within the range of variant within this genus can range from one region to another, from one marker to another. For example, 5S is very conservative, while 18S relatively diverse among different species within the same genera or so on. So I mean, we came up with different hypotheses, but I'm assuming it's mostly the database.
that could be a strain that doesn't exist in the database, and maybe you are the first to identify it, so you have to go further to sequence.
Could be. That's
see.
one of
Yeah.
that's one of the possibility, but it's not that.
Yeah.
Yeah, that could be one of the possibilities.
Thank you. Thank you. I have just small questions for Ashraf. I just want the feedback of Ashraf, because yes, I understand applying EI on coding sequence or promoters or initiation signals, but is it possible to apply it for non-coding DNA? I mean, what we name junk DNA, knowing, because as far as I understand, it is possible to get information by EI if the process got reasons behind. I mean, if we speak about the languages, the words are there for some reasons. And if we speak about the coding regions, it's the same. But if we apply that to the junk DNA, knowing the evolutionary history of random mutation insertions and translocations and all, could we hope using EI to identify some information behind, or could the EI algorithm learn something useful by if we train it on junk DNA? Thank you.
Yes, I'd like to say yes for sure, but other colleagues might. This is the same debate on the NLP, on the natural language processing world, where we are now asking, can genomic language, or can language models learn to think? So precisely you're trying to say, can our genomic language find things that don't necessarily have any, that are not there yet, that we don't understand yet? So you'll have colleagues in the NLP part that tells you both ways. I happen to subscribe to the fact that I think we are going to be able to find to use this for for non-coding regions and to learn a lot of things that we don't know yet, particularly because what we don't know is still large enough than compared to what we know. And I think this is why I think we might be more, genomic language models might benefit more from language models than the other way around, because we created the language and we created all the mathematical thinkings and all of that. So that part to see if a language model will be able to think and prove mathematical, it's some people are debating, but it's a little bit still not clear. But for me, in the genomic language models, we still don't know a lot of things. So just because we call it junk DNA does not mean it's junk. We all know it. And so I think it will find things that we don't know yet, at least. Now, is it going to find something that is not even following any standard? I think that too, but we're still far away.
Okay, yeah, thank you.
A question, I wrote it on the...
I mean, just to make a paralogy, if we give to the model a random event, is he able at the end, when he converts, to say that this is a random event? There's no pattern.
Yeah, so that's what we did actually with our, just to prove what we did to our models is we actually randomized the input and seeing that we dropped the activation. We don't no longer have those activations. Even by just changing the same signal, we moved it 3 to the right or 3 to the left, and it's no longer detecting it. So, so it's learning biological signals. It's not, it's able to, and when we gave it random, it's like even a random that's very close to the reality. The problem why I chose TIS, for example, as my first example of building a genomic language model is it's very hard for regular machine learning because they share the same sequences. If you're trying to choose from this start codon and this start codon and this start codon, they're very close to each other. They share a very common sequence, and so it's very hard to pick. And it's able to do that. So it's not learning any if we make any little modification there, it says, okay, that's not the correct TIS. So that's why I chose that particular problem, is to really see, are we learning biological signals or are we just memorizing data? And I think we were able to show it. Thank you.
Just a question or comment for the Professor Walid regarding the part of the enrichment. media, if we want to run, for example, a metagenomic approach, does not introduce any biases, this enrichment media? For example, enhancing some population, some species rather than another.
Actually, the enrichment itself is actually driving the population into different dynamic, not representing the natural one. So it depends on the media composition. You might enrich for certain species over another. I did not present here, but we were monitoring the dynamic of the population throughout the 14 days. And you can see, for example, day 3, you have certain species become relatively dominant, then they decline later on. So it is very quite dynamic. Yeah, you assume that when you do the 16S or the metagenome, you still have all the DNA, even the population dropped or it completely declined. But it's not giving you the dynamic. So it depends on the enrichment condition. I think you will see different responses. And that we have seen that already. Some species were not surviving at all in certain media compared to the others. But integrating multiple media will improve the chances to recover as much as possible. All right.
Okay. Any other question? Okay. I think it's okay. So thank you very much again for your nice presentations and then and now we will go directly to the next session. Thank you very much again for our estimated speakers. Thank you very much, Professor Al-Lali. And now we will go directly for the next session before going to the break.
انت عندك معطينا غير إيريك كتعرف تدوز جوج صافي واحد بون صافي
So hello everyone. So now we will jump to the next presentation. So it will be done by Professor Eric Jarvis. So Eric Jarvis is an eminent scientist. And his new laboratory investigates vocal learning in songbirds and other animals as a model of understanding spoken language in humans. So the laboratory, which Eric Jarvis integrated in 2016, integrates computational behavior, physiological and molecular techniques to explore the neural genetics of vocal learning. Eric's did a lot of works, he's very productive scientist. So in the recent year, the interest of the professor in songbird learning has expanded into the parallel pursuit of genomics. So as co-leader of an avian genomics consortium, consisting of more than 200 scientists in 20 countries. He oversaw the sequencing of genomes from a representative species of every avian order, in the total 48 genomes in all. So please welcome all Professor Eric Jarvis for his presentation. You have the floor, Professor.
Thank you. Appreciate that introduction. Can I be heard?
Yes, please go ahead, Eric.
Yeah.
Okay, great. I'm going to share my screen to see if that works as well. All right, can my presentation be seen?
Yes, please. It needs to go on slide mode.
Okay. All right, is it fully in slide mode now?
Yes, yes. Please go ahead.
All right. All right, fantastic. I want to apologize for not being available earlier today. There was a mix-up in checking up on emails. And I'm glad to the organizers for being able to fit me in later today for this presentation. I also wish I was there, and I'll make it to Morocco and other places in Africa at some point in the near future, but I'm glad with technology I'm able to communicate from a distance. I will present actually even a broader topic than what I put in the title, which originally had the Vertebrate Genomes Project, but I decided to retitle this presentation to about high quality reference genome projects more broadly, including in AfricaBP, and the lessons learned. I have a lot of slides, and in case I don't get through them all in time, I decided to put here a list of milestones that have been achieved over the years since 2019 that's led up to the projects we're working on now. So in 2009, the Genome 10K Consortium formed with the mission to sequence the genomes of eventually 10,000 vertebrate species. That's what 10K stands for, within a 10-year period. That wasn't achieved, but there were other associated projects that had achievements that helped move this along. One was the Avian Phylogenomics Project, which led to the B10K Consortium for 10,000 bird species. And we started with sequencing 48 bird orders in 2014 and had some initial very successful studies. But we realized that the quality of those genomes weren't as good as they should be. And so in 2017, we formed the Genome 10K Consortium or the Vertebrate Genomes Project, that's what VGP stands for, focused on genome quality more than quantity at that time. And this quality focused effort helped set standards that ushered in long-read phase genomes. And we paired up with the Earthbound Genome Project, that's EBP, for all 1.8 million species with the same kind of mission. And the first VGP studies were published in 2021, and I'll show you some of the highlights from them. And even out of that effort came the T2T consortium that eventually produced the first complete human genome, both haplotypes, maternal and paternal chromosomes, in 2022. And eventually, now we have high quality draft pan-genome of human samples from different parts of the world, including in Africa. And as of this year, as I speak, we're now completing the Vertebrate Genomes Project Phase 1 data freeze of 581 species in 2025, including some contributions of genomes from the AfricaBP collaborators. So in case I don't get through all the talk, I wanted to get that message across. And here is some of the motivation that led my involvement into these projects. I study vocal learning, the ability for us humans and several other species to imitate sounds. It's different from auditory learning, which is the ability to understand sounds, including in your pet animals like dogs. They can understand the word sit, but they can't say, okay, you got it, I will sit. Whereas these other vocal learning species, dolphins, bats, elephants, can imitate sounds of their own species, and some, like parrots, can imitate sounds of other species due to vocal production learning. And so dog usually goes woof, and it can learn to woof in different contexts, but not speak. But yet, here, some of our distant relatives, like this parrot, can talk.
That would surely be absurd.
So I'm gonna skip the rest of this video. You can see some of this online, but it's incredible what some animals can do, and others, like this dog, cannot imitate sounds like that parrot can.
Or shake 'em.
And our closest relatives also don't imitate sounds and have speech, but they understand speech. You can teach, like this gorilla Coco from Africa, how to understand speech.
Coughing. That was good. You did a sneeze and then a cough. Excellent.
So what's going on in Coco's brain and not these other species is Coco has the understanding to imitate sound, to understand human speech, but not to produce it. And we found that in the brains of these vocal learning species, like hummingbirds, songbirds, parrots, and non-human primates versus humans, that we humans and these vocal learning birds have specialized regions I colored in yellow and red here for learning how to imitate sounds that you can't find in the brain of a gorilla, or you can't find in the brain of a chicken or a pigeon. And we thought, some argued that it's independently evolved according to this phylogenetic tree, but we needed to know if the tree was really correct. So when this group formed back in 2009, the Genome 10K Consortium by these three guys here, I joined the effort to sequence the genomes of all those bird species that can imitate and that don't imitate, in collaboration with these two guys here, Goji Zhang and Tom Gilbert. And we formed what we call the Avian Phylogenomics Consortium, where we sequence vocal learners in red, their closest relatives according to different trees here in white dots, 48 species total across the bird family tree of orders, with short reads at that time. And we published a special issue in Science where we revised the family tree of birds according to whole genomes. And these are all the rearrangements in the family tree, these blue dashed lines from the old tree we're using to the new one, including a rearrangements of the vocal learners in these red dots here, the songbirds, parrots, and hummingbirds. But they were still separated in the tree. So one would have to argue there was a common origin of vocal learning here when Neo-Aves bird species were speciating, and then there was like 10 losses in many other species. That's very unlikely. So we think there are 3 independent gains, even with this rearrangement of the tree. Using these genomes, we're able to also find, using gene expression profiling and microarrays at the time, gene expression profiles that were similar to the vocal learning regions of songbirds and parrots and hummingbirds to the human speech motor cortex in these 2 yellow boxes here. Each row here is a gene. Blue is downregulation, red is upregulation compared to the surrounding brain areas. And using this approach, we found similarities between songbird and humans that we could not see between chicken and non-human primates. And so we wanted to study these genes here that show you these red and blue signals here. We wanted to study the function of those genes in songbirds and humans with gene manipulation studies. But when we looked at these assemblies of these genes, like SLIT1, it's a gene that controls neural connectivity in the brain, we find they had gaps. They were missing entire genes, incorrectly assembled. Repetitive regions were not assembled at all in some cases, making us miss biologically important information. This was causing months, sometimes years, for students in my lab to clone and reclone genes all over again because the short read assemblies weren't doing it for us. And if you didn't do that, you were unknowingly working with artifactual gene structure, unknowingly making false conclusions about the biology. And around that time in 2017, I was asked to take on leadership of this project, of the Vertebrate Genomes Project. I said under 2 conditions. We make an effort to generate at least one high-quality, error-free, near-gapless, chromosomal level and haplotype phase genome for all 70,000 vertebrate species, not just 10,000. And we utilize those genomes to address fundamental questions in biology, disease, and conservation. And this is the mission we've had since mainly 2017. We had help from Jonas Korlek at PacBio, who helped us test long-read technologies for this effort. We formed an assembly working group that meets every Friday, every week of the year, to talk about the latest technologies for increasing assembly quality. and brings various projects together, like the Vertebrate Genomes Project with the AfricaBP project and EBP and so forth. And a number of you recognize the names here. I won't go over all of them. This is back in 2018, but we have evolved since then. And so some of the initial findings was that long reads are necessary and scaffolding like link reads and high-C data and filling in gaps is necessary to generate high-quality assemblies. And we tested multiple technologies, the long reads, the high-C data, originally optical math data, and so forth, on each species. And we had to combine data from multiple companies, technologies, in order to get a high-quality assembly. And when doing so, we filled in the missing regions of these genes, like the SLIT1 gene. I finally had a complete gene and discovered that the first exon was incorrectly annotated and assembled, and so was the last exon, or one of the last exons. And this caused us to have mistakes in our gene manipulation experiments. We also discovered that as you get closer to the 5' untranslated region of a gene, you have higher GC content and more missing sequence in the short read assemblies compared to the old read assemblies. In our hummingbird vocal learner, 70% of the promoters were missing in front of the protein coding genes using the short read technology, and they were found with the long read technology. And why is that the case? We find that the closer you get to the promoter region in the 5' UTR, the higher GC content those promoters have. And the short read technology has difficulty sequencing through GC-rich regions, but not the long read technologies. And the more you get through the protein coding sequence in the exons, the GC content goes down, and that's why you're able to sequence them with the short read technology. This does not happen with the introns, shown here in the dashed lines. The GC content of the introns is stable with the rest of the genome. It's the protein coding sequence that goes up. And so now with more complete genes, we did experimental tests on these axon guidance molecules in the brain. Normally, we see high levels of these genes in the brain regions controlling speech. And when these genes are high in their content, and the axons from the forebrain go down to the brainstem to control the neurons down here that control the voice, the muscles of the voice, they don't form a direct projection because they get repelled. However, in songbirds and humans, we hypothesize these genes are turned down, and then by turning it down, that allows for the direct connection to form without being repressed. And we did turn down such a gene in the mouse motor cortex for vocal behavior to make it more human-like. And we did get increased connectivity in these red dashed lines here from the mouse speech-like cortex or vocal cortex, shown in higher power here. And these mice are maturing their vocalizations faster as pups from postnatal day 7 to postnatal day 12. Their vocalizations are changing to become more varied, like a songbird would. And we would not be able to do that gene manipulation experiment if we did not have the full-length sequence. We also found genes that were upregulated here in what we call the song nuclei of songbirds when they sing, but not in the surrounding areas of the brain when they move and they hop and they do other behaviors. And we found in these genes that there was haplotype differences in some repetitive regions that explained why in vocal learners you get this upregulated by singing, but not by other movement behavior. And the reason why we didn't get it correct the first time around is because these repetitive regions were being strung together in short read-based assemblies as duplications of a sequence, false gene duplications, when there really are two different haplotypes. The maternal and the paternal chromosomes have divergence here, and it's not two separate genes. It's the same gene, just more diverged between mother and father origin chromosomes. And so putting all this together, Anna Philippi's team realized if you sequence both maternal and paternal chromosomes, but use the mother's DNA and the father's DNA to sort out the reads in the child's DNA, you can assemble them separately into two separate haplotypes to get two complete assemblies of a diploid genome, reducing the percent of false duplications here from 5% down to 1.3% false duplications. And we're even below that today. So I'm going to skip this here. And just to say this approach of using parental DNA and now high-C DNA to separate out the haplotypes of maternal paternal chromosomes, and so you don't get a mixture of haplotypes in each of the chromosomes, but complete separation is now a standard in the field and the best way to go to produce high-quality assemblies and reduce false haplotype duplications. So the biggest lessons we've learned here is that haplotypes are one giant repeat, and sequencing and accuracy needs to be high enough to prevent the false duplications. Read lengths need to be longer than the repeats within the same haplotype, and the sequencing technology needs to be able to get through complex DNA structures like GC-rich regions. Okay, so I have 5 more minutes left, and I'll talk about the future directions here. So where are we today? We set out to sequence 260 species representing roughly 50 vertebrate orders that are divergent from each other roughly by 50 million years or more in time. And we use that criterion because most taxonomists that consider mammals and birds as orders, we found were species that diverged somewhere around 50 million years ago. So we applied this criterion to all vertebrates and get 260 species. And where we are today, as of September 30th, 2025, is we have, well, a little before that, we created a data freeze back in May of 2025 of 581 species, representing 229 of those 260 orders. And the VGP group now is doing 50 studies that will lead to 50 publications or more on these 581 species that we've been generating over the last 8 years. We're still sequencing more species, and as of today, we have 635 that are now complete, shown in orange here. And the questions we're asking is, we'll redo the family tree of vertebrates using entire genomes. We'll do comparative genomics of specialized traits like vocal learning, like I showed you. We'll publish a data resource paper of all 581 species for the community. Deciphering the origin of chromosomes of vertebrates, the genomics of sex determination, conservation genomics, 3D structure from the high-C data, and come up with a universal gene nomenclature of vertebrates using these high-quality assemblies, and more. And we have all the data in a GenomeArc resource. You can go to there and find it and utilize that data for your studies. And there are multiple projects that contributed to this VGP effort. The BAT1K, Paratus, BIRD 10,000, European Reference Genome Atlas. These are phylogenetic-based across the family tree of some group across the globe. Then there are national-based projects, including AfricaBP, Darwin Tree of Life, Minderoo Foundation Australia, in France project in Canada, and also in more projects in Australia, Denmark, and projects that function to fund us for critically endangered species, from Revive and Restore, Colossal, Chan Zuckerberg Institute, Allen Brain Institute, and Howard Hughes. And Google is now funding us for some work of endangered species. And finally, there's the Earth BioGenome Project that's an umbrella to all these projects here. And these other projects have learned lessons from the VGP, like the telomere one that I told you about earlier. I'm not going to go into details for time, but where we're sequencing high-quality genomes from people around the globe to produce a near-complete telomere-to-telomere human genome reference with lessons learned from the Vertebrate Genomes Project. To look at haplotype structure, each one of these lines is a haplotype from an individual person with deletions, insertions, alternate sequences shown in these bubbles here. And this is an ongoing effort. It's showing here immune-related genes, showing the great diversity in immune genes, even in the same person. Maternal and paternal haplotypes are different for this immune family, and they differ in different populations. We're putting all of our tools in the Galaxy pipeline so people from AfricaBP and other consortiums can utilize these assembly approaches like HiFiasm, Verco, using HiFi data, Hi-C data, ONT data, and so forth. So I'm going to stop here and skip these questions since I'm over time, and just thank the funding sources shown here in stars in particular that funded my group to help lead this effort for the Vertebrate Genomes Project. And if there is time for questions, I will take them now. And how do I stop sharing? Here we go.
Yeah.
Okay, there we go.
Yeah.
As Professor Jarvis have to leave, we will take 1 or 2 questions and jump to the next speaker. So please, if you have any questions, go ahead.
Or if anybody wants to type it in the chat as well.
Yeah. So I think, Bobby, I would just I just have a few comments or questions, partly because I think Eric, we've known you for a while, and just for some who don't really know on the history of AfricaBP, the VGP made initial investments in the AfricaBP, many years ago in terms of sequencing genomes locally, and I think that has also kind of helped in terms of other, investment as well. So it has like a chain reaction. And I think, Eric, your first or second slide, maybe perhaps we could update that, VGP also were very instrumental in it. And then we can mention
I will
Africa.
include that update.
Yeah, because when I go back in time 3-4 years ago when we started, where we wanted to sequence locally, and when you look at, for instance, with the group in South Africa and the investment coming up, we could actually kind of reverse back and trace that initial 18 species kind of, which I'm not sure if we did all, but that helped to snowball things into the current state. And I think your second slide needs to be updated for it, right?
Okay.
Yes. So yeah, I think this is also for the context for those who may not know in the history, and that the VGP, in terms of the tools, also Galaxy tools, we have people using it already. And we've also been working with the VGP, some collaborators as well within the AfricaBP for the past, since 2021, I guess.
Yep.
Yeah. So it's not a question, just a comment on it. And so one question for you, Eric, would be, when will we get the next flow version of the VGP tool available? So I know we've been talking about this, and it currently is on Galaxy. But when is the VGP planning a next flow version?
Oh, a Nextflow version.
Yes.
Yes, there is a plan for it before the end of this year.
Okay.
Yeah. But the assembly tools are up there in Galaxy now in not Nextflow. I'm blanking out on which version of the flow it is, but yeah, Nextflow, because the Sanger Institute uses that.
Yes.
And yes. And so we're going to try to get it up working on Galaxy as well.
On Galaxy, yeah. Okay, because I know the current one, which also the team in UNISA, because the UNISA team will also be speaking later today, they use the Galaxy version, and we wanted to also see if there is the Nextflow version from the VGP coming out, because I know the Sanger team uses Nextflow, but I'm not quite sure if it's the same tool set or
it's the same tool set.
Okay, in that case, then the VGP does not need to have its own Nextflow because it's the same tool set.
That's right.
Yeah, exactly. Okay, over to you.
And Mohammed Hussein had his hand up.
Okay, yeah, Mohammed Hussein, I would give you the unmute. Could you please unmute then? Mohammed Hussein, okay, unmute.
Could you hear me now?
Yes, please go ahead.
Thank you, Professor Jarvis, for the wonderful presentation. My name is Mohammed Hussein. I'm a Master of Science student specializing in molecular cancer biology and bioinformatics from Cairo, Egypt. I really appreciate and like your point about improving genome assembly quality. So my first question was, how can high quality reference genome help us better understanding complex diseases like cancer?
Yeah, so using high quality genomes to understand complex diseases would be important because some of these diseases are the result of, let's say, an extra copy of a gene or a partial copy of a gene, and you get a duplication. And the lower quality of the draft quality genomes don't do well with gene duplications. As I mentioned, it's kind of string together haplotypes. So it could higher probability of getting the assembly wrong. The other is if you have a regulatory region that's changing, that might not be sequenced as well in the draft quality genomes. And then finally, for complex diseases that involve multiple genes, you want the sequence to be accurate for all all those genes involved in the disease. And if one of them is wrong, then you might not actually get your final answer about the genetic associations of a complex disease that involves multiple genes.
Okay, so thank you very much for your answer. My second question was about the conversion gene expression between songbird and the human. Do you think similar study could help us find common regulatory mechanism in human brain disorder?
Yes, so some of these genes that we found convergent evolution between songbirds and humans, those same genes we find are, when mutated in humans, causes speech disorders. All right, one of them being the famous FOXP2 gene, another being a gene called ZEB2, which is a transcription factor. And when they're mutated, humans have difficulty learning how to produce speech, or might have a speech delay. So we could actually then utilize songbirds as a model to study how to cure those genes, how to cure the speech disorders before applying gene therapy to humans.
Okay, so I think we need to move to the thing. Yeah, Mohammed, I hope this answered your question so we can move to those on site. And yeah, before we move to those on site, I just want to mention, Eric, I think I actually do use the VGP AfricaBP collaboration as an example on how to kind of practicalize and do those things. And I think maybe at some point we can have this discussion in the future. But for now, maybe we can just go to the on-site participant. Bobby, are you on site? Yeah.
You have a question? Go, please.
So thank you very much for that interesting, very innovative approach. I find it fascinating that you chose verbalism and speech as your platform in order to start your research. So because of that, I would like to understand, are there any other particular traits that would be interesting to start from as a launch pad in terms of analysis? So for example, instead of speech, would we be talking about predation, or some interesting migratory patterns, or some migratory tendencies? For example, some fish have electroreception. Would that be a particular criteria of interest for future sequencing projects? I would love to understand your opinion about that, and thank you.
Yes, so one reason why we were able to bring a lot of people together in this project is because there were a lot of people that wanted to study their favorite trait, but didn't have a high quality genome assembly to get at the underlying genetics. So although I was motivated to sequence a bunch of genomes of species that are vocal learners and their close relatives, other people utilize those genomes to study gain and loss of flight, or longevity as another trait that people are studying. Long-lived species, like parrots, are long-lived. And so these high-quality genomes have been useful for many traits and diseases as well, not just speech disorder, but cancer genetics. Cancer usually involves a lot of rearrangements in the genome, and you can't get at the right structure without these long-read base assemblies for cancer genomics. So there are a lot of traits. Anything you can think of is going to benefit from these high-quality assemblies.
Good. Any other question? I have myself one question, Professor Jarvis. I have two very small questions. The first one is just I like it very much, the history, I mean, that pushed you to look for accurate reference genomes and later for Hi-Fi and later for Hi-C. I mean, there are scientific reasons that pushed you to go there. My question is actually there is a need to a new technology of sequencing to resolve some problems that you are facing now? I mean, is there any other technology that we need to get to answer specific questions?
Even though for some species we have complete genomes, getting there is very difficult. Why? Because the two different competing long-read technologies, Hi-Fi from PacBio and ONT reads from Oxford Nanopore, they complement each other in that one technology sequences one regions of the genome better, the other technology sequences some other regions of the genome better, and you have to combine them together along with Hi-C to get as complete a genome as possible. And it takes a lot of effort, a lot of work. The ONT data does not sequence well for birds, and the Hi-Fi data misses very even higher GC-rich regions that you also find in birds. So those companies or some other company needs to develop enzymes or other technology that gets through difficult to folded up regions of the genome, as well as the reads are got to be longer. Once those two things happen, then I think we're able to produce genomes at scale that is necessary to sequence all the species in Africa.
Thank you. So just last small questions. You would expect that speech would be governed by many genes rather than a bunch of 1 or 2 genes, because evolutionary, I mean, it's a very important trait. So, I mean, any insight in that?
I think doesn't take too many genetic changes to get speech. And what I think is happening, in answer to the prior question from the other questioner, is that I think predators are selecting against evolution of speech. The more varied sounds you make, the more likely you're detected in the wild and eaten. And so I think simple sounds, innate sounds, are being selected on by predation. And if you bring things into the laboratory, animals in the laboratory, over generations, they start to produce more complex vocalizations. And our evidence we have so far of the several hundred genes that we see different in the speech brain areas of humans and songbird brain areas of songbirds are regulated by 6 or so different transcription factors. So I think if you change 6 transcription factors, you're going to get several hundred changes in gene regulation that'll lead to a brain circuit that's similar for speech. And that's what we're trying to do.
Okay. Thank you very much. Thank you very much, Professor.
You're welcome. Good questions.
Okay, I think it's all. We are really grateful, Professor Jarvis, for this presentation and for your presence. Thank you very much.
You're welcome. Thank you.
So before moving to the next session, because we have the persons that is waiting here, we have to take a break of 5 or 10 minutes so we can come to finish the remaining presentations. So please, you are invited to coffee break here for 5 or 10 minutes so we can continue later. Thank you.
BGI Genomics is a leading global provider of a wide variety of omics services, covering genomic sequencing, mass spectrometry, synthetic biology, bioinformatics, and biodata management solutions. We are relentlessly committed to providing the highest quality and reliability of services. In particular, our unique Dr. Tom Multi-Omics interactive analysis platform has already supported countless researchers to publish papers in some of the world's leading journals. We remain dedicated to advancing genomic discovery and leveraging our world-leading experience to help our partners. We are committed to driving discovery even further. BGI Genomics. We sequence, you discover.
Well, I can't believe it's been 10 years since Illumina Biotech West Africa opened its door for business here in Nigeria and in the large West African region. And for over a decade now, we've been at the forefront of driving genomics and life science across the region.
It's been 10 years of excellence. It's been 10 years of hard work. I remember when I joined the company, we started from a very humble space within high ITA.
It feels just like yesterday. I joined INCABA in 2018. Since then, there has been significant growth.
With Central Africa being the most recent region to have a representation for INCABA Biotech, our creation and existence has benefited considerably from experience and mentorship of INCABA Biotech West Africa Limited, Ibadan, Nigeria.
Reflecting on the last 10 years showcases hard work, diligence, tenacity, excellent leadership, and help from God. At INCABA Biotech Limited, our core values are quality, customer focus, integrity, teamwork, and others.
From groundbreaking research to empowering our team and customers through training and development, we've built a culture of continuous growth, while staying true to our mission to facilitate life science research in West and Central Africa.
My name is Emma, customer service representative from PacBio West Africa Limited, Ghana.
My name is Yusuf, the manager from PacBio Ghana.
Good morning, I am Mamadou Dabo from Mali.
I extend our warmest congratulations on the 10th anniversary of PacBio Nigeria branch.
Congratulations to our counterparts and our colleagues in PacBio Nigeria for their 10th anniversary.
Happy 10th anniversary year to all Inkaba Biotech staff in West Africa region.
Happy 10th years anniversary to all of my colleagues from Nigeria, Cameroon, Benin, Mali, and Senegal. We are the West Africa team.
Having provided sequencing services in Nigeria for about 4 years now, we've come to understand the needs of the research space, and as such we see the need for improvement, and in the future we're looking to take up a bit more responsibility in this space.
We've really achieved a lot. There's still a lot more to achieve. I strongly believe that IBOA as a company in Biotech West Africa has really positioned itself to be a major driver that will facilitate the growth of the biotech industry across West Africa.
We are not only celebrating the past, but looking to an even brighter future ahead. We will continue to collaborate, innovate, and make meaningful contributions to Africa and the world at large. Here's to 10 incredible years and an exciting journey ahead. Happy anniversary Inkaba Biotech West Africa.
Long live Inkaba.
This is to many more years of growth and positive impact to science across Africa. Happy 10th anniversary Inkaba Biotech West Africa. Cheers.
As we look ahead, Inkaba Biotech remains committed to driving innovation and technological advancements in the life science and genomic space. We intend to do this by investing heavily in the latest equipment and sequencing technologies, empowering scientists across the continent so they can continue to do the amazing work they do. And very importantly, we also plan to serve new regions within the territory by expanding our tentacles into countries where we currently do not have a presence. So I hope you can join us in creating this amazing journey.
Despite the fact that resources are limited in Africa, the team has steadily developed the enterprise, including the setup of a genome lab that provides summary sequencing services in Badan. And we look forward to add other genomic services like next generation sequencing using biosequencers like we do here in Pretoria. As Inkaba Biotech, we aim to contribute within our means to the science and economic development on the African continent, a continent that has a vast diversity and needs so much more equality. We have created more than 40 jobs at our offices in Nigeria, Ghana, and Senegal, and this will continue as the need for application in life sciences and diagnostic develop in West Africa.
leveraging high-performance computing for large-scale data analysis. Please welcome with me Dr. Ansia Van Coller.
Hi, just to confirm if everyone can hear me.
Yeah, we can hear you. Go ahead, Ansia.
Okay, perfect.
Maybe you
Thank
want to
so much for the.
speak up a bit.
Okay, perfect.
Can you hear me now?
Yes, we can hear you. Go ahead, please.
Okay, perfect. Thank you so much for the opportunity to present to you guys today. And I can tell you a bit more about the genome assembly that we've been doing for indigenous African plant species, Helichrysum odoratusum. Just for a bit of background on the relevance of this plant, so it was named after the Greek words for sun and gold because it has these characteristic golden yellow flowers, and is named odoratusum because of its strong odor. It's quite fragrant. So it's also known as African incense, and in some of our local languages, it's known as koichut or impepo. So this plant grows quite abundantly throughout Southern Africa, and it is one of the most commonly used traditional remedies. And it grows not only in all South African provinces, but also in the surrounding countries, Mozambique, Zimbabwe, Malawi, and even a bit further north. So it is of great cultural significance in South Africa. And it also has a lot of documented medicinal uses. So it's been used for the treatment of heart disease, chest pain. It can have quite a calming effect, I'm told. Haven't tried it. It's been used for the treatment of coughs, fevers, urinary tract infections. So there's lots of studies that suggest that it could have some antimicrobial properties. It has calming effects. It can also be used to treat psychosis, and it is been hypothesized that this could be due to the presence of phytocannabinoids, which has been picked up in some other Helichrysum species. Because of its strong odor, it's also used as an insect repellent and a perfume. It is one of the most frequently harvested plants and traded in South Africa, so it has great potential for commercialization. So despite this cultural and potential economic relevance of the species, there are no fully assembled genomes for any Helichrysum species. And studies have shown lots of variation in the genome size, the chromosome number, and the ploidy level, even within the genus. And it is likely that the polyploidy plays an important role in the diversification of the genomes and allowing broad ecological adaptability, allowing it to grow in all these different climates and environments across Southern Africa. So given that no assembled genome exists for any of the Helichrysum species, we then sought to sequence and assemble the genome of Helichrysum odoratusum using Oxford Nanopore Technology long reads and high throughput chromosome confirmation capture or high-C sequencing. So I'm sure most of you are already familiar, but just for the benefit of those that perhaps are not. So if an organism doesn't have a defined reference genome, then it would need to be constructed in a de novo manner. And this process would rely on tools that align all of the little fragmented reads to each other to identify overlaps in order to assemble a contiguous sequence known as contigs. And then these contigs can be linked together in larger groups through a process known as scaffolding. So genome assembly can be quite computationally intensive and it can take a while to do. But long reads, so those PacBio hi-fi reads or ONT reads are particularly useful for de novo genome assemblies because it allows us to easier resolve some of these repetitive or complex regions or those with structural variations. So we sampled this plant at this beautiful wine estate called Babylon Sturen, right there at the tip of Africa. So I'm quite a ways away from Morocco at the moment. And then we were able to extract some really high quality DNA and RNA from the plant. And then we did ONT sequencing on our Promethone P2 Solo. And the high-C and RNA libraries we sequenced on the MGI platform. So just for a really brief overview of the assembly workflow that I used, using the ONT data, I did the super accurate base calling model to try and improve the overall quality of the reads, and then did the basic QC trimming of the adapters and ensuring we only use the high quality reads for the assembly. With the Hi-C data, I also did some trimming to trim off bad quality data to use for the assembly. And for the assembly, I used a hybrid type assembly where I used both the ONT data and the Hi-C data using HiFiism, which does really good error correction as well. And it even works really well with ONT data. So after a draft genome was generated, I aligned the Hi-C data to the draft genome and pre-processed this to pull out all the relevant Hi-C contacts, which I then used for scaffolding. And this was then followed by manual curation to kind of put the assembly into defined chromosomes. And then lastly, we integrated the RNA-seq data for some annotation. So in terms of ploidy, so as I briefly mentioned earlier, so previous ploidy estimates in the South African population of Helichrysum odoratissimum have shown that there are both diploid and hexaploid individuals, and the smudge plot analysis revealed that our individual is likely hexaploid, and the genome scope tool agreed, as you can see the 6 KMA peaks. So this is a very, very heterozygous genome, very complex, and this already indicated that we might have some difficulties getting a good chromosome level assembly later on. So I used HiFiism integrating the ONT and the Hi-C data, and it outputs a primary assembly as well as 6 different haplotypes. So it is kind of debatable about how accurate and complete these phased assemblies could be. So I focused most of my further efforts on the collapsed or primary assembly. We can see the draft assembly is quite fragmented in terms of, like, there are over 5,000 different contigs. However, they are, some of them are quite large, and the N50 is about 12 megabases. So 50% of the draft genome is made up of contigs that are at least 12 megabases in size. And we got a really good BUSCO score with a 98.4% genome completion. So due to the large number of small contigs, we anticipated that we would really struggle to integrate all of these while scaffolding. And so I pulled out all of the smaller contigs and compared those to known mitochondrial and chloroplast sequences. And then I pulled out all of those that matched so that we could focus on just building a nuclear genome. And you can see here, after filtering, we removed about 2,000 contigs. The length reduced slightly, but the BUSCO score remained the same. So we did not remove any essential eukaryotic or Unicode plant genes. So the next step was then to integrate the Hi-C data. And again, I just want to kind of go through this again. I'm sure it was mentioned a few times today, but for the benefit of those that are not that familiar with Hi-C, so it allows us to capture the chromatin conformation within a nucleus. So DNA is kind of folded up on itself, and because of that, there are regions that are in close proximity to each other that are not necessarily linear. So through the Hi-C technique, we can link these bits of DNA together and do enzymatic digestion and then ligate them together in a process known as proximity ligation. And then essentially you end up with DNA fragments that contain, well, DNA from different regions of the genome, but that have been ligated together. And you can then sequence this as you would any other DNA library. And then when you analyze the data, you would align these little reads back to your draft genome, and then you would see how this side of a fragment interacts with a specific contig, where the other side interacts with a different contig. And that information is really useful to help show us how the close certain regions are to each other. So you can generate these high C contact maps, and you expect to see a really dark line down the diagonal to show how different regions of the genome interacts with other parts. So I just wanted to show the quality of our high C data. So this was the raw data, and our quality on average was about 36, which is really good if you consider that anything above 30 is considered to be excellent quality. And so as far as I'm aware, this is the first time that high C was done on the MGI platform in Africa. Yeah, and we ended up with really good quality data. We could see the good capture of the chromosome interactions with really good resolution. So we could see interactions at various distances on the genome. So after scaffolding, this is the stats of the scaffolded assembly. So we didn't reduce the number of contigs that significantly. That is in part just because of the presence of the high number of really small contigs. But we did improve the N50 quite a lot, and the L50 and L90. I think this is probably my favorite metric when you're looking at a genome assembly. It essentially tells us that 90% of the genome is contained within 24 contigs. So yeah. The next step was then manual curation of the genome. So as I briefly mentioned earlier, so with HiC you can measure the frequency of interactions of DNA fragments in a 3D space. And when you generate a HiC contact map, you might see something like this, where you see a little bit of a hotspot over here, which indicates that this area of the genome interacts with this area, which means this area likely belongs over there. So then follows a process, a manual process at this moment. There are no AI tools available yet to simplify this process completely. You would need to manually break the contigs and rearrange them, split them, and reorder them. So as you can imagine, this is quite a complex process, especially for really complex large genomes. So here is the HiC contact map of our scaffolded assembly. So you can see the line down the diagonal, as well as several little contact bits off to the end. And even when you zoom into one of these contigs, you can see there's still quite a bit of noise around the edges. But you do expect to see a lot more of this type of thing in really complex and repetitive genomes. Because I'm not an expert at manual curation, I had to phone a friend. So Sadiq really helped me to anchor this assembly into 7 really large chromosomes, as well as 3,900 really small scaffolds that you can hardly even see on this diagram. So yeah, this was a very lengthy process due to the big size and of course the complexities based on the ploidy and the repetitive elements of this genome. You know, it can be difficult to know if you see an interaction off to the end, is it because that region of the genome belongs elsewhere, or is it just because that specific sequence was duplicated on a different chromosome, which can also happen, or often happens, in fact, with plant genomes. When they have these whole genome duplication events, you end up with so many copies of same gene or variations of a gene on different chromosomes. We then did some annotation of the genome. So this was just very broad, and I've tried to visualize it here in this little circus plot. You can see how the gene content is spread out across these different chromosomes. So we identified over 340,000 transcripts. So it could be that there are more, because we only did RNA-seq on leaf tissues, but the RNA expression levels would probably look, or the RNA genes, RNA that is detected would look different in different tissue types. We identified quite a lot of repetitive elements, the most significant being, of course, the transposable elements. And I think that's what was giving us such a headache when we were doing the manual curation. So yeah, in conclusion, we were able to construct a really high quality chromosome level genome assembly for Helichrysum aderatissimum with high levels of genome completeness as assessed by Quast and Busco. And the structure was validated using a high-C contract. So this will contribute to conservation and sustainable use of this plant and kind of assist with future studies on bioactive compound discovery, breeding, and further the commercial development of this plant. But in all, this experience also really just demonstrate that high quality genome assemblies can be produced entirely in Africa. So from sampling to sequencing to analysis. And this study really forms the basis of our future chromosome level assemblies that we will be doing from South Africa in partnership with AfricaBP and the 1KSA initiative. So I know I really probably could have spent more time talking about this, but I know we are a bit behind schedule. So I just want to lastly thank everyone part of the team to help make this possible. And please do let me know if you guys have any questions.
Thank you very much, Dr. Ansia, for showing us this successful example of a complete assembly within our dear continent. If you have any questions for Dr. Ansania, please. I have just small questions, Dr. Ansia. I would like to know, using all softwares that you used, how much time it takes for one person to do the assembly from the raw sequences to the last point of the assembly, more or less, I mean, roughly, for trained bioinformatician?
Gosh, for a trained bioinformatician that's used to doing this, it will probably be a lot quicker. But for me, it was a bit more of a learning experience as I went. I'm more from a human genetics background. So this would be the first plant that I was working on. So I was probably a bit slower than I would be when I do the next one. But the speed really also just depends on the computational power that you have. So these genome assembly steps are really intensive in terms of, the amount of CPUs and memory that you require. And if you can throw more of those things at your assembly, then you can speed up the time. But using the capabilities that I have access to now, I would say at least a week of straight computational power. Yeah, it very much depends on the complexity of the genome and the size of the genome. But yeah, each of these little steps can take quite a while.
Great. Thank you very much. Any question, please? I think it's okay. We are really thankful, Dr. Ansia, for this nice presentation. Thank you very much for being with us. Thank you.
Thank you so much.
Olivia, I think. It's Dr. Lucky who will, okay. Okay. Normally we should have Professor Mafoli, but she was not able to be with us. And Dr. Lucky will replace her. Please, you have the floor, Dr. Lucky.
Can you hear me? Am I audible? Hello?
Yes, yes, of course.
Oh, okay. Thank you. And good evening, good afternoon, everyone. Thank you so much. Like I said, I'm not Professor Mapori. I'm just standing on her behalf. She would really love to be here. She was really disturbed that she couldn't make it because of the university graduations proceedings. She is overseeing that part. But I hope I'll do justice on her behalf. I will give a talk that she was supposed to give, which is titled The Genomic Insight into the African Indigenous Livestock. That is aimed to advancing sustainable food security and biodiversity conservation. If we take a look at the Africa's livestock industry, it is an industry that is characterized by many different breeds that account for at least 1/3 of the global livestock population. And mostly the bigger portion is from the cattle. You find sheep, goats. This indigenous breed, they are characterized by different traits of interest. You talk of the traits of being able to adaptability to most of the challenging environments in the continent, and yet they produce efficiently. The importance of the African livestock, it's also emphasized in when you look in the African Agenda 2026, where it was central when we talk about the poverty alleviation and the potential into providing food serve and nutritional security. Even though when mostly when we talk about the cattle space, we normally focus on the beef and milk production, which is deeply embedded in the continent's livestock production system. But however, there are so many other emerging aspects that are coming into place, and it's very exciting to see some of the projects that are from the livestock that are coming to starting to take shape. In most countries, majority of the countries, let's say in most of developing countries per se, livestock it is integral into their development, and also it contributes significantly into the agricultural GDP in most of in the African countries. So, however, though the significance of livestock sector within the continent, it doesn't exist without some of the challenges. It has long tradition ago that there were some problems that we continue to solve. Some can come and emerge, but how do we collectively address those challenges? Some of the common challenges that we have observed is the genetic dilution or genetic dilution risk that is caused by crossbreeding this indigenous breed with exotic breeds in an effort to improve their production, not looking into some of the benefits that if you look at the efficiency, because there's a difference into when you look at an animal production, you also need to consider the input, that how much do you input on the maintaining that animal. The indigenous animals, they have the advantage where they are able to produce efficiently better than exotic breeds. But that not taking away the issue of the importance of crossbreeding is still important, but it needs to be done in a controlled manner. There's one talk that we were discussing about that we really need to also put more effort in how do we develop a structured breeding programs that will address the questions that we have on the ground. Having said this, we have seen some of these uh, exciting researches, but are they addressing the challenges of the farmers on the ground? Those are some of the challenges that we continue to observe in the livestock production space. Also, the other important that it's still quite a challenge. It's the record keeping. It is difficult to, especially from the small scale farming sector. Record keeping is still a challenge. And the other aspect that it's impacting the livestock industry, it's actually something that it, it sounded more like someone was making a joke. I had to verify it, and I found that it is true actually, where you find that in some part of the regions in the continent, they don't eat animals that are black. They prefer animals based on the skin color. And you find that some of the good animals, you talk about them, maybe the Drakensberger, they have a black coat. They are very good breeding the beef cattle. But because of these social-cultural issues, it becomes a challenge as well. So there are those kind of things that are at play that we need to find a way how to deal with that. And don't ask me how how to advise on terms of the social challenges things. It's quite an interesting part, but yeah, it's one of the challenges that we're facing. And looking at these challenges, what are the possible solutions as we talk, as we have these gatherings? What do we need to start thinking so that we do things that help the people on the ground? In terms of, I will talk from the breeding perspective. We ought to have national or continental breeding policies that can benefit it. There's a good study that was done by some of the colleagues. Isido was part of it, where they were talking about the benefit of having multi-country breeding programs. These are the things that we need to start to implement in order to benefit the industry. How do we get the public-private partnerships to work together? It needs to move from the stage of where we have workshops to talk about it and translate it to the working ground so that we see the progress on the ground. Also, the strengthened farmer cooperatives. How do we invest in genetic infrastructure and extensive services? You will recall that in the previous slide, I mentioned that there is this good research, very expensive research that we're doing as scientists in our labs. But how do we make sure that whatever we're doing, it is translated to the people on the ground? How do the people benefit? There is a very element of extension services that people often neglect. That is one of the most important part that we need to invest in, because they are the people who are going to drive all these tools that we're generating and deliver them to the people so that they benefit. Also talking about the community-based breeding programs to address challenges that are strictly facing the people on the ground. Having said that, I gave you all that information so that we understand the perspective, or we grasp the essence of the livestock production challenges and why do we need to invest more, put more effort in order to guard against the food security issues. There lies most of the opportunities within the industry, within these challenges. And currently, we're talking about the climate smart breeding system. We're talking about the digital record, the importance. I talk about the challenges of record keeping, especially in the smallholder sector. Engaging the youth into the getting involved into the smart livestock farming system. They are the future. We need to start to, whatever we're doing, we need to get them involved. Having said that, I will leave that other topic to the politicians now. I'll come back to let me wear another cap of being a scientist now. But when we talk, we also see the need of developing the genomic tools that will help in improving this indigenous livestock system that we have in Africa that are of precious, so to say. This involves generating high quality reference genome, because like majority of African indigenous species, they don't have quality. You talk of animals, any non-human species indigenous in the continent, they don't have high quality, or they very few have high quality reference genomes. So that is where it was decided there was a birth from the AfricaBP, African BioGenome Project. There was a program, the Generation Project, a program that is a Nelson Mandela Genomes Initiative for Conservation of Nature, which specifically aimed to understand the genetic basis of Africa's cultural and economical significant animals and plants genetic resources. The idea behind it was to build genomics capabilities that can directly address the issues or provide the start to order to answer questions such as food security, biodiversity conservation, biotechnology, as well as in terms of plants, the medicinal purposes. In this project, what we have done so far, we have managed to sequence about 34, it's about 37, it's just 37, that is genomes. And 16 of these genomes have only PacBio Hi-Fi, with 21 have both Hi-Fi and Omni-C. I've heard some of the talks there that the importance of in using the benefit of combining both technologies, and like it was exciting to listen some of the previous talks, and I found that it really translates to what I was to the talk that I'm going to give. So in general, most of the genomes that we do, or the genome, the strategy that we use in this genome, we use the approach of sequencing them through Hi-Fi reads, using PacBio, and we have currently, through the partnership of INGAVA, we have bought the PacBio Revio, and we also use the ONZO for Omni-C. Currently, we have also started to utilize to generate the ONT in an effort to get to the T2T genomes. Just to give a bit of a background of some of the genomes that we have done so far, we have done like in terms of a small stock, we talk about the Nguni goat and Nguni sheep, which have, in general, like I said, they have the most economic important traits that are of importance that we need to guard against the diminution of these genetic resources. Majority of them, like when you talk about the Nguni sheep, they are generally known to be well adapted to different agroecological regions. And they are often, I use the word of a breed of choice to the small stock farmers, because of how it's not difficult, it's not expensive compared to exotic breeds to keep them in the challenging environment where we are. That translates as well to the indigenous goat. They also, though they have small frame, multicolored, they're very beautiful. The coat color and glossy, and they're very glossy. Their short hair and coat is very beautiful goat. Exceptional tolerance to harsh and environmental condition. As well, also we have done the Colebrook pig, which is also similar to the important traits. This indigenous species, they are very well adapted to the harsh condition and the disease that are endemic to the specific region. And they are regarded as one of the South African indigenous pig breeds. Just to give a brief of results in terms of the genome scope, I will go through them quickly, just to see that we are currently the genome size is within the expected. We run the genome scope to see, as well as the we assess the BASCO assessment, which in average, I think most of them, they were about 96 to 90. 96, there are some of them we got even 98 completeness in terms of PASCO and assessment. So in terms of the genome assembly, this is the Nguni sheep and how it compares to other genomes that are available there. It is very much comparable. When you look at the genome size is within the range, the scaffolds, we're not that far off. And we actually produced one of the good qualities of the genomes out there. And when you look at the Nguni goat as well, the genome size is within the expected range, and we got in some cases we got the fewer scaffolds. The scaffold N50, it's within the good range, comparable with the available genome. as well as the Coolbrook, which generated the genome size. It's also within the range, and some of the assembly statistics, it's actually we got the better side of things. Our assembly were quite good. In terms of larger stock, in terms of cattle, we did the Nguni cattle, the three ecotypes. The Nguni cattle have different ecotypes, so we did the three ecotypes, which will be interesting actually to do the genomic comparison going forward, and to looking at that. But sometimes they do look similar, but they are different. When we're talking about the Nguni sheep, how did we can see how they differ with the Nguni Venda, the Nguni Shangan, but in most aspects of the traits of importance, they are all adapted to this challenging environment that we're talking about, and they are tolerant to some of the problematic diseases. You talk of tick-borne diseases. They are tolerant to such problematic and strong disease resistance, and excellent adaptation to this environmental condition. But one interesting part that I looked at, there's a study that actually reported that these ecotypes, in terms of frame size, or body weight, in terms of winning weight, in terms, though other weights, they were not significant different. But when they were comparing the winning weight of this ecotype, they found that they perform differently. They are significantly different. So it was quite an interesting study. And it should be interesting to go further and explore more on the genetic side of these ecotypes. We also looked into the Bosmara cattle, the Tuli cattle, the Drakensberger cattle. Like I said, the Drakensberger cattle, they're generally characterized as black. So in some part of the region in the continent, they might not consume them because of the color of the coat, which is interesting. So the majority of them, they have similar traits of being well adapted and tolerant to some of the problematic conditions and the diseases that we face in the continent. In terms of the genome scope, just to look also on the genome size, we also find the genome size are within range of all the cattle that we did. And for Drakensberger, I forgot to mention that for Drakensberger, we used the trio approach, and we also recently added the ONT. Previously, we didn't add the ONT data on the Drakensberger. This is the genome scope of the Nguni ecotypes, which is the Venda, Pedi, and Shangan. And in terms of PASCO assessment, we also generated most of them, they were above 96 completeness and above. In terms of the summary statistics of the genome assessment, just to give you, and the last side, the last far right, on the genome assembly is the official bovine cattle reference genome, which we compared with what we have generated, and I think we have done justice, and we're standing at a good position in the we have good genomes compared to the available genome reference. In terms of, we look at the genome sizes within the range, the scaffolds, we actually did much better. The scaffold N50, some of them, we did much better than compared what is out there, the contig numbers, as well as you look. And also in some cases, we also employ the strategy that we go further in terms to annotate what the genomes. These are the average number of genes that we found, and the mean length, as well as the exon per gene, and some of the things I'll just quickly do to the time. We do have quite a number of genomes that are other genomes that we are currently working on, or some of them we have, like I said, we have completed 37 so far. We have quite a number of the samples that are still going to be processed, hoping now that we should, in the coming month, we should be able to produce quite more, because we do have our own target of at least reaching 100 before the end of December. I'm still hopeful. I know it's quite late, but with the number of samples that we have, we should be at least have able to cover a significant portion of that target. Yeah, some of the we have started to publish some of the Nguni ship. It's available. Some of the papers, the Nguni goat has recently been accepted. The Colebrook is under review. The indigenous chicken, it's also under review. As well as the tilapia fish, and we also working with the. I mean, it also benefit to, because the other idea is to make these genomes available to everyone at the end of the day. Though we might have had some, as this was a pilot project, we might have had some bureaucratic challenges at the beginning, but I think we are starting to find our food in terms of making these genomes available and accessible to everyone, which it should be an exciting story. And that will benefit the Africans in terms of having access to these genomes. So we are also working with the bovine pangenome consortium, contributing some of the genomes. And like Erich said earlier, that we're also working with the VGP through, but like I said, it's a child of AfricaBP project. So I would like to thank everyone, and I will gladly take questions on behalf of Prof. Mapodi.
Thank you very much, Dr. Lucky. So, Dr. Lucky, you gave us a very good presentation about the approach for breeding for livestock in Africa. And we are very happy to hear about other stories, success stories of complete sequencing of genomes within Africa, from South Africa. If you have any question for Dr. Lucky, I think I have one. Dr. Lucky, I would like to ask you, of course, abstraction that of all that you presented, which is very, very interesting in what should be done. But my question is this: as you know, each country needs to produce maybe more than to conserve species. At least in the short term. So how we can can combine about having intensive breeding programs to produce milk and meat and all, and to have another programs to conserve species, our species, that most likely are less productive. How we can deal with this problem.
Okay, thank you for that question. Well, I don't know where to start. It is one of the questions that the importance part of it, we do have animals that are not performing well, and we see the benefits of crossbreeding in terms of improving, in terms of milk yield, because we know that one of the questions that we were actually talking about, I think it was a month or so ago, it was specifically in terms of milk yield. How do we because we are comparing our indigenous cattle breeds with the exotic breeds in terms of milk production. We want to improve this milk production in the continent. And one of the strategy that is being used currently is through crossbreeding. And this crossbreeding is also bringing its own challenges through the dilution of the genetic resources. But we need to have a controlled crossbreeding programs that we do not completely lose the indigenous genetic material. But also, I feel it's also unfair to put the production of milk of the exotic breeds that went over 1,000 of years of intensive selection programs, where they from to be where they are today. Our indigenous animals, they never went through that. So we also need to have, how do we put the programs that will also within the same in the breeding within the same breeds. How do we improve them genetically? That is where now we have to talk about the multi-country breeding programs. How do we put breeding policies that will benefit our resources without compromising them?
Thank you very much, Dr. Laki. Thank you very much again, and my greeting to you and to Professor Mafoli. Thank you very much.
Thank you so much, and I wish I was there. I've never been to Morocco, but next time,
inshallah.
Thank you so much.
A good day. So we reached the end of the first day of regional workshop for North Africa 2025. So hope to see you tomorrow for the next day of conferences. Thank you very much for your presence. Thank you.